need of the hour dictates that we all work together to protect the environment and educate people about its importance. In this context, the present focus on environmental education is not an entirely novel concept, rather, it is an age old forgotten philosophy of social and cultural practices of many civilizations. The Indian civilization had ashrams and gurukuls as epicenter of its ancient education system. They were located. Sorry, this is speaking. कैसे रिटर्न हो रहा है? The Indian civilization has ashrams and gurukuls as epicenter of its ancient education. They were located in lap of Mother Nature, where sensitiveness towards the environment was not only taught but also embodied in the everyday practices of the people and was embedded in their routine life and was religiously practiced. So, so this one uh, is central idea of the theme that we are going to focus on this conference. Now, I would like to call upon Professor Balram Singh. Professor Balram Singh is a renowned scientist and he has been eminent. He has been professor Department of Chemistry and Biochemistry and Biology at University of Massachusetts Dartmouth. Currently, he is founder of Indic Indo-Vedic philosophy. I request Professor Balram Singh to address the keynote as a keynote speaker to the audience. Professor Balram Singh.
Thank you very much, Professor Rakesh Kumar ji.
इसको क्यों रिवर्ट कर रहे हो?
I'm sharing my slides if that is okay. Okay. Is it okay to share my slides? Can you hear me?
Yes, sir.
Okay. Thank you very much. This is a great opportunity to connect with my alma mater. Obviously, I graduated with my undergraduate degree from Kamla Nehru Institute of Awadh University. And it is always great pleasure to connect. Now, normally I visit my hometown every 2 months, but because of Corona, I've been stuck here for the last 4-5 months. But,
yes, sir.
But this is a great topic. I think it's really, I'm very pleased that this is a topic that is being discussed by institutions like Kamla Nehru Institute. I want to place this in a form where you can see how the modern science has evolved in the first place. How did that start? And that started modern science, foundation of modern science is the European Renaissance. It was a appropriate topic that you have to get physical, biological, social sciences together. But that's not only, I think reality that's what it is. I think we are not divided into physical sciences and biological sciences, social sciences, or any other knowledge system. Knowledge is all continuous integrated. So it's great that you have been able to put your topic in such a way we can discuss all this together. And the European saw, which was in the 16th century, was actually a movement of art and social change. So that also makes a very good sense to have that. And my career is guide. Okay, I'm speaking all right. And the person was so talented and was involved in so many ways was this man Leonardo da Vinci, who is actually known as father of modern science. He painted most of the stuff. He was a painter, a brilliant painter that is, and he actually painted a lot of scientific things, anatomy particularly. So in this example that he drew the thyroid an example. This is an example. He made many anatomical things. So it's important to note that I would say that India is at the cusp, especially in view of Corona. There is a huge social change, huge scientific realization that we are very limited. And so I think it will be a perfect timing for India, a place like India, to get involved in this. Unfortunately, one thing that they don't have is time, because things in 15th, 14th, 16th century, things were very slow. In 15th century, the first time people had books, and so it made huge difference. It was a great equalizer to people, and that helped move things fast. But now today, the world is very competitive, and this internet technology that is allowing us to be here to talk to each other, even though thousands of miles away, I think that is going to be a competition, and there are competitions of ideology, there are competitions of interest, there is a competition of arrogance, all kinds of things are going to be there. So I think India with its philosophical treasure should be rapidly in this area, and particularly they have a huge advantage of young population. Their population nearly 60% population is young, in the world. And if they do it, and which if they do right thing, this would be able to do well. So I would say it's important that we look at it from that perspective. But I want to show you just more history, and that is I want to show that scientific progress the Earth in in AD, it was Earth is the one which and everything is moving. That's what you think about it. The Earth is flat. But Copernicus in the things that we was able to say and Kepler was talking about the ellipse. And Galileo, most of the problem with that, this is a very important thing when we talk about culture and other things, I think we need to think about how that is going to be taken, who's controlling how much. And even though India might be very, very strong culture of science in its philosophies, but that what we are practicing today is question. So there's something like Newton able to come out, and even though he was a very strong person, and he really created that whatever he created religion and in any language that he with him, and so he was able. And when Newton used a different technique where he converting a particle in this line is important. And it acted upon by an force. It was against actually but he wrote mind or maybe not understand. The idea was that anything that moves in circles is mind. And so because restriction was able to violate rules, but he was able to advance science. There is a balance one has to make. The balance has to be say well known, well preserved. We were able to have a discourse and diversity of views are very important. So if you're thinking about this, you have to think about the methodological approach first. And this is very and in which there is a professor from the university writing forward suggested that it is very important that you have a theory that what I'm just trying to suggest previously is that you should have theory and you
Alright.
comprehend their theory for correct use. And this theory rests on philosophical as well as physical assumptions. So always science is based on some assumption.
यहाँ से कुछ था क्या?
I'm hearing some sound, so if you wanted me to stop, please let me know. So when these philosophical assumptions particularly are important, personal and social mentality and behavior is quite different from other countries or cultures. And in this, we talk about this in Middle East and Africa, basically the science that have been developed, it's really European science. And you need to understand that even though you practice that science, there's nothing wrong that with philosophical concepts coming from the West, and you need to then understand what it is in the West. So India's very, very, very old. India was not part of Africa. Over 70 years ago, it moved to Asian continent, and that's what it is. So Himalayas is moving actually 2 million years every year, and the Himalayas has a huge impact on what India's geography is. And the geography is very, very important for what we are. The geography is one another every 100, about ice age. And in the ice age, everything generally is covered with ice, except the area of Africa and India particularly because it is protected because of the humans. And so we have diversity of views that have been very ancient. We have had so many types of ideas, and this days, India is really the minefield of information, knowledge, and all kind of other ideas, and that is very important. And it has been recognized. There is only one country which is in this one. You can see it's called Indian Ocean. There is no other country in the whole world that has any ocean. Maybe some sea, like the Chinese Sea. Those are British Sea. Black Sea, those are the small amount of water, but the recognition of a country with this huge amount of ocean, it is like something designated. It's just people just know that area from that point of view. So I want to just go very quickly over what is very important.
आप वाला बंद कीजिए.
East and West. So the definition, we know the middle school.
There is reverberation.
There is some sound coming. Can you mute, please, those people, anybody who has their audio, the microphone is not. Okay, so this is systematic study of topic, subject, problem, which provides consistency. Very, very normal. Is a non-falsifiable explanation. This definition that was given by Newton, and non-falsifiability implies you will try to falsify your ideas. Those first two fundamental signs that means you have to have control to make sure what you have and the last one is currently in practice. It's science that has the objectivity. Objectivity basically means based on the object that you have seen, that then you don't see it. But the subjective And this is why bias, bias. But actually, there is several studies that people who are doing science, why almost 95 people, even 99% are biased. We have our own hypothesis. Hypothesis generally are biased. I know that the people say that we have to deal with it. But bias in corona time, you may have seen so many reports of BCG against so there is doing whatever precautions we need to take, we need to take, and therefore that's just bias. What's subjective? How do subjective? As far as I know, only in tradition can be very neutral or detached and it's to be subjective. So that and that comes from yoga. You practice yoga. This is you're really if you do it well, you're at least trying to be objective or detached from whatever the fruits of actions are, and then you're much less doing bias. I think scientific can be done much better. Just give you one idea how this so-called activity, and particularly in terms of evolution, there are several main in science, matter, origin of life, evolution. These things are very, very important for researching from my perspective. And so the evolution, and this is I just taken it from text. But initially it was considered that total who is born, obviously then they came, this is a objective bias opinion. They come up with the way became and then it's true, which is in our type of animal is it came out on the top. So then we came up with the encephalization factor. I mean, I know there is an argument to make why the encephalization factor is important and other things, but here is the idea is divided by power 0.69. We are on the top. So this is the kind of biased and objective view you can see. I want to give a little bit peep into my own research. I know you cannot read this probably, but here is one here is the so it is evolved from anaerobic bacteria that not only attacks human beings, it causes paralysis in human beings, but it also goes after the most evolved system of human being, and that is the nervous system. And so I want to just give you a little bit of peep into how we go after this and how maybe Indian tradition might be very important to that perspective to look at. So we have recently, actually this paper is just coming out in a week or so. We have analyzed gene expression in neuronal cells by exposing with the botulinum toxin, and there are humongous number of genes being affected, 1,368 genes being affected, 100 affected, and major pathways, protein, cytoplasm, cytoplasm, all this type of fungal pathways are being affected by several genes being up and down. This is not so that I just from this work. I'm just saying that biology is very complex and it makes very statistic interpretation of this. It's not that boy, it's just only like that we have a huge amount of work, a huge understanding, and the detective view science that so we should be open and humble that is huge amount of work to be done before we can start claiming the problem. This is just so that yeah, of course we have that doesn't mean we have not done very well. We have done something very good. And maybe part of the when I'm talking and be able to use that for injuries. So we can all continue to do work and novel ideas, novel science, new understanding. But it's always important to realize that we have also used the same toxin, just meaning same toxins, some proteins we understand them and dividing the vaccine. So we can use this diet of everything. The major point I'm trying to tell you, we understand, we understand everything. But the thing we cannot use something maybe helpful. And some our head and we think that we are getting so much paper and also important that I can do this. And so I've been talking because this is like an and it's the nervous system. We would like to know more about evolution. And so we have combined chemistry, biology, that is your topic, and to understand not just the biology or the organism level, but also the level, some level, what exactly is happening, to some of the ideas from system. Or bits of members created everything. There are random mutations, which is what the science believes are random mutations selected by the nature, adaptation, and so that gives you survival of your body and the survival of your gene pool, and that makes you to be evolved. And but I want to just question that whole idea with giving you this four gentlemen's example, because it's survival of body and survival of gene pool, because ultimately survival of body leads to the survival of gene pool. So gene pool basically means the more kids you can have. And so if you can see, Osama bin Laden had 24 children. And his father actually, Muhammad bin Laden, had 54 children. And Mother Teresa had no children. Dalai Lama had no children. Now, you tell me, according to this science theory, Osama bin Laden and his father, Muhammad bin Laden, are more evolved. But it will look ridiculous if you really compare that to Mother Teresa or Dalai Lama, who is more evolved. That means there is something wrong in science, and that's where the opportunity is. Something is not right here. This is coming because we think that the science is human beings are selfish. It's about selfless people. What is the evolutionary selfless people that they want to look into, and again gives the opportunity for us to do research. There are evolution has occurred for 4 billion years, and human beings are here only for 2 billion years. That's 4 years all the organism by myself has to it is use what I want to talk this idea is matter, and what happens, one thing happens in this journey of the atoms, molecules, organisms, and tissues, is that there is everywhere is a bipartisan, and so the molecules also always linked, and molecules are linked, and the galaxies are linked. And that is something that my related to Indian you can see that all over. There's professor in MIT who is very interested in chemistry part of the physics of the life, how the life is developed. So I think this can be integrated and what you can see is proteins. Proteins normally structure is supposed to be the material protein, but found if you who are might be knowing that normally is a intermediate when the proteins are being synthesized and becoming native. And but we in our case we found that that's molten globule is the most active form. That means it is against what normal thing is about this I'm talking about this botulinum toxin, which is seems to be evolved from bacteria 4 billion years ago to now today and attacks human beings. And we really want to use that idea as to how that that can be interpreted in today's molecular dynamics and in that kind of analysis. And so I think this is a very interesting thing to find out about evolution and the idea of vibration or oscillation that is being mentioned in Rig Veda. Actually, we just a paper just came out from our group trying to corroborate our initial idea in 2001, and it's like 19 years later. We have done some experiment, and you can see this molecule here on the bottom, and that is shows that it becomes more expanded when it is more active, becomes more expanded, and which is the model that we drew at that time, not knowing this. This is we have used the small angle X-ray scattering or SAX and molecular EMD simulation to find this structure now 19 years later. It looks like that dynamics, and we have published another paper a couple of years ago where the motion of the molecules suggest that is very different compared to the inactive molecule. So that just gives you an idea about evolution. Now the East and West, I have few minutes left in my presentation. I think I have about 10 minutes. East and West is important. Again, there is nothing wrong in what East has or what West has. Like I said, West suffers a lot during Ice Age, and there are many Ice Ages also. Even today, the Europeans and mostly even Americans have only one crop in a year. And whereas in India, you have 3 crops a year. So you have luxury. The weather is more benevolent to people in India and Southeast Asia. And so it is very important to understand that we are coming from different cultures. And so whatever Western science is, it comes from So I think only one person can talk, in my opinion, when we are making presentations. So please either shut up or mute yourself. So important, and just nature and how the East becomes and the one which is dark at like it's 1 a.m. with the and so I get people with you, good morning, that's and everything is wrong with so that sustainability. So is doing geography is rule and Gaia Greek is go. It means oh not a cow, but also it means in Sanskrit. And it is important to understand that Gaia in 1960s, these people scientists were clear about it or not very happy about it. But now concept is considered as very important, especially for the environment and earth sustainability and sustainability of life on the earth. And especially when we are seeing that corona, we should be more sensitive. And that concept of ruby was there for thousands of years. And we have actually not been able to understand our own colors, jars, and what means for science. But I think scientists don't know that. So it's very important to know that. And there is a first so it really is wisdom, but Krisi is always interpreted, but really means means creativity. This means creativity of high order. Any creativity will be considered. Our is the protection of birds. So the sustainability means cow, puts cow also, but in this case, it means sustainable. You be really, you be sustainable, and then only you do the business. And so that will avoid interpretation or explanation of nature. If you fully follow this, and you understand this concept of maybe and east and west and your culture and what in supposedly is further understanding knowledge. So this is very important for us to think about. There one more point I want to say, and I'm my time, so I got maybe 5 more minutes. I do this in 5 minutes. One more thing for us to think about is that we have this 7 days for a week. Where did that come from? That also comes from the West. It comes actually from Bible. Because the word according to Bible, and this is just this is a model that is related let there be water, water, water. And creatures, plants, and things, and 6 days, so they can and now that is just a model, artificial know, I know, and it's not anywhere in scriptures, text, Mahabharata, Ramayana included. But that also totally used to be, which I think in the million people follow. I know they have so much interest, but it is they are all connected to nature. Unless you connect yourself to nature, the moment you become artificial, it will do sustainable because it is not connected. So there is scriptures and texts all over the world. The manuscript books all over the world. There are catalogs of these books. There are catalogs and there are catalogs. And India tops the number of catalogs. There's so much because of our training, British Western enslavement, we are not able to publish our own ideas, which is very, very important. If you want to look at culture that has developed over thousands of years and not used, and culture is only expressing culture, and it is life here. Culture is Bhasa, and but we is very important that That's when the Latin was reused. And people now use all the names still are Latin. And it's doing it. And the party where what I have is very, very important. So this is in Christianity also is said that you have is a sin. But in India, it's very opposite. Every child is divine. And and he started actually opening his eyes and he received Atman and using the Brahman with the connection. This is a people in neuroscience, and they do in the way Atman and same as Brahman, like very important, you know, and act for this Indian government act in the constitution is a fundamental is Article 51. It says you have every the scientific temper, which means those whole issues of entire whole entire life we look at from scientific perspective. And very important science is find that it is this discussion with the message about certain elements, and these elements are not like history, and they allow you to explain certain, you know, medicine, therapeutic, as well as poison, and can be explained. This idea applies to human beings in total space. You have space, then you are in this case we met, and this is based on Ayurveda that you know you practice for all the years. And in the young medical college in India to do this, and so I want to conclude by saying the knowledge you have will become detached. So the knowledge will become, you only become detached if you are biased for whatever religion, anger, whatever you realize, that will create a matter that at the world and but once you have knowledge, then you can test it, and so you can demonstrate like belief or professors, and then you do some technology and you can believe in that. I hope that this, of course, social implications I'm going at this point, but I think might discuss about things. How social is very, very important, and science integrated the society in all and every form, and I think that is a idea. But innovation will happen only from nowhere. It does not have to just copy. And unfortunately, in India, there is a huge habit of copying, not only copying innovation, but also copying that. The society, the best because maybe you're enslaved, and when they are enslaved, but they are not using, not using the other, they are appreciating the yoga, they are appreciating Ayurveda, and I think they will appreciate great theories and schools, scientific knowledge, and brought up by Indian experience. And so, thank you very much for this opportunity again. And I hope if there are any questions, I will be able to answer. I don't have to answer here. I will, सर्वोत्तम गर्मियाँ दिया है। I will start.
The intricacies of life and evolution. Sir, we know that you are a very good and eminent scientist. We are proud to have you with us. Thank you, sir, for being with us. Next one is the eminent speaker, Professor Kishore K. Srivastava. Dr. Srivastava is honorary scientist, ex-chief scientist, and of microbiology. His major expertise area include virology, microbiology, immunology, genetic engineering. Dr. Srivastava has published more than 60 manuscripts. He has 3 patents in his name, reviewed several articles, and wrote many editorials. Dr. Srivastava has been on very administrative and societal responsibilities and membership, being chairperson, Division of Microbiology from 2019 to 2020 in CDRI. He has been head division of microbiology from 2011 to 14, has been coordinator tuberculosis and microbial infections 2011 to 2018. Dr. Singh has been a versatile scientist and teacher also. I have been fortunate to have an association with him in CDRI, though it was for short duration, after which I had to left for Kenai, joining as an assistant professor. Again, I am fortunate to welcome Dr. Srivastava to present his keynote address on this occasion. I welcome Dr. Srivastava to present.
Thank you, Dr. Rakesh. Can you hear us?
Yes, sir.
Can you hear me?
Yes. You are audible, sir. Yes, sir. We can hear you.
Can you see the screen?
Yes, sir. You can share screen, sir.
I'm trying, but I think it's not opening.
Just a minute. Yes. How you can?
It's open now, right?
Yes, sir.
Can you see?
Yeah, it is visible.
Yeah. So thank you, Dr. Rakesh, for giving so beautiful introduction for me. And I think everybody everybody can hear me now, because the earlier talk we were not able to hear things, but the slides were wonderful. We could understand what Dr. Singh wanted to say. So I wanted to make sure that everybody can hear me here and the voice is clear. Can you confirm, Dr. Rakesh, that everybody can hear this? Are there any comments?
Yes, sir, you are absolutely okay. Your voice is clear. No problem.
So what I'm going to speak today is on a new paradigm to drug discovery. And the reason I chose this topic, because if you see like last 4.5 months, people are talking about the drugs, especially for the COVID-19 cases. So I wanted to apprise people at what are the difficulties and what are the successes in getting a drug out of the market. So basically, when we talk about the drug, it is not a very easy process. But we have to be ready because every day, every time the new diseases are coming up. So we cannot have a drug in our hand and just cure the people. So what we have to do is we have to pre-plan a few things before we proceed for a drug discovery. So we're coming to the so you see what is a drug basically? So if you see the drug, this looks very this picture looks very simple. But if you see the complications here, if you see the chemistry, you need a chemistry is hardcore chemistry for several years before you come up with a molecule. You have the pharmacokinetics, we have natural product chemistry, we have the peptide chemistry, and then after coming up on some novel chemical entities, you have to do strong biology, strong evaluation, different animal models, different cell system, cell biology system, different tissue culture system, different enzyme system to find out the drug is the molecule is active against those particular disease. Then these two topics are in the hands of the scientist, chemistry and biology. Then comes the issues of ethics. So in the ethics, you have so many things like biosafety, animal safety, human safety. And before that, you have to talk about the industry. So if you talk to industry and then the industry is going to take that molecule or the drug that you are going to market it. So there's so many complications. So what you see here, this process, it takes around 10 to 15 years. So we'll talk in terms of like COVID-19 at present or maybe other diseases, antiviral diseases. So what happens actually, people are talking, we want a drug next day. How can we get a drug next day? It's not because we have just heard about this disease recently. We know the coronavirus, but the disease we have heard just recently. So what we should do is just see the drug development process. If you see, I don't want to scare people, but if you see a drug development process, it is very tedious process, very time-taking process. So what you do is, Dr. Singh will agree that you have natural product or synthesis of something, some natural compound. Then you have to see whether those are soluble, whether they can go inside the cell, they can go into the bacteria, they can go into yeast, they can go to a parasite. Then you do the primary screening. This primary screening is you have to screen against those particular organism, either it is an organism or a particular cell. Then you determine the minimum inhibitory concentration and the IC50 inhibitory concentration. Then you again check the cytotoxicity, whether compound is not necrotic or just killing everything. So you have to check it whether it is not cytotoxic compound. You have to check the selectivity. Selectivity means it is killing only the bacteria or only the virus or only the cells you are talking about, not every cell or not any non-specific cells. Then you have to do the intracellular cell-based system. Means if you are talking about, suppose intracellular organism is there, you have to make sure the drug is killing the extracellular organism, but at the same time it is also killing inside the cell also. Then you have to find out the structure-activity relationship. So how many drugs, how many compounds, how many novel chemical entities you have synthesized, and how many are going to treat that particular disease. Once you have done this, then you have to go to pharmacokinetics. Pharmacokinetics means where is drug going in the body, how much absorption is there, how much metabolism is there. After that, then you go for animal studies. After getting the animal studies, you have to do a lot of animal toxicity and animal regulatory studies. Then comes to the clinical spectrum. At the clinical spectrum, you have to go for clinical trial phase 1, clinical trial phase 2, clinical trial phase 3, and then comes the industry. So before that you do the clinical spectrum, sometimes you get a target also. What is the actual target? So once you find out a target, where the drug is targeting, it will be easy process. Then you can straight go for synthesis of the drug. So now, if you talk about this slide, if you take every process for 6 months, it will take at least 7 to 8 years to develop something. So suppose our disease comes out next day or maybe tomorrow, how are we going to process it? So the difficulties is because the general public, general society, we don't know. We just want drug. But the thing is, it's a very complicated process. And so what happens? So we have some alternatives also. So suppose we are talking about novel chemical entities. A new drug is a new drug. So what the new drug is a random screening, because you have the known database. You do just phenotypic screening. You have a lot of compounds and just screen against a particular disease. Or maybe target based. You know a certain target where a drug can hit, and then you can develop a drug. But both processes are very difficult, because both process takes also from 2 years to 10 years. So what people are doing now, people are doing a repurposing. That's called drug repurposing or repositioning. So now we see a lot of people are talking about this. We have drug for COVID, we have drug for coronavirus, we have drug for this infection, we have drug to block the infection in the lungs. So what they are doing basically, so what they have been doing is they are extrapolating the common mechanism or the pathway which have earlier known for other diseases or for other viruses or for other bacteria or for other infections. And are also sharing the part of the structure of binding. So what happens if sometimes a particular molecule which has been known maybe 10, 15 years ago, and it is binding to certain protein or certain drug, which is common to this also present disease. So we can share that particular molecule and just find out whether this molecule can hit the particular target and eventually can kill the parasite or the organism or the virus. So what is a target basically? So in the definition for a student, is very important to know what is a biological target. A biological target is a naturally existing cellular or molecular structure involved in the pathology of disease where the drug in development is meant to act on. So basically, you know something in the cell which is essential to the growth of the cells, for the replication of cells, for the metabolism of cells, for the enzyme activity of the cell. And if you stop that, the cell will die, or the process, the mechanism will change. So basically, you identify those particular target, and those particular target once you identify, then it is easy to analyze a drug. So now this slide is covering all the diseases so far exist in the whole world. So in one slide, I have put all the diseases. So look for there are two kinds of diseases, more importantly, lifestyle and metabolic disorder, and then infectious diseases. So in the metabolic disorders or lifestyle disorders, you have cardiovascular, central nervous system diseases, diabetes, cancer, autoimmune disorders. And then there are other one is like infectious diseases, where are the bacterial diseases, parasitic diseases, fungal diseases, viral diseases. In the bacterial, parasite, fungal, most are the microbes. In the viral, they're considered non-microbes because they don't use their assembly, they use a host assembly to multiply or to infect. So basically, when we are talking a drug discovery or drug development, there are two different kinds of drug development, because if you're talking lifestyle disorder, metabolic disorder, you are changing a process into the cell. You are not killing the cell itself. In the cancer, sometimes you kill the infected cells, but most of times in the diabetes or in the autoimmune disorder, CNS, you just modify the mechanism. But in the infectious diseases, you are killing the particular organism. And that time you have to make sure that you are not hitting the normal cell, not killing the normal cell, you are killing the only infected cells. So if it's very important that you precisely target those cells which are infected or the infectious agent. In the cancer, you are also saying you are hitting the cells which are proliferating, not the normal cells. So you have to be very choosy whether you are going to choose how you are going to choose in between a normal cell and infected or unhealthy cells. Similarly, in the diabetes, when you have increased the insulin level, what you are going to do? Either you are going to do increase the beta cells, or you are going to increase the insulin level. So these are there are three different models people are using. So what today I'm going to highlight few things is how you discover a target. What are the target types and variability? What are the families of the biological targets? And how you validate those targets? Because once you have the target in your hand and you have a drug in the other hand, how are you sure that you are going to hit only the target? So you have to make sure that you have to validate it, whether it is specific to that particular target or not. So in the nutshell, you have three processes: synthesis of a new chemical entity or a compound or a natural product. Then you have to look for efficacy, safety, toxicity, pharmacokinetics, metabolism for new chemical entity in humans. This is more like biological studies. And there's a drug development, this human clinical trial. So a lot of industries, a lot of ethical people, a lot of the scientists are involved here. So, but what happens actually, if you design a drug, evaluate it and develop it, again, sometimes there is a problem after 5 years or 10 years, then you have again gone back again design it. So this process is something like a feedback mechanism. You have to design and you develop, and then you will after development, you may have to again design it. So first you have to found the druggability, because not every chemical molecule you synthesize in a laboratory, because every chemist can say I can make a molecule, but the problem is whether that molecule is a drug or not. So how you design it? So what you do is the probability of being able to modulate a drug with a small molecule drug. So target, when you change a target, you change the activity of a target or a position of a target or a mechanism of a target, then you can say this particular molecule is working. Is crucial in determining whether drug process are. So everything is killing, everything is infected cells are killed. But the thing is whether it is a lead. Lead means it is specific to that particular disease or particular mechanism or not. So if you see the human database, there are 20,000 proteins, and 10% of the human genome represent druggable targets. So only about 2,000 proteins or maybe around 3,000 proteins are druggable only. Not every protein is druggable. You cannot kill every protein in the body, because then you compromise the whole body system. So what is a druggable target? Has it sequence domain similar to human target? So known targets. So suppose you have identified a target and you have identified a molecule. So find out whether this target is available in the literature, that somebody else has also worked on this particular target. So, and has it affinity with the appropriate affinity to bind with this one. So now, so you have to look 4 things for doing this. So you have to look the absorption, distribution, metabolism, and excretion. This is very important process for the drug discovery program, because suppose you have a drug in the body staying for maybe several years, then it can be carcinogen, it can do several other things also. So you have to make drug absorb very fast, distributes very fast, metabolize very fast, and excretes very fast. So it doesn't stay longer in the body. So how you design it? So when you design this target sequence, you take the target sequence or structure information from the database from NCBI. Then with the see that this target presence in humans, so make sure this is not going to normal human physiology. It's not going to change normal physiology. So if you are choosing one target, it should kill only the infection, or it should target the mechanism, not the whole process of the body. Visualization of active site, other important, so whether the target has some active sites, can bind with this particular molecule or particular drug or not. Visualization of motifs, domain, ligate, and interaction. So if sometimes what happens actually, there's some kinetic sites also that can have a kinetic activity, enzyme activity. So also you have to see the IC50 also, whether what is IC50. It should not be very high IC50 value, because sometimes it will be overloaded and it can be toxic, highly toxic to the cells. And what other drug resistant information. So in the literature, you find out whether this cannot, suppose you design a drug for 10 years or 5 years, and then I eventually find out after 1.5 years, this drug is resistant. So it is of no use spending so much year in developing a drug. So it is, so if you go in the drug target databases, NCBI is a DTD, there are about 1,300 FDA approved targets, and about almost 390 human targets proteins are approved drugs. So people now are using these particular targets for other diseases or infection. So what are if you see the distribution, which are known distribution on the basis of the diseases, the maximum is on the cancer, 16.17 is a neoplastic disorder. Then again, 16.37 about hormones and hormonal antagonists, and about around 12%, 12.5% for the viral infection. So it is good news that we have around 12.5% for the targets for the viral infections are viral infections are known. So when talking about the current disease like COVID or coronavirus infections, we can be very comfortable that when we have at least 12.5% targets are known, which do not interact with the human proteins. And if you look what we should target, but most of the people target the proteins or the enzymes. There's around 80% of people target enzymes, and then 4.6% people look for the receptors, which bind on the surface of the cells. So these are two major targets to be chosen for a drug, because here one you are direct killing, you are disturbing the protein or inhibiting the protein. In other one, you are inhibiting the binding with the surface of the host cells. So now, what is happening here now? What we are talking about, that's everybody every day, you see the newspaper every day, they take a new article. So we have a drug for COVID, we have a drug for coronavirus. So what is happening here? Why people say that? So if you see this picture, you have almost 21,000 human proteins here. This same paper came in 2004 in Nature Reviews Drug Discovery. So if you see here 20,000 human proteins, and you see they have docked with the several molecules available in the libraries as worldwide, how many molecules and how many libraries, and they superimposed it. So they did docking and they found out there's so many proteins binding to so many molecules, and there's so many molecules are binding the same protein also. So the thing what you see is, so many drug targets more than one protein, and for many targets there are more than one drug basically. So this is important, is a good news that if you have one drug, at least you can think that I can see whether this drug can treat other diseases also. And sometimes if you have one target protein, you can reposition or repurpose your drug and see whether this drug can inhibit other diseases also, or other targets also or not. So look this picture. So if you have like 1200 targets, you see, suppose for example, I'm looking for the X side and Y side. You see the blood and blood form. I'm just giving one example. So there are 13 targets shown. But you see 13 targets, out of the 13 targets, 8 targets are for the circulatory and system diseases also. So there's some common targets also, which are so similarly. You see that I have just done the checkerboard analysis, and you see several targets are common to several diseases also. So this give you very good indication that at least we can talk about the current disease, that we can have some molecules in the market soon. So now we see, so how we do it. So you see how tightly we see how we do the docking also. So look, suppose we have the structure, we have the 3D structure, we have the X-ray structure, and look for the protein. So it is an example of a tuberculosis protein, tuberculosis, and you see the INH is a standard drug for tuberculosis, and see how tightly it is binding. So you know what is the target here. And similarly, a very common protein which is called BCR-ABL, and if you see there's a glibenclamide, and which you can see the very tightly binding. So how then this is the, you see the hit, and then you develop so many a panel of the drugs here from here. So so now so you have come up with the two things, the virtual and actual. So you have virtual targets when you do the docking, and the actual target when you know this virtual target is actually working. So once you have the actual target in your hand, okay, this is the protein, this is the target protein, we can hit it with a certain drug. So what we should do? What we should do is we should find out the biological function, and or we should make a recombinant protein, and we can make a recombinant enzyme and see whether this is really hitting, it is really being targeted by this particular molecule or non-chemical entity. So now coming to this problem, relating to the problem what I have shown you earlier. So this is the article came very recently in Nature. So they have shown the structure of the coronavirus, and everybody knows because every day in the newspaper and everywhere you are seeing this structure. It's very common structure. But what is important here is, so look here, this is one of the biggest genome virus, RNA virus so far known. So it is around 29 kilobase virus, and it encodes for 29 proteins. And out of those 29 proteins, what we have to look at, what are those proteins which are unique to the coronavirus? Because if you are hitting everything, then you may hit to something else in the host also. So you have to make sure what are we hitting. So there are 4 structure proteins which is called E-M, E1, E means envelope, M membrane proteins, N proteins a nucleocapsid which is binding to the viral RNA, and S protein is a spike protein. This spike protein is very important because when people talk about the infection, it goes immediately to trachea, immediately go to the lungs, binding with the epithelial cell, going to the pneumocytes also. So how? So if you suppose we don't have this spike protein, so what will happen? So you cannot stop making spike protein. So what you should do is you target the spike protein. So so if you see, so what people have done is they have identified COVID specific protein to be targeted. So there is a D3 target database in 2019 on norovirus. So they have found out 2 ways to find out whether what are those drugs which are used in the clinic, and find out what are the role of the in vitro and vivo studies, and found out that those drugs which are targeting this particular protein or enzyme, and for identifying lead compounds against potential drug targets via docking. So you can see the structure of this protein and can find out the target. So there are there have been two groups. One is spike, which binds to the surface of the host cell, and other 28 proteins, which are basically the structure proteins or the enzymes or proteases of the virus. So Philip, what is the statement here? So when the virus attacks the cells, it compromises the cell physiology in order to replicate, because virus cannot replicate on its own because doesn't have its own structure to replicate. So it needs a host to multiply. The drugs that have been identified may be able to inhibit these molecular mechanisms so that the virus can no longer use them for its own survival. So you still need to further validate and test it. So now, there are three drugs which recently have been actually repurposed. Earlier they have been used for other diseases also. So coronavirus 1 is SARS-CoV-1, and also for hepatitis, there's another virus disease. So these are the drugs, leponavir, remdesivir, and famotidine. So these are the drugs which are hitting the virus itself. So, but important thing is the virus has a very fast escape, because you don't get much time to hit the virus. So what happens actually, the virus immediately goes to the cells, host cells. So what will happen? Then you cannot use these drugs. You can use these drugs, but at the same time you are also going to use some drug which can also eliminate the virus from the host cell also. So what they did actually, so they have a recent paper in March 27, 2020. So they have found that what are those proteins which interact with the human proteins or human surface proteins, and then it will be easy for targeting the coronavirus infection. So now this is the amalgamation of killing the virus and as well as killing the virus inside the cells, and also activating the cells to eliminate the virus by using some prophylactic or a vaccine candidate. So if you look at what are the drugs acting on human targets, so they have done almost the studies, and they have almost 15 to 16 molecules which are being clinically tried in the phase 1, phase 3, phase 4 trials. So you see the first one is the Giapreza, which is 81 angiotensin receptor blocker. It was used earlier in 2017 for hypotension. Similarly, Davasan or Velastatan, it is also an angiotensin receptor blocker. It was used in 2002. Captopril, which is angiotensin ACE enzyme inhibitor, is got developed in 1982 for hypertension. Ivermectin, there are so many drugs which have been used earlier. So now they are actually extrapolating these drugs for the current diseases, current infection diseases, on basis of some similarity, some basis of some structure based, or some are based on the docking. They found out these drugs can be used for infection or blocking the coronavirus to infect the cells. So what you do is, once you do find out a drug and once you find a target, what you should do is how you confirm it. So first way to confirm it, this is use the enzyme or substrate and see the product. And but once with the inhibitor or drug, there should be either no product or the product is less. So this is the first way to screen. If suppose your drug is not hitting an enzyme, drug is hitting something else, some other mechanism, some other lipid, some other biological process. So then you see the change and reversal in the biological function. And the third one is, suppose you don't know anything, suppose you are talking about something in the diabetes and cancer, so you have to do the reverse biology. You generate knockdown or knockout mouse model to see whether you are hitting that particular specific target or not. So now let's come to very specific, because there are people in today's I think meeting, there are a lot of people from physics, from chemistry. So it will be a very specific subject for people who are working on the biology system. So how you do the study the proof of concept. So now you have a drug, you have a target. So how you do? Suppose you just hitting the target doesn't mean this drug is ready. So you have to make sure this is very specific. It is killing only or just targeting that particular one, or it's not. It is not doing anything else. It is not hitting anything else in the body. It is specific to this particular target or particular disease. But that is a tedious process, because that's why we've been talking about a drug discovery program. This is the most tedious process, that you are taking a specific drug. So people were talking the coronavirus or COVID infection. They are not sure, because they are telling you the drug, but they may hit something else. After maybe 5 years, you'll know this is hitting something else. They are going to kill something else, or they are going to have a lot of side effects also. So in order to that, this process should continue. That specific drug to COVID-19 should continue in the development. So So to do that, you have to do the genetic studies, transcriptomic studies, and proteomic studies to find out this drug has no issue on the in the genotype or phenotype in the body. So what is the functional genomics? And I think most of the students know this. Functional genomics is systematic analysis of gene activity in healthy and diseased tissues. So what is the difference between healthy tissues and diseased tissues? So you have to do the comparative studies by finding out the mutations, by finding out the different levels of proteins. Similarly, the study of an overall picture of genome functions. Functions are different in infected and uninfected, or maybe treated or untreated cells. There's a function of genes, protein, and organism. So you have to see in the organism that different function of this protein is different than the host. And then the integrated biology system, where you have this cancer, parasites, virus, bacteria, and lifestyle disorder diseases also. So basically, when you see talking about the genome, you see the sequence of the DNA and then see whether there is a mutation or not. If you are talking transcriptome, you are looking for the RNA levels and see the whole transcriptome, there is any change or not. And also same thing is the protein, whether the product or the end product is not changed in this case. So when it is the gene array, this is now a whole process, but you say microarray or it's a microarray. So what you do is you treat the cells with a drug, or you treat the cells with untreated, and then you take out the RNA from both of the cells and just see what is the difference. Whether this cell has a different expression of different RNA, and this cell has a different expression. And then identify those particular RNA, and then find out, go back and see whether you are talking the same RNA when you are talking about when you initially started with the target. So you take this is a normal cell or disease cell or treated cells or uninfected cell, and do the extract RNA, label it, and hybridize with the cDNA library of that particular disease or particular host cell, and see the superimposing and see the difference. If you see the difference, and you can see these are particular RNA transcripts are being changed, and then you can find out this particular target. Suppose the 4 green ones are targeting some particular proteins or particular RNA which you are talking initially, then you are through. Then you are happy that at least we have started it. We have found out some indication that this drug is going to be acting specifically on that particular target. So, and then you generate a heat map, and then the heat map will show you how strong is that. So if you are see the G1 and the E1015, then you can see this is a very strong binding, and there's a strong induction of the particular RNA level. So I'm not going to go in much detail because it is available in most of the papers on RNA transcriptome. So similarly, you have when you talk about proteomics, you have the 2D, 2-dimensional electrophoresis, and do the mass spectroscopy, and then you might find out that you have treated a particular cell or particular infectious agent, and then you look at this treated and untreated, and then see the difference in the protein. And if you see the difference in the protein, then go to mass spectroscopy, and you find out, okay, I wanted to hit this enzyme, and we found this enzyme to be hit by this particular drug. So then you are happy that at least we know this. We have found some indication that this is my molecule is hitting that particular enzyme and particular target. So this is the mass spectroscopy, and then how you see the mass fingerprinting, and just see what is the size of this mass, or how is the homology of those particular peptide, and find out where this is. Go back and see in the database is the same protein or not. So then once you have done the transcriptome, the proteomic, then you see going back to the animals and generate a knockout mouse. Suppose, for example, it is a particular gene like deoxygenase, or particularly hitting this maybe this spike protein. So you make a spike mutant and just see this protein is going to, when you infect in the animals, this animal is going to specifically going to react with that particular gene or particular protein. So either you make a knockout, that you remove the gene from the mouse, or you knock in the gene, you add another gene which has complementation to that one, or maybe conditional, which is grown in presence of some particular antibiotic, or in particular conditions, or maybe in the heat, or in the increased temperature. So this is the process of recombination. I'm not going to go in detail because most of the people, I think, in the biologists can understand, and I think the chemistry, physics people should not go in detail for this particular thing. But this is a common homologous recombination process where you put a drug-resistant gene and put it to the cells, and then go in the eventually go the embryonic stem cells in the culture, and then go in the mouse and see and select the mouse whether that particular mouse has got your genetic mutation or not. So this way you can find out, you create a mouse which is not expressing that particular protein and that particular enzyme. And now this is a model you can use with your drug, and that the drug should not react on this one particular, because that model is ready for that particular mutant, and you can screen your drugs with that particular mouse, which is called a knockout mouse. So now, so drug resistant mutants are, you have got drug resistant mutants, you have drug resistance alliance, you make the genomic DNA, and you do the genome sequence of that particular gene. So now coming to this slide. So if you have a drug, suppose for you have a drug, now we are people are talking about a lot of drugs in the market. So once you have a drug, you can identify a target very easily, as I told you. But once you have a target also, you can also identify a drug, because once you have a target, you have the structure, 3D structure, you just dock it with the known drugs in the market, in the library, and see whether this molecule is superimposing with this target, and then you can synthesize in the laboratory this particular. So this is the way you design it, and thank you very much for listening. And I think if you have any query, if you have any question, I'll be available. I have just put on the first slide, my email address is also there, but I'm available here to discuss anything, and any intricacies or any, because I have given a very superficial lecture. So knowing that people are from different disciplines. So if you anyone want to know in detail how the drug is developed, what are the small things in between which I have not told about, I will be happy to tell you. Thank you everyone for patience listening.
Thank you, sir, for your very informative lecture. You have discussed on a great length about the drug discovery and development, about the drug repositioning, important properties of drugs, how distribution of targets, locating targets for drug. You have also delved on great length regarding many targets and one drug, vice versa. This may be providing promising input for development of corona drug. Thank you, sir. We have 10 minutes for discussion, and Dr. Professor Balram Singh is also present, and a elite panel of speakers is available, so we can have a session of discussion for 10 minutes, and thereafter I will invite Professor Arvind Singh, who has joined us. Welcome, sir. So we can have a
Thank
discussion.
you, sir.
Thank you, sir.
Very nice talk, Kishore sir. This is Arvind. So it's a pleasure always to talk to you. So let me ask, to start the conversation. So where do we stand, like from the CDRI perspective of developing a COVID-specific drug, sir?
Actually, we have in Immunophlovir, we are going for this clinical trial now, because we are synthesizing at CDRI. Because if you know, in 2005 and in 1990, China and Russia developed this drug for some other purposes, and we have found actually in the we have a very good structure biology unit, and we have found out that this particular molecule can inhibit the COVID. So if you see the newspaper and they approve DCGI report that CDRI has got the permission from for this Immunophlovir clinical trial. So we are working on that, and I think we should be also very hopeful. And also we have suggested a few people like you also can do it. That like a BCG, using BCG and using HSP60 promoter, you can have like we have earlier done with HIV and other proteins also, because BCG is giving a non-specific cytokine response also. So if you have the BCG model, BCG is a known FDA approved vaccine. So if you have BCG vaccine and putting that particular gene into BCG downstream to HSP60 promoter or some other promoter and having a kind of adjunct vaccine. So the same times you can have the protection against mycobacterium, and the same times you can have protection for other viruses also. So I think Dr. Arvind, you can choose this one because you have a good expertise in this area. So I suggest you to explore more like BCG, and we in CDRI have suggested people, so people are in parallel working on that one. So you can have in parallel in the certain vaccine. And then also the CDRI also working in some neutralizing antibodies also. So we are looking for this tissue culture lab, and once the viral culture lab is developed, I have suggested people to just work on the neutralizing antibody also. And also some recombinant proteins which can be developed in animal models, because CDR has a very strong hold of animal house, so they can do a lot of work.
Very nice, sir. So actually, we have developed a novel viral trap system, like using a approach which can improve the efficacy and the specificity of all these diagnostic kits. So I'll be reaching out to you means to take it further. Like so, essentially, like all the kits that are developed, they are using a diluted sample. So our approach is if we can concentrate the virus from these diluted samples, so increase the specificity and efficacy will be there. So we have filed a patent on it. I'll be reaching out to you.
That will be good. Best of luck.
I can ask a question to Dr. Srivastava. I have not understood exactly. You mentioned how BCG works. I mean, there is a problem with cytokine storm anyway. So is that really a true observation, or just a correlation of some kind?
No, sir, what happened actually in around 1990s, the guys, Ken Stover and I think Richard Young from MIT, they have some developed the gag call and proteins of HIV, and they put in the BCG. So what they did actually, they have made the plethora of several proteins and developed the CMI response. It happened also with the Steve Hoffman also for malaria from NIH. What he did actually, he also made some 9 proteins and put in the BCG and tried to see the CMI response. So what we see in India, if you see, people are saying that people are in India are not more mortal, not dying as fast as in other countries. So people presume that because of the TB infection, because of the TB vaccines, because of the BCG vaccines, people are protected. So this is just a speculation. But what I think is that there's a group, Steve Hoffman's group. I think Dr. Arvind knows the Steve Hoffman group. But they have done that they say that there's a common similarity between BCG and the coronavirus. The reason is because those are the lung infections. So if you look at the lung infection, if you take a dead person and take out the lung, take out the bronchial lavage, and you see there's 100 types of viruses, 100 types of bacteria, 100 types of fungal infections also. So you don't know whether it's only coronavirus or maybe other infections also. There's a lot of opportunistic infection. So when I say use the BCG, because the BCG, if you take the BCG route, it is an easier route because then you don't have to go for long-term clinical trial, because BCG is a well-validated vaccine. So if you use a BCG as a model, at least you have to do phase 3 or phase 4 trial straight away and then can try it. So in BCG, what happens, BCG is a non-infectious. And then if you have a BCG and expressing the coronavirus protein, maybe take example a spike protein. So if you take a spike protein and it is expressed, it will not bind to the particular host. So this is my assumption, could be wrong also, but see what happens, because everybody is talking about serendipitous, so I also thought of that.
You're very
I
much
think
right, sir.
very
If
good.
I may add, like BCG is a good adjuvant. So
Yeah.
for any vaccine, you need a good adjuvant. So essentially, like this will generate a good multi-epitope specific immune response. And I think in India also, GP Talwar's group is taking mLepri vaccine of their own. So they are using it, repurposing it for the COVID.
Yeah, well, that will certainly be a very good idea. But I was just not able to understand how the BCG is working right now against corona. But maybe what you are suggesting, that makes sense.
It should be a recombinant. It's not only BCG, it's a recombinant expressing the coronavirus.
Yeah, that makes sense. Okay, thank you. Well, I since nobody's asking question, I apologize for my voice not being that clear. I didn't realize once you share the slides, you don't see any problems I didn't get to see my comments in the comment section.
Your
So I
slides were very like expressive, sir. So thank you for a nice presentation.
had to read my lips. But the point I wanted to say, basic point is that for innovation, it really needs to be local. Because otherwise you're just copying outside. I mean, copying outside in terms of approach and technology and other things are absolutely important. But India does have a huge resource of holistic system, and that can be developed into a science that Western world is just recognizing it. And they're doing a lot of work on, for example, meditation, yoga, and holistic approach of herbal medicine. And it'll be shame that Indians didn't do it when somebody else did it from outside. So that's the major point I wanted to convey. Everything else was just a detail about it. And this is the general practice of the science development from European Renaissance to today. So I think this is an appropriate time, and I think particularly appropriate time for small colleges like Kamala Nehru Institute of Science and Technology, that they should get involved, because then it become more local anyway. And that will be something that senior professors and scientists like all of you should really encourage that, mentor that, get involved, because this is almost like a national mission for the rest of the world, the national mission for the rest of the world, because the rest of the world always thinks about India very differently. Unfortunately, in last few hundred years, we haven't been up to the mark.
Yes.
So, Dr. Kishore, Rakesh here, I have a question for you.
Sure.
So, yeah, I know this Arbidol is going on. The clinical trial of Arbidol is going on CBI. So, this what is the indication in terms of it is going to be prescribed for mild to moderate symptoms, symptomatic patients, or when the patient gets into the severe situation?
Yeah, actually, Arbidol is a phase 3 and phase 4 trial basically. So until you get it, because sometimes what happens actually, the life-saving drugs are sometimes highly toxic and they are really expensive also. So sometimes when you are taking the drugs, actually the patient is not going to survive. So now these drugs are being used with those patients basically which are highly morbid conditions. So at present, until a phase 4 trial is not completed, you cannot use this drug in the patient which are in the early stage of infection. So this Arbidol and immunofovir or these are drugs which are being repurposed for this coronavirus, are not basically ready for people who are active, who have oxygen level more than 92 or 93. So when you come in the bed and you are using the ventilator, then these drugs are being recommended.
Mm-hmm. So if you have to, I understand, long way to go to understand how it is going to be effective. But there may be some preliminary indications. If you compare the efficacy of Arbidol with Favipiravir and Remdesivir, where it stands.
Yeah, they are, because when in 2003, I was there when SARS-CoV happened in the US, and they are using all these drugs for those particular purposes. And then they have found that there was indication that it was affecting the SARS-CoV-1. So from there, they got an indication that it could be used for Arbidol and used for SARS-CoV-2 also. So this is the indication they are using. Now, they have not confirmed that it is going to, because you don't see that there's a state where something comes out, okay, Arbidol has cleaned this viral infection. So these all suspects are there. Okay, this can be used. There should be maybe particular modifications also. What condition they have to use, what percentage of infection was there, then it was used. So there's so many things have to come out. At present, saying Arbidol can just blank give the treatment, it's not. I think the correct decision.
So there is one point of discussion I wanted to ask from the panel is about the hydroxychloroquine. So there are a lot of conflicting clinical outcomes that we have seen. There is no good news from US, and but in India, ICMR has recommended the use of hydroxychloroquine at least as a prophylactic agent. So any comment on that? How the people are not prescribing in US, but in India it is being prescribed. Any reason?
The important thing is if you are using for prophylactic, anything you are using for prophylactic, it means you are going to work on healthy subjects. And healthy subject will never volunteer until something is validated properly. Suppose if you say, give me a vaccine which is not properly validated and inject in myself, I will not do it until it is properly validated. And if you sue, I'm going to talk somewhere on the vaccines also. So vaccine also take like 10 to 15 years. So sometimes you cannot have that particular immunoprophylactic or immunogen without properly validated. Similarly, a drug, you cannot take a drug knowing that you are going to have a coronavirus infection after maybe 6 months or maybe after 2 days or maybe 3 days. So you have to, so these are drugs that you have to use at a time active infection. Now the second thing is this virus escape is very fast. After 4 days, when you know virus already infected in your body, it has gone to the lungs. And once it is the lungs, it is already encapsulated into a cell. All it has gone to the cell. And then what happens? Now you are not going to hit that particular cell until you are sure that other cells are not being affected. So there are 3-4 things before using a prophylactic drug. Like prophylactic drug, it's very, very difficult to use. Very, very difficult. You can take some Ayurvedic or those kind of things, but a particular chemical drug, taking a particular chemical drug is very good. So when we're talking, ICMR talking about those drugs which have already known they are non-toxic. So if you know some non-toxic drug, that's fine. If you're like taking the Crocin and taking all the paracetamol, and then that's fine. But knowing the drug that can be have even 10% toxicity, you cannot use as a prophylactic. And this particular, let me tell you, Dr. Shankar, the problem is will be that now we are talking a global. We cannot talk about India. We cannot talk about a particular country. So ICMR can talk, but ICMR cannot say like this that you use it in this country and you can do this. But it's not like that because people are migrating from there. People are going back to US. People are going back to Europe and coming from there. So it cannot happen like that. That okay, ICMR is saying to be used. So there should be a proper validation. There should be proper efficacy. There should be a proper treatment. There should be a line of treatment. Then only a drug can come to the market. But the people are dying. Then you cannot do it. You have to give something. So these things which we are talking about, all the 15, 16 drugs, they are being used people who are really in the morbid and mortal stage. So they have to give it this drug. Otherwise, they are totally on the ventilators. And these drugs which we are talking about, they are almost given on that stage of ventilation.
If I can make a couple minutes, I think in the US, nobody has used prophylactic. So all the drugs, all the time chloroquine has been used, either people who are in ventilator or those who have already been identified with having the either infection or disease. I have no idea why they cannot. No, this is not the case that prophylactic drugs cannot be given. Chloroquine is approved here also. FDA has approved it for different purpose. So it's at least safety is known. And I think people are crazy to take anything if it is properly given. So people will take it. There is another aspect in the United States which is political. It's because Trump said so. Because Trump said it so strongly in his press conference that there is an effort to prove him wrong. And so that's why it is not being done. I know a friend of mine who is just submitted IND to FDA on using appropriately this one, and probably it may take 5-6 months. Eventually they will get it done. So hopefully we will have some results, properly done results. And I hope that ICMR has approved it. I hope Indian population is collecting good data about that as well.
Yeah,
I
that's
think
true, sir.
ICMR
That's true.
did point out that the difference between US and Indian study was the dose. So that is one of the factors that was. So I think.
Yeah, that's true. Yeah.
It also causes the retinopathy also. So if you people are not aware, they should be very cautious because chloroquine causes a lot of retinopathy also. There are a lot of side infections also. So I think that's a debatable basically.
Yeah, Dr. Kishore, I think what Professor Singh and mentioned that there is a political issue as well as in India somebody mentioned that there is a dose dependency, right? and this is anti-malarial drug, and the toxicity is well studied. Efficacy is also well studied. There is a tolerance. So, um, you know, how to make use of it depending on the patient's situation. I think it's not bad drug in a given circumstances. So it can be global, but there should be proper studies done. As Professor Singh said that somebody has filed IND, there could be some successful clinical outcome. So
Yes, yes. Something will be done in next 6 months or so. I think IND has been approved from what I understand.
Thank you, sir. There is time constraint. Thank you, Professor Balram Singh. We ultimately have been able to hear your voice very clearly. Now, we have listened you so many times, and you are very good speaker, speaking on a variety of subjects. We have heard you speaking on Audhi language also. You are working from grassroots to very high scientific level. Similarly, Professor Srivastava, actually, I have learned science from him. He has introduced me how to do science in lab. My friend, Dr. Arvind Chavra, and my very close friend, Dr. Rakeshwar Bandichor, we have been together for so many times. Thank you very much for everyone to sparing time. Now I would like to proceed ahead with the conference. I invite Dr. D.K. Tripathi, Associate Professor, Department of Geography, to take over the task to invite next speaker.
Thank you, Dr. Pandey. It is my great pleasure to introduce such a great academician, Professor R.V. Singh, who has been a well-known international personality of the academic arena of geography and earth science. You are a source of continuous inspiration for geographers and earth scientists of this generation, sir. Sir, you started your career as an earth scientist and further served Department of Geography, Delhi School of Economics, Delhi University, as professor and head. Under your leadership, sir, the department was ranked as the best institution of India for geography studies under the QS World University ranking. You had been the vice president of highest world geographical body, International Geographical Union, IGU, from 2012 to 2020. At present, you are looking after as Secretary General of IGU, sir. You are chairperson of Research Council in several CSIR institutions of India and associated with several government organizations of India like Ministry of Environment and Forest, Department of Science and Technology, National Disaster Management Authority, and ICSR. Recently, you nominated as an expert member of Vision India 2035 of NITI Aayog and member of prestigious scientific committee of International Council of Science for Health and Well-being in Changing Urban Environment System. Sir, you have worked and supervised a lot of research work and authored about 50 books and published about 200 research papers in journals of international repute related to environment and health, land use, climate change, urban environment, disaster management, sustainable development, remote sensing, and GIS. You are editor of several prestigious journals like Sustainability, Advances in Meteorology, Physics and Chemistry of the Earth, Naam Today, and General Mountain Science. So I welcome Professor R.V. Singh, sir, to kindly deliver the keynote lecture and enlighten this conference, sir. Professor R.V. Singh, sir.
Can you allow for the sharing the file? Dr. Tripathi ji, please allow sharing the file. Are you able to hear me? Hello?
Yes, sir. Yes, sir. We can hear you.
Okay. So first of all, I would like to congratulate Kamala Nehru Institute of Physical and Social Sciences, Sultanpur, Principal Sahab, Dr. Radhe Shyam Singh ji, Dr. D.K. Tripathi ji, Dr. Avadhesh Kumar Dubey ji, for taking very important initiative, particularly selecting this theme, research and innovation in physical, biological, and social sciences for sustainable development. I would like to take the few important Indian experiences. As you know, when we see the research and innovation, particularly in physical and biological and social sciences, I remember few important initiatives, global and national challenges. Particularly in March 2015, India has signed a very important agreement that is known as disaster risk reduction. Following in September 2015, SDG, Sustainable Development Goals. Then it is followed by the Paris Climate Action. And in the following year, we had Habitat 3. So what I would like to tell you that our research and innovation must take care of these global initiatives, starting from DRR to SDG to Paris Climate Action. And when we are combining, integrating all together, I think that will be a great contribution to physical, biological, and social sciences. Another important point I would like to initial a statement I would like to give that when I heard your topic, I remember a very important term, resilient science for societal implications. So these biological, social, physical processes vary from place to place, district to district. It is not like a North Iraq or the South Iraq or North Italy or the South Italy. India has a pocket of poverty and pocket of prosperity. Every district has a pocket of poverty and pocket of prosperity. So capacity building, increasing awareness towards resilient science for societal implication, it is a very, very important. While integrating, two things always sound in my mind: prosperity of the people and prosperity of the places. And so that is why development means accumulation of human capital and its effective investment in the process of the economy. It encourages improvement in the environmental quality as well as quantity of life. So physically, culturally, and psychologically. So you can see the shift in the development process, how we improve from economic and cultural progress to economic growth and rural development, regional development, integrated rural development, state development. And then now we are living in an era of sustainable development. And 17 goals and 169 targets are the very important aspects where we can bring innovation, good practices in our research and education to meet aspiration and opportunity for younger generation. You know, this fashionable world came before the development, particularly after the and we are looking at the change. What type of change? Change related to technology, change related to economy. So in this case, we have to see the institutional change consistent with future as well as needs. It is very important to understand the immediate complexity in this context. And so here you can see the most important adopted definition published in our common future, development that meets the needs of the present without compromising the ability of future generation to meet their own needs. And so in this context, it is very important to see the India's complexity. I would like to take this, elaborate a little bit more and take from Dr. Abdul Kalam. The concept leads to a unique meaning to development and sets an integrated target for the measurement of development, which has combination of parameters, including economic status, poverty, education, health, women empowerment, harmony, physical connectivity, and environmental aspects. And what are the three key elements? These key elements are environment, where we have to maintain the earth biological system, physical system, ecosystem services, and pollution filtering. Social system, maintaining community capacity that foster effective participation and equitable treatment of all stakeholders. And economic, maintaining economic system that provides a non-declining standard of living for this and future generations. I would like to elaborate a little bit more about this objective of sustainable development. Here you can see this, how biological, economic, and social system are integrated in this. Biological system, where genetic diversity, resilience, and biological productivity are more important. Economic system, reducing poverty, equality enhancing, increasing useful goods and services. And social system, cultural diversity, institutional sustainability, social justice, and participation. These are very important. So you can see these pillars of sustainable development, how these are can be a public science. What your central topic, central theme you look at biological integration, it is a how we can improve our understanding and communicate to decision making. So science policy interface. Here you can see the how sustainability is surrounded by social, economic, environment. And then we have wearable, viable, equity. All pillars are mutually inclusive and interdependent. And so here you can see the major dimension of sustainable development: political, economic, institutional, technological, socio-cultural, ecological. And you can see that how political system, in the present corona time, without having any policy on migrants, we faced a lot of the problem. So that is why our all approach was so during the corona time, we faced economic, we have to bring the lot of packages. Institutional, I think it is very important that the migrant from Mumbai or Delhi or big city, they migrated to the states like Odisha, West Bengal, Madhya Pradesh, Uttar Pradesh, Bihar, Jharkhand. So we have to develop the institutional framework, and for that we have to bring the technological, socio-cultural system of the local people, and ecological, I think we must take care of. How we can measure the sustainability? Carrying capacity. Carrying capacity of biological productivity in an environment is the population size of the species that the environment can sustain infinitely, given the food, habitat, water, and other necessity available in ecosystem. It is possible for a species to exceed its carrying capacity temporarily. Another important measurement is ecological footprint. It compares human demand with planet Earth ecological capacity to regenerate. It represents the amount of biological productive land and sea area needed to regenerate the resources and human population consume and to absorb and render harmless the corresponding waste. So now, coming to 17 Sustainable Development Goals, these are the very, very integrated, and all are integrated under the biological, physical, and social framework. First, I would like to bring before you the carrying capacity of our Indian environment, characteristics of our Indian environment. In global term, India has almost 17% of human population, 15% of the farm animal population, 2.4% of the only geographical area, 1% of the rainfall, 4% of the freshwater resource, 0.5% of the forest land, and 0.5% of grazing land. Now you can understand from your own perception type of carrying capacity we have in our country. So that is why this time we got, key environmental challenges we faced during the corona time. Poverty continues to be a very important challenge. Population growth, inappropriate use of technology. These are the 17 goals transforming our world. No poverty, no hunger, good health, quality education, and so on. I would like to take the one example of poverty. We have very typical nature of regional structure in our country. We have states like Jharkhand, Odisha, Chhattisgarh, where per capita income is very low, but more rich natural resources. We have states like Punjab and Haryana, where per capita income is high, but less natural resources. We have Gujarat and Maharashtra, where per capita income is also high and natural resources also better. And we have states like northeastern states or Himalayan states, where per capita income is less and natural resources are also less. So very typical nature of the poverty we have in our country. So that is why it is called syndrome of collective poverty. Every district has a pocket of prosperity and the pocket of poverty. And for that, we have to bring, I would like to tell you that agriculture is the mainstay of our economy. How you can end the hunger and remove the poverty through the food security, improve the nutrition, and promote sustainable agriculture. It is not a physical availability of the food, but economic and physical access to food, food utilization. So that is why availability, accessibility, and safety, these are very, very. Now I would like to bring a few very important concept. Developing country, barely 30% of agriculture production undergo industrial processing. But in high income country, 98% is the process. In UP and Bihar, we have agriculture, we have animal husbandry, we have forestry, we have mining, we have fishing. What is lacking? We are lacking processing. We are lacking marketing. I think the most important integration is how we can integrate agriculture with processing and marketing. I can tell you, if Uttar Pradesh, all district, every district surrounding to every district, 2 or 3 important agriculture or animal husbandry based outlet can be created, and processing can be there, marketing. I can tell you, on Saturday and Sunday, people from the city, from Kanpur, they will go to the such small places with family, and rural tourism will reach. I would like to bring before you here the concept of 6 industry. 6 industry means 1, secondary, 2, tertiary, 3. So 3, 5, 6. In this process, I think we can have the more important lesson and improve the farming income. I would like to tell you that green technology in production, India and Uttar Pradesh and Bihar can become the important global supplier of the green product. If we can have this type of the process, I think that will be the great contribution. The broader situation I am putting before you, the health. Southern states are faring more better. But the still starting Rajasthan, Madhya Pradesh, to Jharkhand, to reach to Jharkhand and Bihar, all
लीजिए आगे आगे आगे।
I would like to take, you see, the education. What is the situation of the quality education and promote lifelong learning opportunity to all. And still you can see this, some southern state and western states are more better. And these all Rajasthan, they are not performing. So you can see here.
Sir, slides are not visible.
Ensure availability and sustainable management of water and sanitation, and eastern semi-arid area and almost 68% area of our country where precipitation less than the potential evapotranspiration. And you can see the water stress index in Rajasthan and the part of the Haryana. So you can understand from your own perception the type of problem we are getting in our country. And so energy is becoming a very, very important for integrating physical, biological, and the social aspects. And we have to improve. You can see the wind power huge, and we have to improve this. Biomass energy, a small hydro, cogeneration, waste to energy, this all we have to improve. Promoting sustainable, inclusive, sustainable economic growth, full and productive employment and decent work for all. And in this context, you can see this Haryana, Gujarat, Maharashtra, they are more performing more better than, and some southern states. So we have to identify unsustainable production and consumption. You can see climate change, economic growth, pollution, degradation of air and water, soil, depleting resources, population growth, market distortions, unbalanced markets, limited rationality, and biased information. Prices do not reflect the cost. These are the very, very important. And so that is why I would like to quote here sustainable consumption and vision of Gandhiji. Earth provides enough to satisfy every man needs, but not every man greeds. We can say that there is enough for need of every nation, but not enough for the greed of even one nation. And action climate change will bring the lot of integration of physical, biological, and the social system through National Solar Mission, National Mission of Energy and Efficiency, Sustainable Habitat, Water Mission, Sustaining Himalayan Ecosystem, Greening India, Sustainable Agriculture, and knowledge of climate change. And here you can see this how we have to, in 2014, our position of the renewables, 23%, we have to bring 31%. 11% nuclear, not much, 12% only. 21% gas, 23%. 4% oil, 2% decline, and 41%, and only 32%. Although coal is becoming very, very important here. And so practical solution one can adopt individually. We can adopt our value science, producing the fuel efficient vehicles, reducing the vehicle use, improve energy efficiency in building, develop carbon capture and storage processes, increase solar power, decrease deforestation, improve soil carbon management strategy. And you see this, the data of the United States, particularly as for forest policy, 33% area will be in the plain under the forest. Hardly 33% we have in any state. 66% area should be in the mountain area. So some Himalayan states of northeastern, they are farming better here. So I would like to conclude that sustainable environmental education key to implementing SDG, education for life, education through life, education throughout life. And I would like to quote another Gandhiji, educating a woman, educating a generation. Youth can play population in our country, and from the Mahila Mandal to Jyoti Mandal, all we have to organize our different type of clubs of the maybe in the KNI and some other, particularly for achieving the SDG. I would like to tell you that research and innovation is a very, very important. And in this context also, I would like to tell you that public value of science, we must take care of science in private sector also. Gender equality, particularly in the science, agriculture, engineering, we are lacking, the enough gender participation. Knowledge production as public good. Our governance, we have to improve our governance, and so that we can move in the area of direction which may be considered as sustainable development. I would like to stop here. Thank you, Dr. Tripathi ji and other Principal Sahab and other for giving me opportunity. If you have any question, I will be very happy to answer.
बोलेंगे फिर सर हो गया आप बोलो बोलिए क्लियर
Thank you, sir, for the nice speak. Now I request senior faculty member of Department of English, the VPC, to invite our Honorable Vice Chancellor, Dr. Manoj Dixit, for the presidential address. Dr. VPC, sir.
Thank you, Dr. Rakesh ji. This webinar, which is really for a very relevant topic, and sustainable development is possible only with our innovations. And in modern scenario, when we are getting threat from all the corners, especially from China and Pakistan, so we have to be self-reliant and self-dependent. And for this presidential address, I am going to invite Honorable Vice Chancellor, sir. There has been a phrase in English that as busy as bee. But this, our energetic Vice Chancellor, he is belying this phrase, and we used to say that as busy as Mr. Dixit. In this COVID-19, when we have been in lockdown, but his academic activities and administrative activities, he is absolutely committed for the development of all these things. He is very much busy person. Despite his busyness, he is going somewhere, but on the journey, he is on the way to. But he has given his kind consent for the presidential address, Kamala Nehru Institute, and I individually also very much indebted that he always spares time for such academic activities. So for presidential address, I am now going to invite Honorable Vice Chancellor Sir to address us and to enlighten us for this topic. Sir Manoj Dixit sir, over to you.
Thank you. Thank you, Dr. Singh and other faculty members who are attending this webinar. I would have loved if somebody would have briefed me as to what exactly is the composition of the members and the subject that you're discussing. That would have benefited immensely in roots to the webinar that is going on. However, I pick up my threads from what Dr. VP Singh has just mentioned. We are now in the phase where we have to settle down with Corona. It is not going to be a divorce rather than a marriage, that we have to learn to live with this pandemic, though it will hopefully be not as potently threatening as it is now, and gradually will be treatable with our scientists and doctors finding some solutions to this. Second issue is more important than this, that future courses of action with regard to teaching, with regard to imparting education, evaluating the students, interacting with them is going to change. Now, there are two kinds of changes. One set of change that we are undergoing right now is forced upon us because we are not prepared for this. We were doing our conventional job pretty well. But now, having gone through this process for over 3 months and having realized that the conventional modes have to be compromised now with modern digital modes of education as well as instructions. So the digitalization process has to pick up and will pick up very fast after this Corona impact is moderate. Or rather, I would say the curve is flattened considerably. Then this change process has to start. Wearing a mask will be probably a new normal, where number of other precautions would be taken. The classroom scenarios will drastically change as social media or social distancing or physical distancing, what we say, is going to be the norm of the day. And believe it or not, the doctors believe that at least good 30 to 40 crore people will be affected by Corona. Only then it will be safe for us. Now, under these circumstances, both our classroom, or what you call as our colleges, and the students and the faculty members, all three have to acquire a new set of skills, a new set of tools and methodologies and pedagogies for doing the job that we are doing so for last fairly number of decades. And that is going to be our challenge for now. Digital mode of education will be the mode of education that has to be adopted. In a way that I don't see that when I say digital education, I don't believe that it is going to be forced on us, and it will be less effective than the mode that we are right now following. That may not be the case. The reason is simple, that education in the past years, traditionally education in India was learner-centric, who used to learn by doing themselves. in between, when the Macaulay system came, we started having what you call as spoon feeding, where instructions became important. Learning, compromised learner became center. And instructional compromises made him receive many more marks than what he used to get earlier without causing any serious learning. That is going to happen now, probably with the digital mode of technology, where the boy will be taught through technology but has to learn himself. And that will probably distinguish between a good student, a good learner, and a bad learner. And the learning cost has to be evaluated by the what you call as teachers, or I would say the gurus and the acharyas of today. That is probably going to be the case, and that is precisely what should happen now. I think we should take this opportunity with both hands. This is the time for establishing your own methodology, your own, or rather saying, re-establishing the supremacy and the betterness of your own modes of educational system that you had practiced in the past and have forgotten for last 70 odd years, have to actually resurgence again. You have to cause a resurgence in educational mode and technology and methodology. And that will be the, I think, the best part of, the happiest part of this corona tragedy, I would say, that we will be able to understand the vastness, the greatness, the depth of the systems that we created and practiced. Then we lost somewhere in between, and now have an opportunity to gain, given, of course, courtesy the technology is available. The classrooms are now virtual. They have to be made, of course, more virtual than what they are right now, and also that these classrooms have to be more interactive, and that will happen now, because when the choice, there is a no choice situation, it's a choicelessness now, that we cannot have the normal situation coming anytime soon. We have to think in terms of evaluation systems, alternative evaluation mechanisms also, because exams are probably not going to happen the way they used to happen in the past. So we will have to evolve our mechanisms. And this process of evolution, I'm very happy that KNIPSS has taken this opportunity to keep interacting with the students. I don't think they would have interacted with so many people with the limited resources that we used to have without having this technology at our command. Courtesy this technology, courtesy this coronavirus, and courtesy our lockdowns, and courtesy our zeal to do something, we have now venturing into the virtual world with no limits. Our participants are from everywhere, and that is the happy side of this particular pandemic scenario. I wish KNIPSS, particularly Dr. VP Singh and his team, for a very successful sessions on this medium, and I hope that whatever recommendations and suggestions that you evolve out of this will also be circulated among non-participants, and we also, so that we from the knowledge that gained in this process. Thank you. It's an event, and my best wishes for a successful event and exercise that you are holding. Thank you very much, and God bless you all.
Now we have our principal, and I invite him to give vote of thanks to all our distinguished and eminent speaker and guests. Radhesham sir.
धन्यवाद राकेश जी। ये हमारे लिए प्रसन्नता और एक संतोष का विषय है कि इस महत्वपूर्ण विषय पर इस वेबिनार का आयोजन किया जा रहा है। इस इनाग्रल सेशन में अध्यक्षता करते हुए प्रोफेसर मनोज दीक्षित जी का मैं आभार व्यक्त करता हूं, धन्यवाद व्यक्त करता हूं, जो विद्वत्ता और जनप्रियता के समेकित व्यक्तित्व हैं। वो उन्होंने हमारे लिए जब भी हमने निवेदन किया तो थोड़ा समय संस्थान के लिए निकाला है। मैं आभार और धन्यवाद ज्ञापित करना चाहता हूं प्रोफेसर बलराम सिंह जी का, जिन्होंने यूएसए में रहते हुए भी हमने जब भी निवेदन किया तो उन्होंने कमला नेहरू संस्थान को अपना मानते हुए थोड़ा समय दिया और उसके लिए परिश्रम भी किया उन्होंने। यद्यपि हमारे यहाँ का समय और वहाँ के समय में बड़ा अंतर है, फिर भी समय को उन्होंने अपने तरीके से संस्थान के लिए एडजस्ट किया और हमसे जुड़े रहे। मैं प्रोफेसर बलराम सिंह जी को अपना प्रणाम भी निवेदित करता हूँ और आभार भी व्यक्त करता हूँ। इस सेशन में, इनाग्रल सेशन में डॉक्टर किशोर के श्रीवास्तव जी, जिन्होंने बड़ा इनोवेटिव अपना वक्तव्य दिया और उनके वक्तव्य का परिणाम रहा कि पैनलिस्ट बात हुए उनसे कुछ प्रश्नों को कर सकें, उनसे कुछ समाधान पा सकें। वक्तव्य के माध्यम से दूसरे विचारकों को उत्तेजित करने के लिए भी मैं डॉक्टर श्रीवास्तव का धन्यवाद ज्ञापित करता हूँ, प्रोफेसर श्रीवास्तव का धन्यवाद ज्ञापित करता हूँ। प्रोफेसर आरबी सिंह जी हमारे साथ जुड़े, देश के प्रथम कोटि के विद्वानों में उनका नाम है। संस्थान के निवेदन पर उन्होंने अपना वक्तव्य देना स्वीकारा इस वेबिनार के लिए। वो भी हमारे लिए एक पूंजी है और उसे, उन विचारों को हम संभाल के रखना चाहेंगे। आप सबका एक बार फिर से धन्यवाद, आपका सभी का एक बार फिर से आभार। सस्टेनेबल डेवलपमेंट के बारे में अपने अपने विचार प्रदान करने के लिए, कैसे हम भावी पीढ़ी को कम से कम उतना तो छोड़ के जाएं जितना कि हमने उपभोग किया, उतने ही प्राकृतिक संसाधनों के चयन का अधिकार भावी पीढ़ी को भी है, ना कि हमें। आगे आने वाले और विचारों का स्वागत करता हुआ मैं फिर से एक बार इस उद्घाटन सत्र के जितने भी विचारक हैं, प्रोफेसर मनोज दीक्षित जी, प्रोफेसर बलराम सिंह जी, प्रोफेसर किशोर कुमार श्रीवास्तव जी और प्रोफेसर आरबी सिंह का पुनः धन्यवाद ज्ञापित करता हूँ। संस्थान के सभी, आप सबका धन्यवाद।
धन्यवाद सर। बहुत बहुत धन्यवाद सर। इसके बाद हमारा टेक्निकल सेशन, पेपर प्रेजेंटेशन जो पार्टिसिपेंट है वो शुरू होगा। इट विल बी चेयर्ड बाय डॉक्टर हर्ष विक्रम सिंह एंड डॉक्टर आर्यन सिंह, डिपार्टमेंट ऑफ जूलॉजी। और हमारा पैनल डिस्कशन फिर 12:50 पे 12:50 PM पर फिर से शुरू होगा। थोड़ी देर के लिए we have a break. Thank you and for next meeting at 12:50. Thank you. मैसेज सर अभी आपका काम
सर बोल दीजिए ना, आवाज कर देने से कोई सर प्यार हो जाता है यार।
कैसे आवाज
बीचों बीचों में दो बार
आवाज Dear participants, the technical session has started. Separate link has been sent to all those who are willing to present their paper. So please open the link and log in for the presentation of your paper. The maximum time given for the presentation is 5 minutes for each individual. Dr. Aryan Singh and Dr. Lalit Kumar being chairperson of the technical session.
next
The next session of the conference is going to start within 5 minutes. We invite all the speakers to log in and join us. ऑप्शन दे दो।
एक करके जाएगा सर ऑप्शन।
हाँ शुरू कर
Welcome to second session of this first day of conference. Our next speaker is Professor R.C. Dubey, who is Professor and Head Botany and Microbiology Department, Gurukul Kangri, Haridwar. For his introduction, I would like to invite our senior faculty, Dr. A.K. Singh, Head Department of Zoology. Dr. A.K. sir.
Hello. Hello.
Yes.
Hello.
हाँ आप बोलिए सर.
हाँ hello. Hello.
Yes sir आप बोलिए.
Yes, myself A.K. Singh. I feel great pleasure on this occasion to introduce Professor R.C. Dubey, Head Division of Microbiology and Gurukul Kangri University, Haridwar. Professor R.C. Dubey completed his education from Banaras University, and after completing his PhD degree in 1986, he joined Kumail University as assistant professor in botany and worked there up to 1996. Thereafter, Professor Dubey joined Gurukul Kangri University, Haridwar in 1996 and became professor during 2004. Professor Dubey's field of work is rhizosphere microbiology and phytopathology. Most important thing which I took mention here, he has published 4 important books, that is Vedic Microbiology, Textbook of Biotechnology, and Textbook of Microbiology, which is well known in all over India. Professor Dubey is not simply an eminent botanist, microbiologist, but also a great poet. And a book, Gitayan, is published in 2014. Professor Dubey has published a number of research papers in national and international journals, and also he has been member of different eminent academic bodies like Indian Botanical Society and Indian Science Congress. At present, Professor Dubey is working at Gurukul Kangri University, Haridwar, and at this moment, I welcome. He has been my senior colleague at Banaras University also. So I welcome on the behalf of Kamala Institute, Professor Dubey, and invite him to provide his valuable lecture to all of us. Thank you. Hello.
Hello. Hello.
Yes sir. Yes sir आप बोल सकते.
I am coming to the point and going to share the screen.
Yes sir, you can.
It is disabled. Kindly make it enable.
Just a minute. Yeah, just. Yes sir. Now you can share your slides. Yes sir, you can see it.
It is visible to all of you.
Yeah.
The sound is vibrating. May I start? Hello? Hello? Hello?
Hello, sir. Yes, yes, sir. You can start your lecture, sir.
Okay. Thank you, Dr. A.K. Singh, Head of the Department of Botany, Dr. Rakesh Pandey, Head of the Department of Zoology, and the organizing team of Kamala Nehru Institute of Physical and Social Sciences, Sultanpur. Thank you so much to all of you for inviting me to be a speaker of this webinar, which has been started since today. So I extend my thanks to all of you. I have selected a topic on which I am unknown. So with your help and the help of our ancient text, I want to give some of the most important information regarding the Vedic microbiology and how this Vedic microbiology, the themes of the Vedas, the principles of the Vedas can be helpful in warding off the diseases like COVID and other diseases also. My dear friends, we have forgotten our treasure of knowledge. This is several thousand years old script, which is the oldest one in the whole world. And still, we Indian, we teacher, we students do not know that these scientific ethos have been mentioned in our Vedic literature. So with this view, I want to make you available some very small number of the slok mantra with the help of this slide and webinar. And I do hope that all of you will enjoy with this new type of information. So starting with the COVID. All of us, we know that this dangerous virus has come from bat eater country, that is China. So this disease is known as China-oriented virus disease 2019. But China has changed its name as COVID, the coronavirus disease 2019. I don't know why does he is hiding his name. So it is said that the wet market of Wuhan city is the hot spot where the bats are being sold and they are eaten raw and where the viruses reside. So nowadays, including India, 188 countries are facing this problem. So this coronavirus is not a new virus. It is already known. This virus is already known. It is found in all type of bat species which are found globally. So the viruses found in all the bats, they are not so dangerous because there are some genetic changes. And due to these genetic changes, the viruses may or may not be harmful, may or may not be virulent. So it came into existence for the first time in 1933, but in 1960s, it was known as human coronavirus because it caused disease in the respiratory system. In 1975, this virus was classified into a family Coronaviridae by International Committee on Taxonomy of Viruses. Then further, this family was divided into two families, coronaviruses and toroviruses. The coronaviruses has been put into two types of viruses, alpha viruses and beta viruses. My dear friend, the COVID belongs to beta viruses, which is very much dangerous to human society. The structure of this virus is such that the capsid is covered by an envelope, and this envelope is of host origin. It is of mammalian origin. And there are certain proteins, 4 important proteins, surface protein 1 and 2, nucleocapsid protein 1, the matrix protein, envelope protein, and single-stranded RNA as genetic material. What is the uniqueness in this virus? Uniqueness is that the surface protein 1 consists of angiostatin covering enzyme 2, which is abbreviated as ACE2. And when this enzyme ACE2 becomes complementary to the ACE2 of our gut cells, of our respiratory tract cells, then this virus becomes compatible to our system and it enters in the respiratory tract, and it does. People are the opinion that in RNA containing viruses, there is possibility of 198 variants. So my dear friend, this is one variant. It means that 197 variants are possible because it is RNA containing viruses, where uracil is present in genetic material. And in DNA viruses, instead of uracil, there is a thymine. So uracil very frequently can grow a genetic cyst. So through genetic drift or genetic cyst, variations are brought out into the coronavirus genome sequence. That's why it becomes, it takes a dangerous shape. Now, when this virus is present in our system, then people start sneezing, or they cough, they discharge their cough, they discharge their sputum. So through sneeze, the coronaviral particles are transmitted from one person to other person. The size of droplet varies. So therefore, this distance traveled by the viral particle also varies. This distance traveled by this aerosol may be 2 to 14 feet. And the direction of sneeze also decides the distance, how much it will travel. The contaminated droplets, when get settled on others, but the droplets are heavy, usually fall on this surface and do not travel for a longer distance. So the size and weight of the droplet decide how much distance it will travel. That's why always it is broadcasted, and always we people are informed that one should maintain a social distance of at least 3 feet or 6 feet. But when a person speaks loudly, so they also discharge the droplet to a longer distance. Therefore, when two people are talking, they should not talk very loudly. They must talk slowly. Uncovered sneeze discharge a droplet traveling up to 15 feet. That's why it is recommended that one must use the mask. So once a person is sneezing, the mask will protect, not to allow the droplet to travel in the air for a longer distance. And even the sputum and saliva, they carry the viral particle large in number. This is today's figure. There was a time when the virus, the infected person, to come to the level of 10,000, it takes 75 days. And now at the end, in about 6 or 7 days, this number is doubling. So it takes 7 days from to increase the number from 4 lakh to 5 lakh. So number of patients are increasing day by day, and there is no end. So what we have to do, we have to live in such a society where there are challenges. So we have to develop our habit to live with coronavirus. The coronavirus never will be eliminated from the human society. Once it has entered in our society, it will live for a longer life. It will travel from one person to other person. In this way, in human society, it will always be traveling. So what we have to do? We have to adopt our Indian culture, which we have forgotten. We have never response just handshake or any other measures to give respect to our elders, because the hand carries a large number of the virus particle, bacterial particle, fungal spores, and other type of germs. So one must say, just stop this is some sound is coming. I don't know where from. Anyway, so one must make a distance between two persons so that through this saliva, through this sneeze, the viral particle should not travel from one person to other person. One must say Namaste. Namaste is Anjali Mudra or Pranam Ashan. Namaste means I bow to the divine in you, because in each and every person, the supreme divine exists. So we pay a respect to the divinity which is present in the human. That's why Namaste means Namaste. I bow down to you. You means the divine power which is present in you, not a person. So there is a lot of measure which has been given. Nowadays, stay at home. Unnecessarily, we should not come out. Otherwise, there would be chances of receiving the inocula of the viruses. We must maintain a distance of 6 to 15 feet, not 6, because in society, some of the person speak very loudly and they sneeze very forcibly. It is the force that decides the distance of the traveling of the droplet. One must use mask and frequently use 70% alcohol-based sanitizer, not others, because since hundreds of years, ethanols have been used in the microbiology laboratory. That's why I am recommending you. My dear friends, the use of mask is not new. Masks have been used about 3,005 years ago. Maybe that during the start of the Jainism, there would have been a spread of the pandemic. So it is the pandemic that has occurred in the past also. Don't think that this pandemic has come for the first time and we are facing the problem. Several pandemics have occurred in the past also. I will give some of the example. So this Jainism philosophy also say the use of the mask, leading the vegetarian life, maintaining social distance, adopting quarantine, and putting in the isolation. According to Dr. Soumya Swaminathan, who is the representative of India in the World Health Organization, she says that coronavirus cannot be eliminated up to 5 years, even after vaccination. Hence, we have to live with COVID-19 with certain precautions. And what are those precautions? Which I told earlier. So people have claimed, including India, that they have discovered vaccine, but so far this vaccine has not come in the market. Even mass production has not been started. So what we have to do? We know that vaccine is within you. We don't think this vaccine is coming outside, because the person who have recovered from the infection of the coronavirus, they have developed their own immunity. It means that vaccine is in your intestine. Vaccine is present within you. What you have to do to secrete more and more vaccine, we have to perform daily yoga, we have to exercise or play any game. We must use vegetarian food, sattu, khichdi or any other local food material. We must take one fruit daily at least, preferably juicy fruit like orange. We must consume spinach, radish, chenopodium is green vegetable daily. They contain lot of mineral. The use of dal, good jaggery. Good or jaggery must be used, not the sugar. Pure vegetable oil like mustard oil, linseed, groundnut should be consumed, not the refined oil. Tulsi or Indian wassail and other medicinal drink must be taken each and every day. And pure milk-based product like lassi, chach, curd, and milk should be taken each and every day. My dear friends, this will help you increasing and maintaining your immune power. No, human microbiome is such that our system externally as well as internally bears a lot of microorganisms, and these microorganisms, they help in developing the immunity. For example, 100 species of microbe lives on the skin, 1,000 species in the intestine, generally 100 trillion bacteria lives in human intestine, and 600 on the chest here. So in this way, large number of microorganisms, they are present in our system, either outside or inside. So we have to adopt the Vedic philosophy just to lead a longer life. What is that? We must follow the brahmacharya, celibatism, because we have been taught as brahmacharya tapsa deva mrityum upagnata. So brahmacharya must be followed by each and everyone. We must perform yoga, exercise, daily agnihotra, oblation of the medicinal plant in the fire. We must keep our environment clean, and we must take each and every day a balanced diet, because this balanced diet will help in increasing our immunity. How to enhance our immunity? There are 3 ways: by performing yajna therapy. Yajna is known as therapy. Agnihotra is now known as therapy. Natural remedies, God has given, the nature has given all type of plant all around you where we are living. So one must use natural remedies and yoga therapy. By performing all these, we can enhance our immunity, immune power. Now, we know our kitchen is also having a large number of these spices, the medicinal product, and we have forgotten all those like ginger, fenugreek, garlic, clove, black cumin, cinnamon, saffron, the black pepper, long pepper, all these are having immune power. So one must use these in the form of kadha. So in the kadha, what we have to do? We must use lemongrass or lemon juice, the ginger means adrak, black pepper, kali mirch, long pepper, pipli, cinnamon, dalchini, and tulsi. And the dose can be controlled. It can be formulated according to the choice. You know how our rishis, our sages, they live a long life. Samved says, Pavamaniryo adhetri rishivi sambratam risham tasmai saraswati duhe shiram sarpim udutkam. Means the sages, our rishis, live long life by consuming daily pure milk, pure ghee, pure honey, and pure water. My dear friend, keep in mind, our rishis, they were not having any type of very flexible life. They were leading their life by their own way, and they lead a longer life only by consuming pure milk, pure ghee, pure honey, and pure water. This has been said in Samveda. Now, as far as water purification is concerned, in our culture, there was a system. Still, it exists in the villages, that we throw a small coin of copper in the river, like river Ganga, like river Yamuna, all these 7 holy rivers of India. But we have forgotten all these because silver coin is not available. What does silver coin do? Silver coin precipitates the organic material, the solids material, suspended particle down. Number 1, it kills the diarrheogenic bacteria that lives in the water, and at that time Ganga water was drunken as such a raw. Still, you know, I am sitting in Haridwar, and I know people coming from throughout the world, they come here, they have faith in the Ganga water, and they consume raw water as such. So this copper coin kills the pathogenic bacteria that causes diarrhea if it is living there. As you know that this social distancing is not a new term. This is a very old term. In Skanda Puran, there are 10 slok, and in 10 slok, 2 important diseases have been mentioned. One disease is viral disease called as mump, and this mump was known as Galgand or Gandamala. And second disease is smallpox. Smallpox, you know, common name in Hindi, North India, known as Shitala. And temples have been built up in different part of the country with different name. So in the for months it has been written Gandamala Galganda Graha Ghora E Chante Darunam Nimam. It means if there is a Galgand, means mumps. Mumps disease has been described in Skanda Puran. In Ved also it has been described. And for social distancing, for smallpox, it has been told as Bisfotak Bhayam Ghoram Grihat Tasana Jayate. When this smallpox has taken the very bursting phase, one should not go to others' home. What does it mean? One must maintain this social distance. one should not go to other home because it has taken the very bad shape, very dangerous shape. So Visfotak bhayam ghoram grihe tasya na jayate. We should not go to the home of other where the smallpox has taken very bursting phase. So this is the mention of mumps or Gandamala in Atharveda. Apachitam lohini nam Krishna mate sustam munir devas mulena sarva vidyamita aham. Apachitam means Gandamal. Gandamal means the mumps. And this mumps or Gandamal is caused by paramyxoviruses, which is called as minor sense RNA containing virus. And genetically, generic name is rubella virus. So when rubella virus causes disease, all around of our gland, there are parotid gland, then infected person cannot swallow the saliva, cannot drink the water, cannot eat the food. So such infected person can die within a few days. So there is a medicine. Medicine has also been mentioned in Atharveda. Muner devas mulena. By using the root of Butria monosperma. Root of Butria monosperma can be taken out. It be crushed properly. And if you are applying the paste on both sides of the gallagund or where there are parotid gland, so this will be cured naturally. We have also forgotten the Vedic technology performing Agnihotra. So I recommend that one should perform Agnihotra at least once in a week. By doing this, we put the medicinal plant in the fire. And what has happened? There is an increase in temperature. The temperature goes to about 12,000, 1,200 to 1,300 temperature. At this temperature, when we will put the oblation along with the ghee, then at different temperature, there will be burning of those medicinal plant. And smoke will come out. And these smoke will spread all around the environment. And these smokes, most of them, they are having the antibacterial, antifungal, antimicrobial property. So that smoke will purify our environment. It will kill microorganisms. It will increase the mental health of the Agnihotra performers. It will eliminate the ghost problem. If there is a problem of a ghost in the house of anybody, you know that in this oblation, Google gum powder of the Google gum is used. So once we are using the Google gum powder, the problem of ghost will be eliminated, and ashes can be used as water purifier. My dear friend, all these are the techniques which have been done during the Vedic period in India. Now, it has also been mentioned that we should sit in the sun. We should take sun bath, because what will happen? Atharveda says, Udyan aditya krimin hantu nimorchana hantu rashmibi. Once you are sitting in the sunlight only in morning and evening, not in a day, for about 20 minutes, then what will happen? Krimin hantu, all the krimi, all the germs present on the skin will die. So what is uniqueness in the sun and evening sun, in the morning and evening sun rays? In the morning sun rays and evening sun rays appears red in color. And in red in color, where from this red color comes? Red color is only because of the presence of infrared. So in the presence of infrared, our skin will produce more and more vitamin D3. There would be synthesis of D3. In the presence of vitamin D3, there would be absorption of calcium. Because calcium is absorbed only due to presence of vitamin D3. And calcium combines with vitamin 3, help in combining with our protein present in the heart muscles and all the muscles. So this is called as calmodulin. And calmodulin is a part of our heart muscle as well as liver muscle. My dear friend, when there would be deficiency of calcium in our heart muscle or caudulin or the liver, the people will suffer from heart disease like heart attack or yellowishness of the body, means jaundice. So sitting in the sun rays, we can cure this type of disease automatically. This our Atharveda says. The external part of our body consists of a large number of microorganisms. For example, you will find 1,000 bacteria per centimeter square on hand. So we should not perform hand sex. Forehead contains 100 million bacteria. Scalp contains 1 million bacteria. Nose mucus 10 million bacteria. Armpit contains 10 million. Saliva 100 million bacteria per centimeter square. Fecal material 100 million bacteria per centimeter square. So our life is possible only because of microorganisms. In other words, and some of the microorganisms, they become very dangerous if our immunity is very weak. So what we have to do, we don't kiss the others. Unnecessarily don't kiss the other person. Because other person, if they have infection like oral candidiasis or herpes simplex virus, this will spread into the healthy one. So this is the first lesson which I want to give to all of you, all the participants. Don't kiss the others. Then prevention of microbial diseases can be done by changing our habit. We have bad habit. So we have to change our bad habit. Our spiritual script says, Lavanam Benjanam Chaiva, Dhatam Tailam Tathaiva cha, Leyam Peyam cha Bibdam, Hasdattam na Bhakshet, which means salt, ghee, oil, food, vegetable or milk or food material should not be taken with your bare hand. So hand must be washed properly, or it should be taken out only by using the spoon, because the hand contains a lot of microorganisms. I told you 1,000 bacteria per centimeter square, they are found only on hand. Unnecessarily, we should not put our fingers into eyes, nose, ears. These are the gateway of the microbe. The microbe will enter inside our body. Anatura swani khani na isped nimittatah. Don't touch your own organ such as eyes, nose, ears without a reason. And Atharveda also says, yo akshau parasarpati, yo nase parasarpati, datam yo madhyam gatim tam krimim jamahamyam. It means akshau means eyes, naso means nose, dat means mouth and teeth. This should not be touched unnecessarily. And you know, the small children, the youngster, all they have bad habit. The habit of putting their nose, finger in the nose, or cutting their nail with the teeth. Washing of hand, feet and mouth should be done before eating the food material. Our script says, Hasta pade mukham chaiva panchadre bhojanam charet. Na prakshalit pani pado bhunjit. Wash your hand, feet and mouth before you eat and after you eat. Before and after eating, one must wash the hand and mouth, even the feet also. Because during those days, people used to take meal food material only sitting on the ground. Now that time there was no dining table. Avoid the use of wet cloth. Na adram paridadhita. Don't wear the wet cloth, because the wet cloth, now this winter season, this rainy season has come. Due to presence of moisture, there would be transfer of ringworm caused by the fungi, dangerous fungi, Taenia corporis, caused by Trichophyta rubrum, the sporotrichosis, sporotrichium, this causes a disease. So we should not use the wet cloth. We must avoid use of others' cloth. Na dharayet parasva snanam vastram kadachana. Don't use the cloth used by other for drying your body after a bath. And tatha na anya dhritam dharyam. Don't wear cloth worn by the other. You know, if you are using others' cloth, then certainly there will be chance of transfer of microorganism, dangerous microbe on the head. If you are using the socks of other person, there may be chance of development of Taenia pedis, and the skin will start decaying, and hardly such type of disease can be cured. I say hardly they can be cured, because immune system becomes very weak. We must avoid the own used cloth. Again and again, we are using our own cloth. So one must avoid by to use our own cloth again. Apabrijjan na snato datrambar panibhi. Don't use cloth already worn by you and dry yourself after a bath. Na aprakshalitam purvadhritam basanam vibriyad. Clothes once old should not be worn again before washing. So after each and every use, the clothes must be washed. Anyadeva bhavadvasa saniye narottama. Use different cloths while sleeping, while going out, while performing puja. Same cloth cannot be used while doing three different type of work: sleeping, going out, and performing puja. Different type of cloth must be used by us. There are certain precautionary measures as well. What are those? Chita dhuma sevane sarve varna snanam achareyu. Bamne sham shuddhi karmane cha. Take a bath on return from the place of cremation. You know, this is the last stage of the ritual which is done in a life of a person. The cremation. When a person is returning from Smashan Ghat, from the place of cremation, we should not enter inside our home. We must take bath first, either outside of the home or in the river where from we are returning. Only then we should enter our home, because there is a chances of carrying a large number of microorganisms. So these things have been mentioned in our old Indian literature. Take a bath after every haircut. Even you are coming from the salon or beauty parlor, one must take a bath and wash all your cloth into the pure water. Otherwise, the barber uses the same contaminated cloth again and again. So there is a chance of transmitting the microorganism from the infected one to healthy one. So my dear friends, all these are the very important informations that have been mentioned our Vedic literature, our Puranic literature, in the Smriti and other different type of books. I think and hope that we have forgotten these things which have already been told by our rishis, our sages. 1,000 years ago, it means that during those times also several epidemics have come. Don't think this is the first epidemic. In the past also, most of the human society has been vanished. Only 150 million people died of only smallpox in the European country in 1 year. During that time, the population was very low. Just to imagine, 150 million people died in 1 year only because of the smallpox. So microbes are very dangerous. Microbes are very helpful. They are a friend. Without microbe, our life is not possible. Microbes are living inside and outside of our body. So we have to live with the coronavirus, with other viruses which are present in the society. We cannot eliminate all the microbes from our body, from the society. So thank you very much for listening me, and namaste to all of you. If you have any question, please you can ask me. Thank you.
Thank you, sir, for delivering such a nice lecture. I call upon, I request Dr. A.K. Singh to say a few words regarding this topic. A.K. Singh.
Hello. Hello, sir. Good morning to you. I express my deep sense of gratitude for your nice information and providing me an occasion in respect to the highlight the Vedic microbiology, role of Vedic microbiology in our life. No doubt, you have given a extensive information regarding the development of coronavirus, mode of infection, symptoms of the disease, and with preventive measures. No doubt, the Vedic philosophy, Vedic principles, and Vedic technology have been fruitful as a natural therapy and keep our healthy life. Thank you, sir, very much for your presentation and our kind cooperation. The Institute is grateful to all of you. Thank you, sir.
Thank you, all of you also.
Thank you, sir. We have 5 minutes. We can have an interactive session. Any panelist or participant wish to ask a question on is invited. Welcome. And after that, we will proceed to the next lecture. And I will invite Dr. Sunil Kateria from JNA to deliver his next talk.
कोई क्वेश्चन नहीं आगे बढ़ो नहीं है सर
Okay. Yes, I expect that there is no question. So thank you very much for delivering such a nice lecture. And it was really very thoughtful that how we have changed ourselves from our ancient civilization and ancient culture. The Vedic philosophy, the Vedic era where we live, was everything so we proceed to the next. Dr. Ajay Vikram Singh has joined us from Germany, and I call upon him. Ajay Vikram Singh, is he ready?
Yes, sir, I'm ready. So can I start now?
Yeah, you can. You can. After that, there was some confusion because some time back, I requested Dr. Sunil Kateria to present his talk, but as per his schedule, I request him to be prepared for his talk. So I invite Dr. Ajay Vikram Singh to be here and proceed with his lecture. Dr. Ajay Vikram Singh is working in Max Planck Institute, Germany. Ajay Vikram Singh, you are most welcome to deliver your talk. Thank you very much, and you can proceed.
Yes, thank you, sir, for the introduction. So I will start now, and basically this talk will be little technical, so I want to apologize in advance for that. So this is a talk recently I delivered last week. I work at German Federal Institute, Germany as a senior scientist, and basically we are working in the area of advanced testing strategies for inhalation or anything which enters in our lung. It could be toxic material, it could be infectious material like this coronavirus. So I mean, this talk is in the context, so you take this talk in context with the coronavirus. So inhalation toxicology is basically our inhalation itself is a gateway of many different types of events that happen to us daily to daily life. So few example I'm presenting here. The upper left corner here, you can see that every year almost 7 million people around the world die because of the air pollution. They inhale a lot of these toxic air with the different kinds of characterized and uncharacterized material across the world. And this lower left panel, you might be aware of that there is a increasing infection rate in India also. Almost every day 16,000 people are getting infected, and the main cause of infection is basically the aerial transmission of these infectious droplets, which have these tiny nanoparticle like coronaviruses. So basically coronaviruses, you can say that they are organic genetic nanomaterials. And right hand side, there are two pictures. Basically they represents the problems more in developed countries like in Europe, in United States. So 3D printing is getting very popular. Here in Germany, every home, these kids have these 3D printers. So if they want a toy, they can print their favorite toy. And also to get rid of the cigarette, Rakesh sir, everybody is listening. I'm quite audible, yes?
Yeah, you are.
Okay. So in European Union, they introduced this electronic cigarette. Electronic cigarette is basically what they do when we, okay, so first quickly I will explain you. When we smoke normally, we burn the cigarette with matchstick, and the temperature of that red zone, when we just take the cigarette in our mouth and apply the air inside, the temperature goes 900 degrees centigrade. And that higher degree of temperature converts this nicotine in benzpyrene and many severely toxic chemicals. Those cause cancers, those cause mutagenicity inside our body. So in Europe and United States, basically they introduced e-cigarette. E-cigarette means there is an electronic coil, you insert the cigarette and you burn at lower temperature. So what happens, toxicity is significantly reduced. And as a modern expansion of this brand came into many different types of products as e-liquids, electronic liquids. So basically what they do, they replace the nicotine with some organic liquid, which when you inhale inside, it gives you it satisfies your mind like cigarette. So these are some of the toxic scenario or infectious scenario that are involved in our inhalation pathways. Before I go, just I want to quickly show that to also the understanding is very important for this, because not everything what we inhale goes to the deeper lung or creates problem. So on the left hand side, you see three different regions: extrathoracic, which is basically our oral cavity, and this vocal cord, larynx and pharynx basically. And then when you go little deeper, you see these trachea, tracheoles and bronchi. And the most important is the pulmonary region, which is involved in case of this coronavirus. Basically, this tiny virus migrates not from upper respiratory tract, but goes to this air liquid interface. And what happens once it reaches to air liquid interface, it gets exchanged with the circulatory system and reaches to blood and infects everything. So this is a very important thing to understand exact the physiology of this. And also this mysterious coronavirus is known to found in every part. Not initially it was considered that it infects majorly to the lungs and creates pneumonia, and this is the reason of the major death. But if you will see closely, the sensory organs, they are connected internally. All the sensory organs are connected basically through these airways or tiny tube-like structure inside our. For example, if something you will inhale, there is a high probability it will reach to the brain, eyes, upper and lower lining of oral epithelium, ears. So same thing happens with corona also. Since this is airborne, once we inhale, slowly it reaches to every organ and creates problem. And this is the reason in severely or elderly, immunocompromised people or elderly people, the death is 100%, mortality is 100%. Okay, on this right hand side, this is rather a complex diagram. I will not go in much. So basically it shows the fate of coronavirus when it reaches to the lung. So coronavirus, as I explained, this is here equivalent to these nanoparticles. So once it reaches to the air liquid interface where it makes, where it get exchanged with the blood system, a lot of things happen at that point. One thing is that macrophages are the policemen of our body. These immune cells, you can call it policemen, and they keep wandering all over the body. And whatever infectious or toxic nanoparticle they get, they inhaled them, sorry, they engulf them, and they take them to the clearance port or debris port, which is our lymph node or reticuloendothelial system. And some of these coronavirus reach to the RBCs, they infect RBCs also. So this complex picture shows there are different fates of this coronavirus. Once it reaches, maybe it goes to circulation and interact with the viruses, may not be. In that case, it will just eaten by the macrophages or policemen of the body and remain there and get dumped. And basically the clustering or the how well-being is our respiratory tract also matters a lot in this infection. For example, here are 3 scenarios: impaction, sedimentation, and diffusion. So if the coronavirus get aggregated like nanoparticles, they get deposited into the upper and lower respiratory tract. And this is what happening in most of the Asian countries where pollution rate is high. So what happens during normal inhalation, the virus goes and it get mixed up with the pollutant and they become larger particles. And larger particles, you know that as per Einstein's law, their sedimentation rate is high and their diffusivity is less. Contrary, in the clean air in the United States, you can say, and also in the Europe regions where the air is pretty clean, what happens? These nanoparticle like coronaviruses, they enter into the deeper lung through the rapid diffusion, and they are less prone to the gravity. And depending on the particle size where they get deposited, you can say the head region, if they get aggregated in form of 10 micron to 100 micron particle, they get deposited in these trachea and bronchiole if the size is between 10 micron to 100 nanometer, and they go very deep into the lung and get exchanged with the blood if the particle size is 1 micron to 10 nanometer. So the size, the air quality, these things also matter where this virus is going to interact and bind. So this, I would like to skip this slide because this is our regulatory slide that in developed country, basically they are trying to remove these animal testing or in vivo testing following this 3R principle, which means reduce, refine, and replace the animal testing. So we are developing as a scientist, for example, my job is to develop these advanced strategies to find out the equivalent in vitro system where we can examine or test these virus or nanomaterials or these smoking products and say that, okay, their effect is equivalent to the in vivo system or inside the body system when they are taken. And this is a very difficult because developing in vitro system exactly copying the in vivo scenario is very difficult. So few slides I will share in coming presentation. So what we are doing, like when we go outside, so there is a air pollution, maybe somebody has sneezed, droplets have spread into the air, and this flowing air goes inside our nose. So to mimic that scenario, sorry. So this is a system that we have developed. So what we do, it's like a nebulizer. We put the cells or tissues pieces inside, and we put this coronaviruses laden thing like aerosol. So what happens, like we inhale, it get deposited into the individual tissues or the lung cells, those are present there. So we are basically upgrading the in vitro testing strategies. And those classical kind of submerged culture, if some of you might be doing in cell culture or tissue culture there, you might be aware of that. We take the pieces of tissues and we put into feeding media where they grow normally. Contrary this, we are actually developing not these water-filled chambers, but we have very advanced, as I shown in this video, advanced culture system where we can directly expose them to the aerosol containing these corona to understand how they enter into the cell. All right, so and this is just a quick overview because as I said that this talk could be little more technical and out of your hand. So because I delivered it last week and I couldn't get time to convert in context with the sustainability and other issues that that is the main theme of this particular conference. So some of example here I'm showing that 2D versus 3D culture system. So in the lower panel, you see that basically these are the two-dimensional cell culture. So normal practice is that you take the cells and put in a plastic plate and flood them with the media, and these cells grow on this plastic like tiles, you know. But this is not how our cells remain in our body. Our body is basically a viscoelastic soft material. And inside, other than the surface skin cells, if you will go deep, you will find that any cell inside the deep, they are exposed to the three-dimensional molecular distribution of these signals. And because of that, they grow in 3 dimension, not in 2 dimension when we put them into the plates, such kind of plastic plates. So inspired by these 3D cultures, some of the recent advances are spheroids. We can generate a sphere-like 3D like of culture. We can make organ type culture. We can take organ slices, as I told you that, and you can test directly the infection and other scenarios on these transfer system. And other important thing is that in our body, everything is connected through the blood flow or network or circulatory system. So we are also connecting these different cellular compartment with the microfluidic channels, which is a kind of simulating our body condition. And these are some slides that we are doing in our lab. So basically, I will just skip because we use different molecular marker to test them regularly, that they are doing their job like they do inside the body. So basically, we take the endothelial marker, the barrier function marker, and different kind of protein marker. We also routinely inspect them through the microscope. And these are some more stuff related with the 3D cell model. Okay, so what we are doing currently, so I'm showing you a few slides of advanced technologies that we are developing here. These are called organs on chip. So basically our different organs, vital organs such as the liver, kidney, heart, brain, lungs, all these skin, all these organs, basically we can simulate or we can manufacture them on a small chip. And we can test these drug molecules as infectious material or nanoparticle, how they affect in real scenario inside our body. So in this particular slide, it's very complex to understand here. So I'm just showing one by one. So on this chip, basically, we can copy exactly the function, structure, and physiology of the organs inside our body. So this left-hand side, this where my mouse marking here. So this is the chip and this is equivalent organ. So equivalent organ here, I'm trying to simulate or copy the lung. So lung has a diaphragm that makes this breathing motion, expansion and contraction regularly. And it has a air-filled lung, and outside this lung, there are the blood capillaries. So there is an air-liquid interface. So same thing we can copy by culturing the cells. As you can see that we can culture the lung cells here, and here we can culture the blood vessel cells, and these both are separated by this thin porous layer. And this is the real-time imaging and staining of these tissue, and you can find this green with the lung layer and this red with the blood layer. And also, like inside our body, they have this different kind of tight junctions. So this lung system or air side in the lung, basically they have the cells in a very tight control or combination. So they don't allow other than air to go outside this. So such kind of expression we see on this system. And like the diaphragm moves up and down, we can also apply the pressure into these side chambers of these chip, and they will expand and contract in a breathing motion. And these are some complex characteristics that have been studied by the Donald Ingber lab in MIT. So these are basically tiny chip-like systems. Those are called as the organ on chip. So maybe it is more, you can understand more in this video that is called lung on a chip device. So these are like the SIM card size of devices which have these fluidic channels. Those have the air channels. So fluidic channel, we can flow the blood. And as you know that lung expands and contracts regularly, and the oxygen is transferred from capillary to the bloodstream through these small, tiny balloon-like structures. Those are called as the alveoli. So to copy this thing into chip format, you can see into the cross section, we can generate two chambers, upper like a lung, lower like a blood, and these two chambers will be separated by this porous membrane, you can see. So this is a kind of boundary between two, our air liquid interface, and we can culture the cells in both the chambers, and this arrangement is like the same into the lung, it happens. And on the side channels, we can apply the vacuum, and it will make contraction relaxation like the abdomen does. So it's a quasi lung, tiny lung, micro lung, you can say, in a chip format. And we can carry out the important experiments to replace the animal testing, because they have all the ingredients that is found inside our body. So inspired from this, basically these lung on chip devices, I want to quickly discuss that they have this problem of that they need a lot of expensive instrument and arrangement. So I was working 4 years in Max Planck Institute of Micro Nano Robotics, and my job was to design those tiny slug snail-like soft robots, those crawls on the surface. So two examples I'm presenting here. One on the left-hand side is the light-fueled soft robots, and on the right-hand side, these are wireless magnetic soft robots. So basically, light-fueled robots are thin sheets of the transparent polymer films, and these polymer films are called as liquid crystal gel. When you shine the light, they are very sensitive, are responsive to the light, and their crystal arrangement get disturbed, and they make this bending motion. Like the bending motion is done by the different creatures, you know, are the larvae which eat those plant things. And right hand side is the example is the soft jellyfish like robot. So this is a jellyfish. This is not the real jellyfish that is found in ocean or in the aquarium, but these are the magnetic soft jellyfish. And these jellyfish have the magnetic material, and when you apply the magnetic field, for example, alternating magnetic field or electromagnetic coil we used in Max Planck, they make this very interesting motion you will see in this video. So you will be amazed that these are synthetic structures. These are not the real jellyfish, but these are synthetic structure made up of the soft polymers, and you can see that they move like a jellyfish. And Metin and our collaborators in Max Planck, they did very detailed analysis of these jellyfish, and basically they work like they can carry small particles, and they can swim from the bottom to top of the ocean. For example, this is more clearly seen when, so this is not jellyfish, this is a robot. I want to tell you. So the field is so advanced that you have very precise control over the motion, rhythm, frequency, and periodicity of the soft structure. So, and it is just controlled by the magnetic field. And on the right hand side, you see one example, that light fueled soft robot. You shine the light, they will bend, and they will make this versatile motion. You switch off the light, they will become flat. And by switching on and off, you can make very nice jumping, crawling. So what is the idea of discussing this here? So what we want to do, we want to capture this technology and take the next level of the heart and lung, because these two structures, when we are born until our death, these two structures keep beating throughout our life, you know. And such kind of beating or rhythmic motion, which was so into the organ on chip devices, is not feasible because those are very tiny. And if you will see the lung and heart, they are the big organs. So we need something more robust and peculiar system and soft system that can support. And so we are basically developing here a tabletop or table on lung kind of devices for corona infection study, for nanomaterial infection study. And the idea is that we are 3D printing the different parts of the heart. You know that our heart is segmented. Our right heart has 3 segments, left has the 2 segments. So each segment we will 3D print of these soft supportive materials, and we will put some QCM sensors that can capture the images and concentration of these viruses or particles entering in each part of the lung. And since these are synthetic structures, so we will take the cells and tissues that exist inside our lung or heart to culture in a tiny devices called as the MEMS, micro electromechanical system. And in this way, we will make each region of these organs inside our body in a real scenario. So this is something very interesting. And some of the work in progress in my lab here is in, the different aspect of this project, how to control the dose, how to measure the dose, because single point optimization characterization takes almost 3 to 6 months. You know, 2-3 students are working tirelessly on such projects. So some of these are work in progress. And yeah, with this, I would like to wind up my talk. And I'm looking forward any questions if you have. Hello.
And we know you are a very good orator.
Thank you, sir.
The session is open for discussion. I think you have very good lecture. You delivered a nanoparticle, the aerosol. There are so many questions in my mind, and I hope that all the panelists, all the participants must be having such questions, such queries in their mind. Taking this opportunity, I invite all the participants to ask and have some healthy discussion on questions.
Yeah. So Rakesh, and I think the talk was wonderful and delivered by Dr. Ajay. So I have got a couple of questions. Not question, but it's kind of a query in terms of COVID diagnostic kit. So if you look at today RT-PCR based kits that we have, the sensitivity or accuracy level is not that very high.
Yes.
There are a lot of cases are false positive.
Yes.
So in nano robotics that you are working on, can you think of developing a primer which is labeled with the fluorescent material, and if it is transformed into the nano size, so probably there may be a chance to have RT-PCR techniques more accurate in terms of analyzing the samples. This is one. And second one was like, in my collaboration, we had written a project in Indo-US program where we are trying to propose antibody based kit, this coronavirus testing kit, where we are thinking to develop very sensitive technique where it can be mimicked just like a pregnancy kit. You just add a drop of swab and the result will be instantaneous. So there we are trying to exploit this nanotechnology. So do you have some thoughts along these two, questions or thought process?
Yeah, so I think it's a very good questions, and I think sensitivity matters a lot to give this false positive or the real diagnosis about this corona. And I think this was one of the mysterious scenario which ran all over the world. I mean, whether it is America, United States, here in Germany, for example, we are working in very closely with the Charité Hospital in Berlin. And the same issue, because the problem is this coronavirus, which is like nanoparticle, as I mentioned, the antibody-based test can be developed only after infection. The patient has a significant titer value of the viruses inside the body, and against that, the immune cells make a very alarming signal, and they raise significant amount of immunoglobulin M, IgM, and then you can extract and test them. So that is, I mean, something different and very robust test that can replace this PCR. But again, that is a time taking. And from the swab-based test and sensitivity, I think it depends on different readouts. So like you mentioned that maybe fluorescent particle could be helpful to make it more sensitive, and take a little swab and just scratch it, and even presence of few particles will make the equivalent binding and show the fluorescent signals. But I think more advanced, that is the, I think none of us have put focus. These luminescent, so luminescent based testing is the most sensitive, and they need very low concentration of the virus particles. I mean, up to, I mean, femtomolar or something. But still, I'm not aware of, and couple of groups are working here. So fluorescent based testing is still in our in our plate, and we are working here. But still, I mean, we haven't came up with the system that can improve the corresponding sensitivity. And still, after initial screening, I think PCR is the only way to go for the final diagnosis. It will take a while, I think, to unfold the mystery and get more sensitive test. I think it will take a while, at least 2 to 3 months. Yeah.
Yeah. So it's a good discussion. So I can tell you the primary idea that we have. We are trying to label a nanomaterial of this with the fluorescent material, and further nanonize it, and then label it with the your primer piece, so that when there is a number of RNA copies comes in, and you go through the genetic machinery, then there is a fluorescent amplification. So since it's going to be kind of nanonized, the possibility of getting amplification much higher, even though there is a less number of copies of RNA.
Yes, true.
So that is the idea. So since you are expert, seems like you are doing very in-depth studies in nanoscience area, and that too in Max Planck. So you should be able to pull it out. Certainly, I will get in touch with you offline.
Sure, definitely send me an email. I will have a long discussion with you. Yeah, it is very interesting you point out this. Yeah. Any other question?
From my side, I'm done. Thank you so much.
Okay, thank you.
I have just one question.
Yes.
Also, Dr. Singh, so you are doing very advanced work, but I got just relevant thing when you have a 3D bioprinting versus the organoid which you prepare with the conventional method. So have you ever checked the molecular signatures and their basically comparative analysis?
So yeah, I mean, it is another good question, and the field is new basically. So
I mean, organoid itself is very new. We are trying to make it for last 1 year.
Yeah. So we have introduced actually a kind of so nature inspired thing, like in the spinach, this is palak. Palak kind of thing. So if you will decellularize this Palak, so all the chlorophyll will go and you will see nice skeleton, and this is quite biocompatible. So what we are doing, we are making one layer of the cells, putting this network of the Palak on the, I mean the without chlorophyll top, and so we are using different strategies. This is one example I told you that. So here,
this is kind of scaffolding that you are
scaffolding, yeah. So we are making layer by layer scaffold the blood vessels, cells, because if you will see inside our body, if you will hit with the knife, you will see that, okay, there is a muscle and the blood vessel. So it's a scaffolding strategy we are using, but still we are in initial stages. It will take a while, yeah. But this is very interesting field because we can in real time produce the whole organs. So maybe after 10 years, it will be like an organ shop. I mean, the donors and other things, I think it will be less required compared to current scenarios, you know.
Hello.
Hello.
Can you hear me? So no, actually anatomical wise and morphological wise, you are right. I was saying that if you compare their basically protein profiling, looking at RNA profiling, and I'm sure that you will find very different signature with what we see even at the single cell level.
Yeah, true.
Okay. So are you having any plan to check it at that level and make sure that if not all, at least 80% physiologically it is relevant?
Yeah. Actually, that is a very important part of such development or such initiative, which is up to next advanced technology, especially in 3D printing. So we have for each organ, we have a set of molecular markers that is that we compare at single cell versus in the 3-dimensional scaffold cultured cells. Yes. And very often we have the target that as a target to reach. When we get the equivalent profile like they exist in vivo, or those are available into the clinics, or the in the hospitals, or the surgeons, those use these marker as a important point to say that, okay, there is a normal tissue growth or something. So we take them as an organ, but you are right. I mean, we don't get often the similar results like the single cell level versus these 3D scaffolded. And in each scenario, I would say the answer is different. And some particular organ or 3D scaffolded structures or 3D printed structures or organs or tissues are easy compared to the other organ. For example, you take kidney. So particularly the kidney and liver is easy to make. And so we are working a lot on the lungs. It's very difficult. Heart, I mean, some of collaboration in the Case Western University in the United States, it's more difficult. So it depends on different organs, such kind of analysis, comparison, and getting the right fit, you know, to develop such 3D structures. So it will take, I think, another 10 years because, yeah.
I'm sure that your group must be having the molecular biologist as well, who will take
Yes.
care about this issue for sure.
Yeah.
And in Germany, where you are situated?
In Berlin.
Okay. Oh, Berlin is my favorite place. I go to Humboldt University quite often.
Good.
So
maybe next time you can visit us. Yeah.
Yeah, I did PhD from Regis.
Okay. I think his video is hanged. Rakesh sir.
So let me add on. I think Sunil, myself, and there is another guy, Sobi. We all were in Regensburg during PhD and postdoc.
Mm-hmm.
So I'm familiar of Germany. That's the reason I like your work. We visited a couple of times to Max Planck.
Okay. Yeah. It's a even
Amazing
for the
place.
research, you know, so much funding they have. Yeah.
So much unrestricted, literally unrestricted funding.
Yeah.
So Sunil is also leading in some sort of molecular biology in JNU.
Okay.
He's doing pretty great.
Okay. Great,
So
sir.
Sunil,
I would request send an email, you know, and then we can discuss. Actually, I want to make a network of, you know, all the colleagues in India, so we can come up with something very very good, you know, solution and help to the, I mean, our local people also. Yeah, yeah.
Sure.
I think that is what about this conference is. This is to unite all the countries and eminent person who are working in very high grade technology that can come to the mass and at the bottom level help society benefit.
Yeah, thank you, Rakesh, for organizing this. I'm really happy to be part of this. Yeah.
We also.
So Dr. Ajay, are KI product?
Yeah.
Oh, that's
Dr. Ajay is an innovator of this institute. He has been here to deliver another talk, and we have listened him earlier also. So thank you, Ajay. Actually, in any conference, discussion is the most important part, and fetch the maximum results. I think even better than the talk itself. So thank you, Ajay. Be with us.
Sure, sir.
If you have time, I know you have some constraints, time constraint. Now I would like to invite Dr. D.K. Tripathi ji to invite next speaker. After that, I will invite Dr. Sunil Kataria, but before him, Dr. D.K. Tripathi will call upon Dr. A.R. Siddiqui to take on the dais.
Thank you, Dr. Pandey. I invite our next guest speaker, a great academician, Professor A.R. Siddiqui, Head Department of Geography, University of Allahabad. Professor Siddiqui is looking after Indian Institute of Geomorphologists as its Secretary General, and your works in the field of geohazards and geoinformation science always enlighten researchers working in the field of geohazards, environment, and sustainability sciences. Sir, I welcome you. Please deliver your talk. Professor A.R. Siddiqui.
गुड आफ्टरनून एवरीवन. डॉक्टर त्रिपाठी, आपको आवाज आ रही मेरी?
Yes, hearing. Yeah.
थैंक यू. सबसे पहले मैं कमला नेहरू इंस्टीट्यूट ऑफ फिजिकल एंड सोशल साइंसेज की पूरी टीम को धन्यवाद देना चाहूंगा कि आपने इतने रिलेवेंट टॉपिक पे जो वेबिनार ऑर्गेनाइज कर रहे हैं और अभी डॉक्टर साहब को सुनने को मौका मिला और इतना बढ़िया पेपर उन्होंने प्रेजेंट किया यहाँ पर और बहुत सारी इंफॉर्मेशन मिली. तो मैं इस टॉपिक के लिए आपको धन्यवाद देना चाहता हूँ और मैं जितने भी पार्टिसिपेंट हैं उनसे मैं कहना चाहता हूँ कि डॉक्टर दिनेश कुमार त्रिपाठी जी, D.K. त्रिपाठी जी, इनको मैं कहता हूँ डायनामिक एंड नॉलेजेबल त्रिपाठी, D फॉर डायनामिक एंड K फॉर नॉलेजेबल. तो त्रिपाठी जी, मैं आपको भी बहुत ही बधाई देना चाहता हूँ क्योंकि आपने जो टॉपिक चुना है निश्चित रूप से मतलब सारी चीजों का कहीं ना कहीं से इसमें सार मिलेगा लोगों को. रिसर्च एंड इनोवेशंस इन फिजिकल, बायोलॉजिकल एंड सोशल साइंसेज फॉर सस्टेनेबल डेवलपमेंट. पिछले 20-25 सालों से अगर आप देखें तो सस्टेनेबल डेवलपमेंट में जितनी भी यूनिवर्सिटीज हैं, जितनी भी सेंट्रल यूनिवर्सिटीज हैं, इंस्टीट्यूट हैं, कहीं ना कहीं से क्लाइमेट चेंज पर या सस्टेनेबल डेवलपमेंट पे या गुड गवर्नेंस पे या कुछ इस तरह का थीम होता है कि वो अप्लाई करते हैं और बड़ी बड़ी कॉन्फ्रेंस करवा रहे हैं. लेकिन जो सस्टेनेबल डेवलपमेंट जैसे कि हम लोग ज्योग्राफी और सोशल साइंसेज में हैं और जब 1987 में जाते हैं और आवर कॉमन फ्यूचर जो एक आर्टिकल पब्लिश होता है उसमें अगर 3 जो कंपोनेंट अगर आप देखें कि ये कंसेप्ट आया और उसमें जो 3 मेजर जो कंपोनेंट्स हैं उसमें किन चीजों की बात होनी चाहिए. इनोवेशन में हम अभी आएंगे लेकिन चूंकि सस्टेनेबल डेवलपमेंट एक बड़ा एस्पेक्ट है और डेवलपिंग कंट्रीज के लिए खास तौर से, इसलिए इसके जो 3 इंपॉर्टेंट कंपोनेंट है उस पर जाएंगे. पहली चीज फुल केयर ऑफ एनवायरनमेंट की बात. इसमें पूरी दुनिया के जो वैज्ञानिक हैं, शोधकर्ता हैं, जो हमारे रिसर्च स्टूडेंट्स हैं, ये सारे लोग जो है फुल केयर ऑफ एनवायरनमेंट की बात करते हैं क्योंकि एनवायरनमेंट मैंने इसलिए शब्द का नाम लिया है कि हम किसी भी चीज के साथ इस वर्ड को जोड़ देते हैं. जैसे मान लीजिए पॉलिटिकल इन्वायरमेंट, एग्रीकल्चरल इन्वायरमेंट, कल्चरल इन्वायरमेंट, सोशल इन्वायरमेंट, अर्बन इन्वायरमेंट, रूरल इन्वायरमेंट. तो हमको इसका मतलब समझना चाहिए कि आखिर पर्यावरण जो इन्वायरमेंट का वर्ड है, तो पहली बात जो उन्होंने कहा कि फुल केयर ऑफ इन्वायरमेंट, जो कि आवर कॉमन फ्यूचर में बात कही गई. दूसरी बात कही गई थी उसमें जुडिशियस यूज ऑफ रिसोर्सेज. जो भी हमारे संसाधन है उनका जो इस्तेमाल हो जुडिशियस हो। और तीसरी बात जो कही गई थी उसमें इक्विटेबल अप्रोच की बात कही गई थी, इक्वल की बात नहीं कही गई थी, बल्कि इक्विटेबल की, जिसको जितनी आवश्यकता हो, जिसको जितनी जरूरत हो, उसके पास उतने संसाधन होने चाहिए। अब बात आती है कलाम साहब की, कि कलाम साहब की विजन 2020 में ये सपना देखा गया था और उसमें अपने विजन 2020 में उन्होंने कैलेंडर किताब में दिया था और दुनिया भर के आविष्कारों की बात की थी और 80% 90% आविष्कार हम लोगों की पीढ़ी ने देखा है और हम लोगों से पहले की पीढ़ी ने देखा है। यानी इनोवेशंस का जो कैलेंडर है और आविष्कारों का जो इतिहास है बहुत पुराना नहीं है, यानी बहुत नया है। तो उन्होंने भी एक बहुत इंपॉर्टेंट बात कही थी, किसी भी राष्ट्र की शक्ति जो है वो प्रौद्योगिकी में छिपी होती है, टेक्नोलॉजी में छिपी होती है। अगर हमारी टेक्नोलॉजिकल एबिलिटी है और चीजों को अडॉप्ट करने के लिए हम लोग सक्षम हैं, तो हम एक विकसित राष्ट्र हैं। तो टेक्नोलॉजी को उन्होंने बड़ी अहमियत दी। और उसके अलावा दूसरी बात उन्होंने जो कही थी वो वेलफेयर ऑफ द सोसाइटी की बात की थी और ये कहा था कि कोई भी डेवलपमेंट तब तक डेवलपमेंट नहीं है जब तक कि वो समाज कल्याण की बात न करता हो। अगर हम इस टॉपिक को ले, रिसर्च एंड इनोवेशन इन फिजिकल, बायोलॉजिकल एंड सोशल साइंसेज फॉर सस्टेनेबल डेवलपमेंट, तो लास्ट में जो हमारा जिस्ट है, जो हमारा फोकस एरिया है, जो थ्रस्ट है, वो यही है कि सोसाइटी का वेलफेयर या डेवलपमेंट। तो जो उन्होंने बात कही थी कि वेलफेयर ऑफ द सोसाइटी, और तीसरी बड़ी इंपॉर्टेंट बात कही थी टाइम की। अगर समय के अनुसार, और जो चाहे वो टेक्नोलॉजिकल डेवलपमेंट हो या किसी भी चीजों का एडाप्टेशन हो, समय के साथ अगर हम अपडेट नहीं करते हैं तो वो डेवलपमेंट उसमें नहीं आएगा। आज आप देखिए, समाजशास्त्री भी हमारे साथ इसमें बहुत होंगे क्योंकि सोशल साइंस में भी इनोवेशन की बात आपने की है। तो सोशल साइंस में तो एक विचार ही क्रांति ले आता है। जैसे मान लीजिए कहा गया कि वर्कर्स ऑफ द वर्ल्ड यूनाइट, डू और डाई, दुनिया भर के मजदूरों एक हो, डू और डाई। तो ये एक विचार ही क्रांति ले आते हैं। तो उसमें किसी लेबोरेटरी की जरूरत नहीं होती है। लेकिन अगर आपने ये कहा कि पीढ़ी, तो आज भी अगर हम देखें तो 3-4 पीढ़ियां एक साथ चल रही हैं। आज घरों में बच्चे, अगर आप उनसे कहें दाल चावल रोटी सब्जी, तो मेरा ख्याल है वो चाइनीज और न जाने क्या क्या फास्ट फूड खाते हैं। और उसके बाद जो रोटी दाल सब्जी खाने वाले लोग हैं उनको पुरानी जनरेशन का मानते हैं। तो फूड हैबिट हो, चाहे रहन सहन हो, चाहे पहनावा हो, इन सारी चीजों की बात हमने की है। लेकिन पूरी दुनिया में जो ट्रांसफॉर्मेशन आया है, कहीं ना कहीं जो आया है वो नॉलेज के अपग्रेडेशन से आया है। नॉलेज के ट्रांसफॉर्मेशन और डिफ्यूजन की वजह से उसका ट्रांसफॉर्मेशन हुआ है। लैंग्वेज का डिफ्यूजन हुआ है। अगर हम एक सेंटेंस बोलते हैं, मान लीजिए कुर्सी पर बैठ जाइए, अब कुर्सी पता चला अरबिक वर्ड है, लैम जो है इंग्लिश वर्ड है, तो कमीज कोई और शब्द है। एक ही सेंटेंस में पूरी दुनिया के कई कई शब्द आ जाते हैं। तो मैंने कहा कि चाहे वो नॉलेज हो, लैंग्वेज हो, टेक्नोलॉजी हो, कल्चर हो, रिलीजन हो, कहीं ना कहीं से इसके डिफ्यूजन की बात आती है। जब सस्टेनेबल डेवलपमेंट का जो एस्पेक्ट है, तो दो नाम बड़े इस शताब्दी में आते हैं। अगर वर्ल्ड के कॉन्टेक्स्ट में देखें तो वर्ल्ड के कॉन्टेक्स्ट में नॉर्मन बोरलॉग का आता है, जो क्या कहते हैं, उन्होंने ग्रीन रिवॉल्यूशन के क्षेत्र में पूरी दुनिया में उन्होंने जो काम किया है। अगर आप देखें तो 1970-80 के दशक में जो गांवों में जो स्थिति थी, लोगों को गेहूं नहीं मुहैया हो पाता था, जौ बाजरा इस तरह के अनाज वगैरह हुआ करते थे। बहुत आसान नहीं है कहना कि वो बहुत सारी चीजें अवेलेबल थी, क्योंकि भारतवर्ष में भी आविष्कारों का इतिहास जो है वो बहुत नया है। तो अगर आप MS स्वामीनाथन जी और नॉर्मन बोरलॉग जी का ही योगदान अगर देख ले, तो वही ट्रांसफॉर्म कर देने वाला एक बहुत बड़ा इनोवेशन है। लेकिन इसके तह में जाने के लिए ये जरूरी है कि हम 8000 साल या उससे पहले की बात करें, जब इंसान जो है गिनती करना भी नहीं जानता था, बाद में जो नियोलिथिक एग्रीकल्चरल जो एज आई, जब से हम सेडेंटरी एग्रीकल्चर में हम आए, कृषि करना जाने, तो उससे और उसके बाद डोमेस्टिकेशन ऑफ एनिमल हुआ, पशुपालन करना सीखे। तो आपको याद होगा कि इंसान उससे पहले गिनती करना भी नहीं जानता था और जानवरों को लेके जाता था जब जंगलों में, तो लकड़ी की छोटी छोटी टुकड़े करके जाता था और वो गिनता था, और शाम को जब लौट के आते थे तो उन्हीं लकड़ियों को गिनता था, और अगर एक दो रह जाती थी तो उसको ये पता चलता था कि हमारे 2 जानवर कहीं ना कहीं जंगलों में छूट गए हैं। तो अब 8,000 साल पहले या 5,000 साल पहले आप एग्रीकल्चर करना जाने, और पूरी दुनिया में जहां जिस प्रकार का आपको ज्योग्राफिकल कंडीशंस मिली, उसी तरह से ह्यूमन ने अपना रिस्पॉन्स दिया। और आप जानते हैं कि जो मनुष्य की इकोनॉमिक एक्टिविटीज हैं वो डाइवर्सिफाइड हैं, तो आर्थिक क्रियाएं विविध हैं, क्योंकि ज्योग्राफिकल जो बेस है वो भी विविध है, उससे हमारी आर्थिक क्रियाएं विविध हुई। अगर हमको एग्रीकल्चरल बेस मिला तो हम कृषि करना जाने, अगर हमको जो है मिनरल रिसोर्स मिला तो हमने संसाधनों को एक्सप्लोर किया और इंडस्ट्रियलाइजेशन हुआ। लेकिन 1924 तक ये सारी चीजें हम नहीं जानते थे। उसके बाद एक बॉटनी के प्रोफेसर थे लेनिनग्राड यूनिवर्सिटी में, आपने नाम सुना होगा, N.I. वाविलोव। उन्होंने दुनिया के प्राचीनतम कृषि के उद्भव के केंद्र के बारे में बताया, और वाविलोव ने जो जानकारी दी कि कृषि के उद्भव जो क्षेत्र थे, ओरिजिन जो प्लेसेस थे, उसमें साउथ वेस्ट एशिया और इंडिया, उसके अलावा जो है ईस्टर्न एंड सेंट्रल पार्ट ऑफ चाइना, साउथ ईस्टर्न पार्ट ऑफ चाइना, सेंट्रल पार्ट ऑफ चाइना, मेडिटेरेनियन रीजन, उसके अलावा हिली अफ्रीका जो रीजन है, ईस्टर्न जो पार्ट है उसका, उसके अलावा साउथ मेक्सिको, पेरू और बोलिविया। तो इतने स्थान बताया, और उन्होंने बताया कि यहाँ पर इस प्रकार की कृषि होती है। गेहूं कहीं पर होता है, मक्का कहीं पर होता है, जौ कहीं पर होता है, फलों का उत्पादन कहीं होता है, मेडिटेरेनियन रीजन में बताया उन्होंने, साइट्रस फ्रूट वगैरह के बारे में बताया। तो आप जाने, लेकिन अब आप ये देखिए कि जो लैंडमार्क हैं, अगर हम जाएं तो हम वहां नहीं जा रहे हैं अभी कि मिनोअन सिविलाइजेशन में, चार्डियन सिविलाइजेशन में, सुमेरियन सिविलाइजेशन में, इजिप्शियन सिविलाइजेशन में, बेबीलोनियन में नहीं जा रहे हैं, क्योंकि उससे पहले अगर आप इन सिविलाइजेशन में जाते हैं, चाहे मिनोअन में जाएं, चाहे चार्डियन में जाएं, सुमेरियन में जाएं, इजिप्शियन में जाएं, तो एस्ट्रोनॉमी आपको मिलेगी, नेविगेशन में आपको मिलेगा, जियोमेट्री मिलेगी, अर्थमेटिक मिलेगी। ये सारी चीजें हम ग्रीक और रोमन्स के टाइम में हम पढ़ चुके हैं। जैसे जैसे आगे आप बढ़े, चाहे वो ग्रीक हो, रोमन्स हो, इंडियन स्कॉलर्स हो, आपने देखा ये डेसिमल सिस्टम के बारे में, या भारत के जो लोग थे उन्होंने बहुत सारे इनोवेशंस किए जो आज तक भी उनको जो है सराहा जाता है, क्योंकि उनके बगैर कुछ काम ही नहीं चल सकता। चाहे आप भास्कराचार्य को ले लें, बारामीर को ले लें, धनवंतरी को ले लें, चरक को ले लें, जितने भी हमारे इनोवेशंस हैं आज तक उनको चुनौती नहीं दी जा सकी। अब बात आती है कि जो मोटी मोटी बातें हैं जो हम स्कूल में हम लोग पढ़ के आए हैं, जैसे आप कहते हैं इनोवेशंस जो फिजिकल साइंसेज में, क्योंकि बाद में इस सारी चीजों को हमको इंटरलिंक्ड करना है। अगर आप एग्जाम्पल के तौर पे अगर एक हवाई जहाज उड़ा रहे हैं तो इसमें कोई एक साइंस का वो नहीं है, इसमें फिजिकल साइंसेस भी है, यानी फिजिक्स भी है, इसमें केमिस्ट्री भी है। अगर आप एलपी उसका जो है पेट्रोल यूज करते हैं, इसमें केमिस्ट्री भी है, इसमें फिजिक्स भी है, इसमें हर चीज है। और अगर इसको एंशिएंट क्लासिकल टाइम से लेके जाते हैं और आपको जो है पवन हंस के बारे में अगर आपने पढ़ा है तो वो भी है, वो सारी चीजें भी हैं। तो मैं आपसे कहना चाहता हूं कि जो मोटी मोटी बातें हैं, अगर आप जाते हैं 1664 में और न्यूटन को याद करें तो आप ग्रेविटी, न्यूटन के लॉ को आपने पढ़ा है। उसके अलावा लॉ ऑफ प्लेनेटरी मोशन जो मेरा ख्याल है 1609 के आसपास जो केपलर का जो सिद्धांत था, उसके अलावा जो है टेलीस्कोप भी 1609 के आसपास ही उसका आविष्कार हुआ। ये सारी थ्योरीज आती रही, E = mc स्क्वायर या थ्योरी ऑफ रिलेटिविटी, तो ये सारी चीजें आते रही। लेकिन सबसे ज्यादा जो आविष्कारों का जो इंपॉर्टेंस हुई, क्योंकि अगर आप ह्यूमन हिस्ट्री में जाएंगे और ह्यूमन हिस्ट्री में आप जाएंगे तो आपको 1800 ईस्वी तक आपको ये लगेगा कि 1800 ईस्वी में वर्ल्ड की जो पॉपुलेशन थी वो 1 बिलियन पॉपुलेशन थी। अब जरा सोचिए कि 1800 में वर्ल्ड की जो पॉपुलेशन थी वो 1 बिलियन पॉपुलेशन थी, लेकिन 1798 में आपको याद होगा एक पेपर पब्लिश हुआ था, एसे ऑन पॉपुलेशन ज्योग्राफी, माल्थus की थ्योरी आई थी, जिसमें उन्होंने कहा था कि जो पॉपुलेशन है उसकी जो ग्रोथ है वो ज्योमेट्रिक है, लेकिन जो खाद्यान्न है, फूड ग्रेन है, उसका जो प्रोडक्शन है वो अर्थमेटिक है। यानी जो फूड ग्रेन है 1 2 3 4 5 6 इस तरह से हो रहा है, और पॉपुलेशन जो ग्रोथ है वो ज्योमेट्रिक है, 1, 4, 16, 64 इस तरह से बढ़ रही है। तो जब वर्ल्ड की पॉपुलेशन क्या कहते हैं, वर्ल्ड की पॉपुलेशन 1 बिलियन थी, उस जमाने में ये माल्थus का जो है थ्योरी आ गई थी। तो आप जरा सोचिए कि वर्ल्ड की पॉपुलेशन में किस तरह से चेंज आया। 127 साल बाद यानी 1927 में वर्ल्ड की पॉपुलेशन बढ़ के 1 मिलियन से 2 मिलियन हो गई। उसके बाद जो है 1960 में यही पॉपुलेशन 3 बिलियन हो गई। उसके बाद 1974 में यही पॉपुलेशन 4 बिलियन हो गई। 1987 में यही पॉपुलेशन 5 बिलियन हो गई और आज जो है 7.35 बिलियन पॉपुलेशन वर्ल्ड की है। तो अब आप ये देखिए कि जो चिंता हुई उस समय में, लेकिन अब जो फिजिकल साइंसेस की बात हुई उसमें जो लैंडमार्क है, जो आपने कहा कि इतनी जल्दी बाद में 15 साल में पॉपुलेशन की डबलिंग हो जा रही है, उसके पीछे रीजन क्या है? पहले भी हमारे पास जो है नेचुरल हैजर्ड हुआ करते थे, बहुत सारी चुनौतियां हुआ करती थी। हो सकता है सुनामी, अर्थक्वेक और ये साइक्लोन और डिजर्टिफिकेशन, डिजास्टर, नेचुरल डिजास्टर, ये सारी चीजें पहले भी मनुष्य अपने पर्यावरण से उसको कॉम्बैट करता आया है। लेकिन अगर आप देखें तो मेडिकल साइंसेस में अगर आप हिस्ट्री में जाते हैं तो वैक्सीनोलॉजी, जो अभी कोविड-19 के बाद अभी हम लोग इंतजार कर रहे हैं कि दुनिया का कौन सा देश जो है वैक्सीन लेके आता है और इस बड़ी डिजास्टर से हमको निजात मिलती है। तो आपको याद होगा एडवर्ड जेनर का नाम आया 1776 में और उन्होंने वैक्सीनोलॉजी वैक्सीन इजाद किया। लेकिन उसके अलावा जो अभी हम लोगों के बचपन में और उससे थोड़ा सा पहले पूरे पूरे गांव खत्म हो जाते थे, बच्चों की डेथ हो जाती थी। और याद होगा कि स्मॉल पॉक्स जैसी महामारी आई और उसका भी वैक्सीन आपको मैं बता दूं कि 1798 में आ चुका था। इसी तरह से कॉलरा वगैरह हुआ करता था, पूरे पूरे गांव खत्म हुआ जाया करते थे। और कॉलरा का भी 1897 ईस्वी में जो है उसकी वैक्सीन आई थी। अब सवाल इस बात का है कि पोलियो आपने याद होगा कि बचपन में दीवारों पे लिखा जाता था कि चेचा कि आप पोलियो का कोई भी केस बताने के लिए। लेकिन हमारे यहाँ देश में थोड़ा सा ये है कि अगर पोलियो का ड्रॉप पिलाना है तो उसके लिए अमिताभ बच्चन और ऐश्वर्या राय को पैसे देके या फ्री में उनसे रिक्वेस्ट करके कि आप जो है अगर एडवर्टीजमेंट करेगी तो जनता अपने बच्चों को जो है पोलियो का ड्रॉप पिलाएंगे। तो बहुत सारी चीजों के लिए हमको एडवर्टीजमेंट करना पड़ता है। अभी आपने देखा होगा कि कोरोना का टाइम चल रहा था तो हमारे पुलिस के जो सहयोगी, हमारे पुलिस के जो सिपाही और जो उनके अधिकारी जो लोग थे गाना गा रहे थे, किशोर कुमार का, मोहम्मद रफी का, और नए भी गाने सुना रहे थे कि देखिए आपको इस वक्त सोसाइटी को आपके कोऑपरेशन की जरूरत है। तो ये जो कम्युनिटी है, कम्युनिटी में जब हम लोग जाते हैं तो उनको अवेयर करना पड़ता है। तो पूरी दुनिया में जो अवेयरनेस का मामला है वो बराबर से नहीं पहुंचा। जो अभी हम डिफ्यूजन की बात कर रहे थे तो टेक्नोलॉजी का डिफ्यूजन हो या लैंग्वेज का डिफ्यूजन हो या नवाचारों का जो प्रचार प्रसार है, पूरी दुनिया में बराबर नहीं पहुंचा है। अलग अलग इसकी स्टेजेस रही हैं, इसलिए जो है डेवलपमेंट का जो वो है वो भी अलग अलग दिखाई देता है। बाद में फिजिकल साइंसेस में आपने देखा हो आज लोग सिगरेट पी रहे हैं, तंबाकू खा रहे हैं, उसके लिए हमको एडवर्टीजमेंट करना पड़ता है। लेकिन अब जब इसके इनोवेशन में जाएंगे, इन्वेंशन में, तो एक्सरे वगैरह के बारे में भी आविष्कार हुआ। और मैं आपको बता दूं कि 1870 तक अमेरिका की जो लाइफ एक्सपेक्टेंसी थी, अमेरिका की जहां की बात कर रहे हैं, वो 43 वर्ष थी। जरा सोच सकते हैं। अब आजादी से पहले जब आप पॉपुलेशन ज्योग्राफी में जाएंगे और आप एवरेज एज या लाइफ एक्सपेक्टेंसी देखेंगे तो बहुत कम थी। लेकिन मेडिकल साइंसेस में इन्वेंशन के बाद देखा होगा कि आपने जो है सारी चीजें हुई। शुरू में मैंने नाम लिया था कि हम सस्टेनेबल डेवलपमेंट की बात करते हैं, लेकिन जब हाई यील्डिंग वैरायटी ऑफ सीड और मैकेनाइजेशन एग्रीकल्चर में हुआ और कैनाल के इनोवेशंस हुए और जहां पर पानी नहीं पहुंच पाता था वहां पर आपने पहुंचाया, तो आपने देखा कि सोसाइटी जो है ट्रांसफॉर्म हुई और एग्रीकल्चर वहां पर मुहैया हुआ। तो हाई स्टैंडर्ड ऑफ लिविंग हो या इम्प्रूव्ड न्यूट्रिशन की बात हो या बेटर हाइजीन की बात हो या एनवायरमेंटल जो मॉडिफिकेशन की बात हो और जो लाइफ एक्सपेक्टेशन की बात हो और चाहे नॉलेज एक्सपेंशन की बात हो, कहीं ना कहीं इन्वेंशन का ही रोल है। भारतवर्ष में अगर आप जाएंगे तो मैं आपको बता देता हूँ, ज्योग्राफी के डॉक्टर डीके जी बैठे हुए हमारे साथ रिमोट सेंसिंग इंस्टीट्यूट में थे देहरादून में। मैं आपको बता देना चाहता हूँ कि अगर आप जानते हैं कि GLCF, जहां से हमको लैंडसेट डेटा फ्री मिलता है, अमेरिका की जो वेबसाइट है और जियोलॉजिकल सर्वे की जो वेबसाइट है, यूनाइटेड स्टेट जियोलॉजिकल सर्वे की जो वेबसाइट है, वहां से डेटा हमको फ्री मिलता है। लेकिन भारत में अगर आप देखते हैं तो ये रिमोट सेंसिंग का भी इतिहास बहुत ही नया है। लेकिन एरियल सर्वे जो है फ्रांस में शुरू हो चुका था 1840 के आसपास। लेकिन भारत में बहुत जो संस्थान है उनका अगर आप इतिहास देखेंगे तो बहुत ही नया है। जैसे मान लीजिए इसरो की बात कर दी है, इंडियन स्पेस ऑर्गेनाइजेशन की 1960 में, या नीरी जो नेशनल एनवायरमेंटल इंजीनियरिंग संस्थान है वहां पर नागपुर में, उसको आप देख लीजिए 1958 का। सेंट्रल एरी ड्रोन रिसर्च इंस्टीट्यूट जो है वो भी 1958 का है। इसी तरह से जो है FRI 1906 का है और नाटमो 1958 का है। अब आप जरा देखिए कि जो वर्ल्ड की सेकेंड लार्जेस्ट पॉपुलेशन यानी विश्व का 6वां व्यक्ति भारतीय है और वो 2.4% ज्योग्राफिकल एरिया में रह रहा है, उस देश में रिसोर्स मॉनिटरिंग और मैपिंग के लिए जितने भी बड़े संस्थान हैं वो इसी शताब्दी में हुए। तो इसका मतलब वर्ल्ड वार फर्स्ट और वर्ल्ड वार सेकेंड के आसपास पूरी दुनिया में लोगों को यही नहीं पता था। अगर आप कोई भी किताब ज्योग्राफी की देखेंगे तो उसमें 3 चीजें नहीं होती थी, टेबुलेशन नहीं होता था, इलस्ट्रेशन नहीं होता था, समराइजेशन नहीं होता था, सारणी नाम की कोई चीज नहीं होती थी कि दुनिया में कितना लोहा, दुनिया में कितना तांबा, दुनिया में कितना गेहूं क्या प्रतिशत है। अगर ये क्वांटिटेटिव रिवोल्यूशन नहीं आता तो हो सकता है बहुत सारी चीजें हम लोग नहीं जानते। बात की जो है पुस्तकों में आपने देखा होगा कि हमने सारणी देना शुरू कर दिया। अब मैपिंग और मॉनिटरिंग इसलिए भी जरूरी है क्योंकि ऐसे तो आपको सारी चीजें हरा भरा लगता है, लेकिन कैन यू इमेजिन कि 2.38% फॉरेस्ट एरिया है इलाहाबाद का, वाराणसी का 2% से भी कम है। अब 6.09% जो है यूपी में फॉरेस्ट कवर है। तो मैपिंग और मॉनिटरिंग क्यों नहीं होनी चाहिए संसाधनों की? आप डिजर्ट की बात कर रहे हैं, आपके दिमाग में डिजर्ट का नाम लेते ही जो है वहां रेगिस्तान आ जाता है और धीरे धीरे यहाँ पर वनों का आच्छादन कम होता जा रहा है। उसकी चिंता हमको क्यों नहीं होनी चाहिए? तो रिमोट सेंसिंग में बहुत सारी 4 चीजें इंपॉर्टेंट है और सारी चीजें पढ़ी हैं जिनका बड़ा रोल है आज के टाइम में। और मैं आपको बताने देना चाहता हूँ कि जो डेटा है कौन इस्तेमाल कर रहा है ये बड़ा इंपॉर्टेंट है। समाजशास्त्री कर रहा है कि भूगोलवेत्ता कर रहा है कि भूगर्भ शास्त्री कर रहा है, कौन कर रहा है इंपॉर्टेंट। तो इसीलिए मैं कहता हूं कि पहली बात तो हमारे पास सबसे ज्यादा डेटा होनी चाहिए। उससे रिलेटेड आपने देखा कि हमने खुद भी दुनिया के टॉप 5 कंट्रीज में हम आते हैं और भुवन की वेबसाइट में किसी जमाने में आपको अपने पिताजी और आप उनके पिताजी के खेत के बारे में नहीं मालूम हो पाता था। आज जो है भूलेख वेबसाइट है, आज जो है बहुत सारा डेटा आपको मिल जाता है इंडियन स्पेस रिसर्च ऑर्गेनाइजेशन से। तो डेटा, टेक्नोलॉजी, एप्लीकेशन और पीपल, ये 4 कंपोनेंट GIS के हैं। तो मैं ये कहना चाहता हूं, अभी 4 नाम अगर आप ले लीजिए तो पिछले 15-20 सालों में तो उसने पूरी दुनिया को ट्रांसफॉर्म करके रख दिया है। हमारे दिनेश कुमार त्रिपाठी जी दिन में 2 घंटे फेसबुक और गूगल में 1-1.5 घंटे देते होंगे। अब जरा सोचिए, मैं तो बड़ा चौंक रहा था कि जब मार्क जुकरबर्ग की मैंने एज देखी कि 1988 में पैदा हुए और 2004 में पूरी दुनिया को उन्होंने फेसबुक दे दिया। जरा सोचिए आप, ये 2020 चल रहा है और 88 में उनकी पैदाइश हुई थी। 12-4-16 साल की उम्र में पूरी दुनिया को उन्होंने फेसबुक दे दिया। उसके अलावा जो है अगर आप बात करें पॉल एलन की और क्या कहते हैं बिल गेट्स की बात कर लीजिए। माइक्रोसॉफ्ट आपको मिल गया 1975 में और जो है उसके अलावा बहुत सारे सॉफ्टवेयर मिले आपको। एल्विस मिला 1996 में, ArcGIS मिला 1999 में, एडास इमेजिन आपको मिल गया 1990 में। तो आप बताइए, जो आपके पास सॉफ्टवेयर है, चाहे एल्विस हो, चाहे वो माइक्रोसॉफ्ट हो, चाहे ArcGIS हो, कभी आपने देखा कि ये इनका इतिहास कितना नया है? तो आप ये बताइए, जब हमारे संस्थान ही नहीं थे, या ज्योग्राफी में भी आप देख लीजिए कि दुनिया भर के, अगर आप वान थुनेन की 1826 की एग्रीकल्चरल लोकेशन की थ्योरी छोड़ दें, जो कि एक अर्थशास्त्री में 1826 में दिया गया था, वो भी डी रिकार्डो के प्रिंसिपल पे था। तो आज अगर आप ज्योग्राफी में देखें तो 1909 में थ्योरी ऑफ एग्रीकल्चरल लोकेशन, उसके बाद 1933 में जो है थ्योरी ऑफ ये क्रिस्टालर की वाल्टर क्रिस्टालर की सेंट्रल प्लेस थ्योरी, उसके अलावा अगर आप तो बहुत सारी चीजें आपके सामने हुई। ओपेन का क्लाइमेटिक क्लासिफिकेशन नहीं था, हर्बर्टसन का मेजर नेचुरल रीजन नहीं था, तो बहुत सारी चीजें आप देखें, थंडर्स नहीं थे, तो बहुत सारी चीजें अगर आप देखें तो आपकी और हमारी जो ये शताब्दी है वो कहीं ना कहीं से उसको देख रही थी। तो जैसे मैंने बिल गेट्स की बात की, वैसे ही मैं लैरी पेज की बात करूंगा और सार्गे ब्रिन की बात करूंगा, जिन्होंने आपको गूगल दे दिया। हमारे टाइम की बात याद है कि जब मैं हॉस्टल में हुआ करता था तो ईमेल बनाना नहीं आता था, या डीके त्रिपाठी जी जानते होंगे, प्रोफेसर आलोक दुबे जी हमारे ज्योग्राफी डिपार्टमेंट में थे, पहला उन्होंने मोटरोला का मोबाइल लिया तो हम लोग बड़ी गौर से देखते थे। और आपको तो बिल्कुल याद होगा कि जब डे टाइम में फोन करते थे केबिन में घुस के, और 7 बजे से आधा हो जाता था और 11 बजे से 1/4 हो जाता था, तो हम लोग अपने हॉस्टल में जाके जब जाते थे और घर में बात करनी होती थी, घर में फोन था नहीं, पड़ोसियों के यहाँ से, तो ये 7 बजे वाला फोन करते थे कि 11 बजे कौन जाके बुलाएगा। तो आप ये देखिए कि बहुत सारे आविष्कार जो मतलब आज से 15-20 साल पहले अगर कहा जाता कि एक मशीन आएगी जिसमें आप कार्ड लगाएंगे और पैसा गिरेगा, तो ये जनरेशन कभी नहीं मानती। लेकिन आप देखिए कि किस प्रकार से जो हमारे नवाचार है उसने पूरी दुनिया को चेंज किया। अभी एप्पल का मोबाइल और एप्पल का लैपटॉप जो है 2007 में जो है स्टीव जॉब्स, ये सारी चीजें आप सामने देख रहे हैं। तो ये जो इनोवेशन है उसका सबसे बड़ा जो इम्पैक्ट पड़ा हमारे नजरिए पे, हमारे परसेप्शन पे, हमारा चीजों को देखने का नजरिया बदल गया, परसेप्शन बदल गया। और नजरिया आप जानते हैं कि परसेप्शन में ही सारा मामला है। और लिटरेचर में तो इतना बड़ा इनोवेशन है कि अगर आपको एक लाइन अगर किसी शायर ने और किसी लेखक ने कह दिया, हो सकता है आपकी पूरी जिंदगी बदल जाए। तो आप सोशल साइंसेस को के रिसर्चेस को भी आप हल्के में नहीं ले सकते। आप ये नहीं कह सकते कि हिंदी का जो साहित्यकार है और उर्दू का जो नगमा निगार है उसको आप हल्के में नहीं ले सकते। और समाज को बदलने का काम सबसे ज्यादा फिल्म इंडस्ट्री ने किया है। समाज को सबसे बदलने का काम जो है फिल्म इंडस्ट्री ने काम किया। अभी कोविड-19 में लेबर की आप बात कर रहे हैं, रूरल सोसाइटी की बात कर रहे हैं, अर्बन सोसाइटी की बात कर रहे हैं, तो आजादी के बाद से ही अगर हम आपसे शुरू कर दें तो शुरुआत ही हो जाती है। और वहां पर आप देखेंगे कि चाहे वो नया दौर फिल्म हो, चाहे वो मजदूर फिल्म हो, चाहे वो क्या कहते हैं नरगिस की जो फिल्म थी जो पूरे परिवेश को जो है देती थी। तो आप ये समझ लीजिए कि उपकार को ले लीजिए, मनोज कुमार की जो फिल्म थी, मेरे देश की धरती सोना उगले उगले हीरे मोती है। तो इस तरह की जो फिल्में थी पूरे भारतीय परिषद उसको उन्होंने दिखाया था। इसलिए कि फिल्में भी समाज का जो है आईना है। जिस प्रकार का समाज होता है उस प्रकार से डायरेक्टर और एक्टर और गीतकार ये सारी चीजें लिखते हैं। तो चाहे और आप उसको भी बराबर ही ले सकते हैं, जैसे मतलब E=mc², ठीक है, उन्होंने किया, आपने देखा कि पता चला हिरोशिमा और वहां पर जो है वर्ल्ड वार हुई और उन्होंने बम गिरा दिया। लेकिन ये जो सोशल साइंसेस की रिसर्च है, जहां पर आपके जज्बात बदल रहे हो, जहां आपका नजरिया बदल रहा हो, वो सबसे बड़ी रिसर्च मानी जाती है। और आज समाज को इसी प्रकार के शोध की आवश्यकता है। आजकल जो है फेसबुक पे और उस पे प्रेमचंद जी पे चल रहा था, कभी आप जाइए इन साहित्यकारों पे, कभी आप जाइए जो उन्होंने गजल लिखी है, चाहे वो गालिब रहे हो, चाहे वो ज़ौक़ रहे हो, चाहे जोश मलिहाबादी रहे हो, हर दौर अपने जमाने का पीक होता है। आप ये नहीं कह सकते कि कौन सा जमाना बैकवर्ड था, कौन सा जमाना फॉरवर्ड है। आज सारी चीजें आज की आवश्यकता के अनुसार हमारे पास है। लेकिन अगर आप उस जमाने में जाएंगे तो जिन लोगों का भी आप नाम लेंगे वो अपने जमाने के पीक हुआ करते हैं। तो आज जरूरत सिर्फ इस बात की है कि चाहे वो फिजिकल साइंसेस हो, चाहे वो सोशल साइंसेस हो, चाहे वो बायोलॉजिकल साइंसेस हो, चाहे ह्यूमैनिटीज और ये आर्ट्स की फील्ड में जो होने वाली रिसर्च हो, इन सबका ताना बाना है और इन सबका जो है सामंजस्य होना चाहिए। कोई भी डिसिप्लिन अलग रह के सर्वाइव नहीं कर सकती। आप उसके कोरोना वायरस के उस पे जाएंगे या बायोलॉजिकल साइंसेस पे जाएंगे, कोई भी शाखा जो है वो स्वतंत्र होके सर्वाइव नहीं कर सकते हैं। और आप जानते हैं कि इन इन रिसर्चेस को, इन शोध को जो है, इन इनोवेशन को एक दूसरे की इंटरडिपेंडेंसी है और बनी रहेगी। तो मैं आपसे कहना चाहता हूं कि जो जितना पोटेंशियल भारतवर्ष में, मैंने आपसे कहा था कि किसी वेबिनार में कि भारत का जो ज्योग्राफिकल एरिया है वो केवल 2.4% एरिया है, लेकिन भारत के संदर्भ में जब हम बात करते हैं तो ये भारत नहीं है। भारत जो है उत्तर प्रदेश में बिहार को अगर मिला दीजिए, उसकी टोटल पॉपुलेशन कर दीजिए, तो ये अमेरिका की पॉपुलेशन के बराबर आप हो जाते हैं। केवल उत्तर प्रदेश की पॉपुलेशन जो है ब्राजील के बराबर में है, मध्य प्रदेश की पॉपुलेशन टर्की के बराबर में है, यूरोपियन यूनियन की पॉपुलेशन राजस्थान के बराबर में है, गुजरात के बराबर में है। तो आप इतने बड़े देश को लेके चल रहे हैं और इतनी ज्यादा प्रोबेबल पोटेंशियल है यहाँ पर कि अगर इन चीज को एक्सप्लोर करेंगे, और मुझे ये लगता है कि जो हम संभावनाओं को तलाश कर रहे हैं और हमारी संभावनाएं स्कूलों में है, प्राथमिक विद्यालय में है। जैसा कि कलाम साहब ने कहा था कि अगर हम अपना एक बहुत बड़ा इन्वेस्टमेंट स्कूलों में करें तो सोशल कैपिटल के रूप में, मनुष्य एक सामाजिक पूंजी के रूप में भारतवर्ष के जब ये 10, 12, 15 साल में जब बच्चे बढ़ेंगे तो ये जो डेमोग्राफिक डिविडेंड है इससे पूरा परिदृश्य बदल जाएगा। और ये भारत जो है जिस प्रकार से सुपर पावर है और सुपर पावर थे, आने वाले दिनों में भी आज आप यही से अंदाजा लगा लीजिए, अमेरिका का इंटरेस्ट हम में है, चाइना का इंटरेस्ट हम में है, जर्मनी का इंटरेस्ट हम में है। दुनिया के सारे देश लोग जो है भारत में क्यों आ रहे हैं? क्योंकि सेकेंड लार्जेस्ट पॉपुलेशन एक डिमांड लेके उठ रही है और अपनी इकोनॉमी को मजबूत करके उठ रही है। वो जो भी तैयार करेंगे तो वो यहाँ पर खरीद लेने की उनकी क्षमता, परचेजिंग पावर बढ़ रही है, तो इसलिए उन देशों का इंटरेस्ट हमें है। लेकिन कोविड-19 के बाद कम से कम इस वेबिनार के दौर में हम लोगों को और सब लोगों को मौका मिल रहा है अपनी अपनी बात खुल के कहने की कि कोई सामने यहाँ पर नहीं है। अगर हमने थोड़ी बहुत जबान फिसल गई तो कम से कम आज रिस्क नहीं है कि कोई जो है 2-4 सवाल पूछ लेगा, लेकिन हम सबको मौका मिल रहा है अपनी बात को कहने का। और हमें लगता है कि हम लोग रिविजिट कर सकते हैं कि कहाँ पर गलतियां रह गई हैं और आने वाले दिनों में इस देश को जो है किस प्रकार से मजबूत किया जा सकता है। और जैसे कि मनुष्य को जो है हमेशा में लिया गया है कि ये अर्थ चेंजर है, मैन इज द मैनेजर एंड मैन इज आल्सो मिसयूजर। तो ये मैनेज करने का भी काम करता है, मिसयूज भी कर रहा है और मैनिपुलेट भी कर रहा है। तो कुल मिला के जो है हमको अपनी जो इतनी बड़ी पॉपुलेशन है उसमें जो डाइवर्सिटी है और उसको देखने की जरूरत है कि क्या वजह है कि बिहार और उत्तर प्रदेश और उड़ीसा और वेस्ट बंगाल जैसे राज्य जहां पर दुनिया भर की संपन्नता भरी हुई है, जहां पर दुनिया भर की ज्योग्राफिकल कंडीशंस हैं कृषि करने के लिए, उद्योग लगाने के लिए, नगरीकरण करने के लिए, और ऐसी क्या वजह रह गई कि जब इस कोविड-19 में मजदूर को आना पड़ा तो इन प्रदेशों में उसकी मोबिलिटी रही, ऐसी क्या वजह रही? क्योंकि होना तो ये चाहिए था कि ये प्रॉस्परस रीजन है, यहाँ तो ऑलरेडी डेवलपमेंट दिखाई देना चाहिए था, लेकिन आपने देखा कि सबसे ज्यादा बदतर हालात जो है हमारे इस रीजन की रही। तो आने वाले दिनों में जो न्यूक्लियस हैं पॉपुलेशन के, छोटे छोटे लघु स्तर के जो छोटे छोटे गांव हैं और मध्यम स्तर के जो टाउन हैं, वहां पर जो है गवर्नमेंट की जो पॉलिसीज है उसको डिसेंट्रलाइज करके वहां पर इन्वेस्टमेंट की जरूरत है और उनको मजबूत बनाने की आवश्यकता है। तो डीके त्रिपाठी जी सामने दिखाई दे रहे हैं, उनके प्रेशर में बहुत ज्यादा नहीं बोल रहा हूँ क्योंकि वेबिनार में एक लिमिटेड टाइम होता है और मुझे बहुत देर से सुन रहा था, सुबह भी एक वेबिनार था, तो मुझे अपनी जीबी और अपनी बैटरी की चिंता है कि अचानक बीच में बंद ना हो जाए। तो मैंने अपनी बात कही है और इसके साथ मैं कंक्लूड करूंगा कि जो फिजिकल साइंसेस, सोशल साइंसेस और जो बायोलॉजिकल साइंसेस, इनका जो समन्वय है, इनका जो इंटीग्रेशन है, उससे हमारी नजरिया बदलता है और नजरिया जब बदलेगा तो आपको हर चीज अच्छी दिखाई देगी। और रही बात भारत की, तो भारत आज से नहीं, भारत को बहुत साल पहले कह दिया गया है कि सारे जहां से अच्छा है हिंदुस्तान हमारा। तो उसमें यूरोप और जर्मनी और न्यूयॉर्क और शिकागो की बात नहीं की गई, सारे जहां से अच्छा हिंदुस्तान हमारा। और आप भी कभी गूगल अर्थ लेके बैठिएगा, जो अभी हम बात कर रहे थे कि 2004 में गूगल अर्थ आया, गूगल अर्थ इंजन लेके बैठिएगा, ज्योग्राफी के आप हैं, दुनिया में कोई भी हिस्सा ऐसा अगर आप दिखा दीजिएगा भारत के लिए, चाहे वो गंगा का मैदान हो, चाहे यहाँ पर ग्लेशियर्स हो, चाहे यहाँ का हार्ड डिजर्ट हो, चाहे यहाँ की कोस्टल लोकेशन हो, चाहे यहाँ का प्लेटू हो, दुनिया का कोई भी हिस्सा आप दिखा दीजिएगा तो मैं वाकई मान जाऊंगा आपको, क्योंकि मैंने तो एक्सप्लोर किया है, मुझे तो दुनिया का कोई हिस्सा नजर नहीं आया जो भारत जैसी भौगोलिक जो सुंदरता लिए हो। और मैं अपनी बात यहीं से कहना चाहता हूँ कि तू अपने मन में डूब के पाजा सुरागे जिंदगी, मेरा नहीं बनता तो ना बन, अपना तो बन। क्योंकि आज के जमाने में हमारे जो छात्र हैं उनके लिए मैंने ये शेर कहा है कि ये जो छात्र स्कूलों में हैं, जो छात्र विश्वविद्यालय में हैं, इंस्टीट्यूट में हैं, आप बन जाएंगे तो भारत तो अपने आप बदल जाएगा। और गवर्नमेंट के लोग जब कभी यूनिवर्सिटी में आते हैं तो हम लोग यही बात करते हैं कि सबसे बड़ा इन्वेस्टमेंट जो है हेल्थ के इनोवेशन में आना चाहिए, इन्वेंशन में आना चाहिए, और सबसे बड़ा इन्वेस्टमेंट जो है एजुकेशन के इन्वेंशन में आना चाहिए। और अगर हम सबसे बड़ा इन्वेस्टमेंट अपने बच्चों पे करें तो एक विकसित राष्ट्र का सपना जो हमारे कलाम साहब ने और लोगों ने देखा है, तो उस विकसित राष्ट्र में सपना तो छोड़िए, हम लोग हैं, अगर हम अपना इन्वेस्टमेंट उसमें करें तो आने वाले दिनों में भारत एक सुपर पावर होगा, है और था। धन्यवाद, जय हिंद।
प्रोफेसर रियाज सिद्दीकी सर, जिस तरह से आप सरल व्यक्तित्व के धनी हैं, इस तरह से आप सरलता से पूरे सस्टेनेबल डेवलपमेंट के जो सोशल एस्पेक्ट्स हैं उसको बहुत सरल शब्दों में इतिहास के धरोहर से लाते हुए आपने प्रस्तुत किया। निश्चित तौर पर इस समय बहुत सारे पार्टिसिपेंट जो हमारे छात्र हैं वो जुड़े हुए हैं, तो लग रहा होगा जब ज्योग्राफी के लेक्चर क्लास में हैं और इलाहाबाद यूनिवर्सिटी के हेड ऑफ डिपार्टमेंट ज्योग्राफी के उनको सुन रहे हैं, वो जरूर आनंदित हो रहे होंगे। और बहुत बहुत धन्यवाद आपको, आप जो है जुड़े, और फिर आपसे समय लेंगे कभी हमारे यहाँ KM सिंह लेक्चर सीरीज और आप अवश्य समय देंगे। बहुत बहुत धन्यवाद।
थैंक यू, थैंक यू।
Thank you, Professor Siddiqui ji. Thank you, Dr. Tripathi ji. Professor Siddiqui has delivered very nice and informative, beautifully delivered talk. The science is never separated from arts, and in art there is always a science. So both are integrated inseparably. Now I request Dr. Sunil Kachiria from JNU. He is professor in the Department of Biotechnology, and he is very intensive researcher. I know him from the days when he was appointed in LA University, and now he has moved to JNU. Very good afternoon, Dr. Kachiria. You are most welcome, and I hope you get my voice.
Yeah, I can hear you. I just want to share these things, so you allow me.
Dr. Kachiria.
Yeah.
You are most welcome to deliver your talk.
Thank you very much. So I'm trying to share my screen. Host disabled. So can you enable me, please?
Can you share your slide, Dr. Kachiria? Do you any help? Do you need any help?
No, actually I'm trying, but disabled me. So you have to enable me.
Yeah.
Can you hear me? Can you hear me, sir?
Yeah.
So I am trying to share my screen, but host is disenabling me. So you have to enable me.
Yeah, you are enabled now.
Now it is okay. Fine. Can you see my screen, please?
Yeah.
Oh, can you see?
Slides are visible.
Slides are visible.
Yeah, you can go ahead, Dr. Secretary.
But I can't see. Wait a minute. There's some problem with my.
We'll first you in presentation mode, then share.
Okay, now?
Yeah.
Now it's fine.
Okay, so good afternoon to everyone. Actually, I'm very thankful to organizer, especially Professor Pandey, Professor Tripathi, and Dr. Bandichor. So can you see my slides now?
Yes, yes.
Yeah. So it's really very nice webinar covering very diverse area, and I'm going to talk something very different, and maybe I'm an odd man. So this thing started with very basic biology, and that basic biology is coming really from algae, but you will see how that basically information coming from algae is really helping us to understand biology, and moreover also provide cure for certain basically disorders. So now next slide. So what you will see, my topic is mostly on photoreceptor, and these are natural photoreceptors which you basically see that functional in the biological system all the time. And what we see that this photosynthesis, which is very important component on the planet. Hello? Is everything okay with the slides, sir?
Yes, 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 yes,
Okay.
sir.
Yeah, it's okay.
Okay. So this thing, because of this, basically this planet has oxygen and life is surviving. And photoreceptor also very much important in vision. And this vision exists start from bacteria to human. But of course, in bacteria or lower organism, the vision is not like camera-like vision. They have chemical sensing or other kind of sensing, but nevertheless they see. And third very important component, which is basically photoreceptor operating in all biological system, that is circadian clock. And this circadian clock is a biological clock which is operated in all kind of living organism, start from bacteria to human. And this is coming from a very classical biological science part, that is photobiology. And in photobiology, which we have learned from the 12th class biology and even higher place, so it is having main two components. One is light, and we talk about light all the time. And then second component is photoreceptor. But I just want to tell one thing, this is little different than the photosynthesis, because in photosynthesis, you have a photopigment, not photoreceptor. So today my work will especially focus on photoreceptors, not on photopigment. But the message is that light is very important for the biological system and survival of life on Earth. And this is basically three very important things. So when we talk about sensory photoreceptors, these basically, there are different kinds of sensory photoreceptor in the nature, and photoreceptor always have two components. One is protein component, another is chromophore. And what we can see at the chromophore side, that we have basically tryptophan chromophore, flavin chromophore, retinal phytochromoglobulin. And these different chromophore combined with the different protein, and then they give you UVR photoreceptors, light oxygen voltage photoreceptor, blue light using molybdenum photoreceptor, cryptochrome, rhodopsins, and phytochrome. So today I will talk only about two proteins, that is blue light using flavin domain and rhodopsin. So all photoreceptor have two components. One is chromophore, another is photoreceptor protein. And another very important hallmark for the photoreceptor, that they have a photocycle. Photocycle means molecule is in a dark state where basically it is not giving any information to cell, organism, and system. Another is signaling. So when it basically absorbs appropriate light, and that absorption of light is done through chromophore, then it goes into signaling state, and then signaling is happening in the system. So a very important point we're going to bring, that sometimes very irrelevant basic biology can give you entirely new thing, and sometimes solutions for human being. So this basically thing started with very basic science that we were looking for photoreceptor, which is important for this green algae, which is present in our pond all the time in around. And this organism shows two kinds of response. One is phototaxic, that is moving towards light or away from light. Another is photophobic. So photophobic means when light is too much, then organism just stop and then take a decision where to go and what to do. And this kind of basically behavior is important for survival because it try to find out amicable condition where it can go and survive. So these two basically systems are operated by a protein which is rhodopsin, and rhodopsin, by the way, is also present in our human eyes. But definitely these organisms have different kind of rhodopsin what we have. So this was known, but the thing is it was not known whether that molecule is really functional or not. So this is little bit from the history. So history in sense that it's my PhD work. So we were given a task to find out photoreceptor for the photobehavioral responses which are operated in this green algae. And green algae is all doing to try to find right light so that it can do appropriate for photosynthesis and survive. So we found gene by doing bioinformatics, molecular biology, and then we try to assess the function. We try different systems, and finally we got solution from a very strange organism, and this is basically Genopus frog, which is South African frog. And in this South African frog, so can you hear me now? Hello. So the South African frog, basically we isolate these basically O cells and then inject the mRNA. Once mRNA is injected, and then we do electrophysiology, and from electrophysiology we can characterize whether this is a functional or not. So what we have got here, that when you show light, where which is basically the gene which was encoding rhodopsin from algae, upon light basically give a current, and then same oocyte if you give with the red light, you don't see any current. So this particular experiment gave information that protein can have the electrical activity when you express in your right system. And this was a revolutionary point for 2 points of thing. One is that for basic science that we identified the photoreceptor, and second thing, soon people realized that such protein can be expressed in other human cell or other kind of cells where we can change electrical activity. So by doing this basic molecular biology, biochemistry, cell biology, we have identified many rhodopsins, and I am not going to talk about all these. So these are the basically different type of rhodopsins which are present in this green algae. So the first thing was solved that, okay, this photoreceptor is for phototaxic and photophobic response. And then people have realized application. And now the new thing is started, application in the area of optogenetics. And soon I will be talking about that optogenetics. So here optogenetics, as it basically says, opto means light and genetics means genetically encodable. So you have a gene, you take this gene, go to a cell or system, and when you express this thing, then you can immediately basically get the function which you want. So light regulated gene is transferred to a system, and once it is transferred and it is expressing in that system, you can sign appropriate light, and once you sign light, then you can control the cellular response. So this was the concept which was taken to neuroscience first. So now just remember for student side that we started with the green algae and we reached up to imagining human brain. So in human brain, what happened when we want to learn about the signaling, we try to get the electrical activity from outside and then initiate a signaling and then get the signal. So this method was going on, but the drawback with this method is that when you insert electrode in cell tissue or organism, that time what happened that you are basically, so you are basically so you are increasing background electrical current as well as it is interfering in the electrical thing. So when we got our protein, so now what will happen, you take that protein and put into the neuron where you want, and then you can have signal only from that neuron. So this concept was just for the one protein, that rhodopsin, but people have also realized that other light sensitive protein can be utilized for this purpose. For example, here you see light-gated ion channel. That is our channel option which identified from Alki. This phytochrome coming from the plant, cryptochrome is also coming from plant, and then light oxygen voltage domain coming from yeah. So somebody is raising hand. So any question at this stage? Somebody is raising hand.
Continue.
Okay, I will continue. Okay, so here what you see that these different proteins can bring different function. For example, light-gated ion channel, when you express in different cells, you can control neuron. And then if you express phytochrome, then you can change the protein-protein interaction. And by cryptochrome, you can change gene expression. And here with this one, you can modulate basically modulated gene expression system. So light-sensitive proteins are used in the system to control cellular events and externally. So we went to another very interesting organism, I think botanist knows very well, and some geologist, with Euglena. And in Euglena, what happened that this also shows phototaxic. And around 20-30 years, people were trying to find out photoreceptor, which one is responsible for this phototaxic. Finally, basically, it is done by a Japanese group who basically found. Hello, is there any question? Because again, I see somebody raising hand.
Again, continue.
Okay, so we will discuss question at the end. Then it is nice. Okay. So
no problem, Priya, you may continue.
Yeah. So here basically you see that this organism shows phototaxic, and then people have identified the protein, and this protein is having two domains. Here blue light you see flavin and cyclase. And when you take this protein to a system, here basically human embryo kidney cell, and then you can see when I am having no gene, then there is a less calcium current. And how calcium current is coming into picture, because if it is a light-gated alanine cyclase, so it can change the CMP or CGMP concentration in the cell. And if it is changing that one, then CMP and CGMP can bind with the CGMP gated channel. And once it is binding with the channel get open, and then you have a calcium influx. And this calcium influx is measured by fluorescent dye. And so what you can see in the B panel, that when we are expressing this gene and shining light on it, so then the calcium fluorescence is coming up. So this was the first time when we showed that yes, you can take the light sensitive protein and you can express them in different systems, and you can basically operate that activity what was happening in very different organisms. So creating basically opportunity to control other biological systems. So with this concept, we move to some more organism, because more organism and more protein. So here the lower panel where you have two blood domain and two cyclase domain. So you have two basically light sensor and two enzymatic. And this normally give a problem in terms of kinetics and controlling. So we thought of finding something which is having a simpler system. So what you see in upper panel, we have a one sensor and one cyclase, and these proteins were present in a Nigeria blueberry. This is another small protozoa, and which is basically very much free-living. But the one species is also causing disease, that is brain dead disease. So we are not working on the pathogenic one, but we were working on the non-pathogenic one. So we isolated these three genes, and then of course this is the workflow which we adopted. So we identified the gene, and then we basically did all bioinformatics analysis, and then we took that gene and express it, purify it, and we do electrophysiology and all other kind of characterization, especially spectroscopy and enzymatic one. So what you can see in bioinformatics is, so do not get worried about this complicated picture, but we just mention that the amino acids which are marked by the different, basically the shape here, pink shape or real kind of thing. So these are the amino acids which you can see common in all basically blood domain, irrespective whether they are coming from bacteria or protozoa or it is coming from other system, Euglena. So this suggests that, okay, yes, if you have such protein, that it may get expressed and you may have the function of the protein. Then we also have analyzed the cyclase domain. In cyclase domain also you can see these important basically amino acids which are marked with the triangles, they are conserved. And these are the amino acids which are important for the binding of your ATP or GTP, and that is important for the enzymatic function. So that was also conserved. So it gave us basically very good confidence that when you move to expression system, probably you get the protein and you can control your biological system by the light. So what you see here, this was the classical basically spectra. When you have a photoreceptor function, then you will see these are the basically flavin biopronic peak associated with the protein, and here you have a protein, and this should happen with the functional protein. So we adopted similar system, and what you can see, we have all these three proteins which are expressed, purified, and all three proteins have the typical characteristic for the block domain, that is like using flavin. But of course, they have different kind of lambda max, they have different kind of the absorbance, and that is happening because these are having different protein sequence than the one. So we have characterized their basically lambda max, and you can see that they show at least some plasticity at the lambda max, and which basically enable you, when you go to different system, you can use different light, and you can control the biological system with a different light system. So as I told you earlier, that all photoreceptor have two systems where dark state and light state, and this can go back again into dark state, and this cycle can go million times, and that's why this is the hallmark. So at the right panel upper side, what you see that this was the published data where you see when you have a photoreceptor, you give dark state spectra, then you give a light, it goes into signaling, and it come back into again dark state. So we adopted the same method, that you have a purified protein, then you acquire the dark state spectra, and then you go to UV visible spectra with the light, and then after that you can see whether difference is happening or not. So in these all to light, and they do the basically shift towards the right shift, and they all have different kind of photodynamics, and that is basically summarized in a table. So the different dynamics means now the different protein have different kind of signaling state. So it means that you shine a light, and then that particular protein will open for that much time to give you cyclase inside the cell, and that may be sufficient for your initiating biological function. So this also basically suggests that this can be a functional protein, and they can have different recovery time, and that gives different toolkit to operate into the system. So now the question is, we have a light sensitivity, it does basically dynamic, do we have enzymatic activity? So this panel ABC on the right side basically shows what we are doing here. We take that expressed purified protein, and then basically we do analyze cyclase activity with the dark, black is dark, and blue, when we give the blue light, that is basically blue bar. So you can see when I'm giving light for different time window, then it is increasing the CMP concentration in the in vitro system. So now this is an enzymatic proof, and giving basically in vitro proof that yes, all three proteins are giving you light-gated cyclase activity, and this can be utilized for the system when you are using it. And these are the basically different numbers which tell that how much CMP will be produced when you are working on these things. And that will be important because all cells may not need same level of CMP and cGMP. They all need different kind of CMP and cGMP. So that time you need different kind of toolkit. So we taken that concept, now we go to real thing, whether we can do something in mammalian cell or not. This was all in the cell. So what you can see in mammalian cell, we can basically increase this one. So here concept is that CMP is a very important molecule, play a prominent role in mammalian cell physiology. And they basically work on three different proteins. You have a GEF, CNG channel, and PK. So what will happen that you activate the CMP somehow, and in our case, we will activate with the light, and then this will basically activate the PKA, protein kinase A, and that goes under autophosphorylation, and then it can regulate the gene expression. So let's see whether we can achieve that thing in human cell or not. So this is the classical basically CMP signaling, that you increase the CMP by either internal input, or it is biochemical, or here light-gated basically cyclase one. So we are expressing this cyclase into the cell, and then by shining light we increase the CMP. This will activate basically protein kinase A, and protein kinase A will phosphorylate CRV transcription factor, and that will regulate all other genes. And these genes control development of learning, memory, metabolic regulation, immune regulation, cell growth. So what it means, principle, that if I am able to control cell CMP by using this optically active gene, then I can learn about development of memory, learning, metabolic regulation, immune response, cell growth. So we have focused only on one aspect, cell growth and survival for proof of concept. And then we were seeing that whether this kind of signaling can increase expression of cyclooxygenase and followed by prostaglandin, which is basically downstream molecule. So let's see what we have here. So what we have done, we have expressed this gene into mammalian system. So this A panel suggests that yes, now you are able to express these different gene, and this is the internal control where I am showing that I have loaded protein in every cell, and then you can see that only in the cells where we have expressing this thing, we see protein presence. And this basically is so once we got the protein, then we have asked another question whether this light regulated CMP production is happening in the mammalian cell or not. So we did similar experiment that you take cells with and without their different construct, and then you keep them in dark, and also you give the blue light, and then by shining blue light, what you can see here with the blue bar, that CMP is increasing. But of course, the amplitude of the CMP increased by different protein is different because the protein sequence is different, and that sometimes is very important, very good, because you can have different amount of CMP when you want to think of application purpose. So these are the basically different number which you can have 10-fold, 3-fold, and 17-fold CMP in the system, which you can regulate by this one. So so far I have shown that. So the upper panel is the method where we are working on that, and then what you can see this here lower. So we are able to express this one blue light activated cyclase, increase the CMP, PK. Now we want to see whether this transcription factor is phosphorylated or not. So we are at that step, the CRV, that's why we are highlighting. So what you can see here on the right panel, upper panel, that the different basically panel suggests that when you express this gene into mammalian cell and you shine the light, then the CRV transcription factor phosphorylation is increasing as you increase the duration of light and expression of this one. And in the F panel, we have quantified that how much CGM basically phosphorylation is happening. So in some case, you can see that the difference is not much, but we have to see whether this much difference is sufficient to initiate downstream gene regulation followed by overexpression of cyclooxygenase, and then having the prostaglandin thing. So we are able to control this phosphorylation of the transcription factor by using light gated cyclase. Then we move to next level whether we can have a cyclooxygenase production or not. So what you can see here that we have quantified by the by this one, and then upon increasing light, and so black bar is very little, and when we give the light, this blue light, and then you can see cyclooxygenase is increasing in all three constructs. So we are able to initiate cyclooxygenase expression by shining light on the different system. So we can do this thing. Now next question is, if that is the case, can you control it downstream signaling, that is prostaglandin synthesis? So this was the final experiment from this study. So we were able to do it, and then it was published in Nature Scientific Report 2017. So this was the concept that we started with the basic photobiology, and in basic photobiology we focus on the photoreceptors, and that two photoreceptors coming from the green algae and Eugleena, and how this green algae photoreceptor was useful for the optogenetic modulation in neuroscience. And then we shift to one light activated cyclase coming from the Eugleena and Protozoa to utilize and change the CMP, CGMP in a given cell, tissue or system, and finally basically have this thing. So then question is, it is only for basically neuroscience or cell work? That is not the case. It can be basically utilized for the basic science research, that is neuroscience, cell biology, cardiac physiology. So concept is very simple. If you want to change the electrical activity, you can change using this system in a biological setting. If you want to change the CMP, CGMP, you can use this one. So then the clinical application wise, this thing is utilized nowadays for Parkinson's disease, epilepsy, depression, obsessive compulsive disorder, chronic pain, vision disorder, and I will highlight one of these vision disorder soon, and then we will see. Also, people are trying to correct spinal cord injury by utilizing this opportunity. And the last thing which we are developing in our lab, can we do drug discovery? Today I do not have time to explain all those things, but at least I can focus on one, that is vision disorder. And this is very relevant to our society because unfortunately, India is the largest, basically India is having largest population which cannot see properly, or we can say blind population. So this is a very sad thing. And another basically disappointing point, not much research is going on to do that because people are able to manage their life. So their morbidity is there, but it is manageable, so people do not care. So I'm showing just one vision disorder, which was basically showing promise to correct the human vision, and that is retinitis pigmentosa. So what happened in this one? So on the left side, when my vision is right, I will see full picture. There is no problem. But if I have genetic disorder, and because of that retinitis pigmentosa, what happened that rhodopsin is degraded in my eyes around this basically area, and that's why I have a ring-like vision. I cannot see full picture, I just see ring, and this grows on with the time, and then eventually I get completely blind, I can't see much. So what people have shown in the mice first, and then now it is up to human clinical trial, and this human clinical trial is also going on in Switzerland. They have taken that algal channel rhodopsin protein, and they put into basically these mice, and after that these retinitis pigmentosa mice were able to initiate the vision evoked potential. So this was really a very nice piece of work, which I can say that it started with the algal basic science, and then we reached up to this level. Okay, so I will be finishing quickly. So this is basically human trial which is going on in Switzerland, and they are already on track, and hopefully that someday it will be available for our Indian population or other places same way. So the question is, then we are having just only these things. We are also developing some more optogenetic tool where we can change the nucleus thing, we can change transcription factor, we can change metabolite, we can change cation and anion, we can change cytoskeleton by using light tweeters and light actuators basically. And so I'm coming to my second last slide, which is very important for the basically research purpose and all other things, acknowledgement. So this kind of work is possible only because of two things, that I have very great collaboration with people, and also I am well supported by different funding agencies at national and international level. So because of these two things, I'm able to deliver such interesting research in India. So, and I'm also very thankful to basically these beautiful natural organelle systems, which are basically given opportunity us to find something interesting and useful for human being. And I'm thankful to all of you for paying attention and listening to my talk. And now I'm ready to take questions if any question is there. Thanks a lot.
Thank you, Dr. Sunil Kataria, for very informative and highly relevant study you have presented. I request Dr. Ajay Vikram to express his views on your presentation and for a healthy discussion.
Yeah, thank you, Sunil. It was very interesting, actually, how you took a basic science and you guys are, translating this into the medicine and to cure such a massive disorders in, especially in developing countries. And we need such vision, basically to pull the basic science to such a high level. So I really congratulate you, and it's really impressive. So few of the questions that I have in mind. So this clinical trial, I mean, you guys have already shown in vitro that system works in vitro system, and when it is incorporated and expressed in vivo, it shall work something similar.
Okay, I tell you. So here many things. So first of all, this is not that I have all the studies done. So my basic research was that to show that such protein exists for basic science purpose, and then it was taken to optogenetics, and then people took to neuroscience. So the mice experiment were done by a group, and the human trial is done by the company. So this is, and of course, India is not involved because this was work done at Germany, and so it was taken by the European system. But nevertheless, I'm happy that this is seeing basically product light and very translational one, and they are showing promising. Okay, now coming back to this thing, whether it is working, so it works in not only in vitro, because if you have in vitro, then there is no question for clinical and all those things. It works in vitro, it works in mice system, and then once it was built or characterized at the molecular level, electrophysiology level, and also pathophysiology level, disease biology level. Then it was taken to human trial, because getting human trial is not straightforward. They also want to see all kind of evidences. So this was done, but then the thing is that now the outcome of human trial will tell how successful it is. And the only thing, if you are asking whether it is working similar way or not, so definitely it is in a different way, because our rhodopsin in eye is different than the bacterial one.
True.
But what they are doing, so this particular therapy is working at the neural level. So not that the control is done at the imaging level, it is done at the neural level. So you got changing the electrical current only. So these people will be able to see only in a bright light. But it's okay. Even if they see in a bright
Yeah,
light,
of course, that's a big achievement. Yeah.
a big achievement. So this was basically a thing. So definitely it worked in a different way, because in our eye at the end you need hyperpolarization, and this basically do depolarization. So they are working at the place where the depolarization is needed in the mammalian system, okay? Not that hyperpolarization system. So like then right now. So definitely it is at the little bit upper level of the rhodopsin, because the rhodopsin signaling is over, and this gives the electrical current. So you are trying to change electrical current directly without passing that rhodopsin business. Yeah. So that's why it is basically not that we are depend on the classical human signaling pathway. We are depend on the electrical current per se. So as long as electrical current is well coordinated with the optical neural system, it should integrate.
Yeah. And of course, I mean, the light optogenetics, I mean, something optical controlled thing will be a very novel technology, not in medicine, but also in, I mean, different fields. For example, we work in micro nano robotics, and we are always looking for tiny animals. Those can propel the things into fluid like the blood. So basically, the biggest challenge currently, even in the medicine, I mean, normal pharmacology, is deliver the drug at the target site. So the idea is to design these tiny robots filled with the drug and some well-controlled, optically controlled, magnetic controlled thing that can push them inside, with so I mean, this is a very fascinating system. Good one.
Actually, now a lot of things are merging together. So even nanoparticles, so you can, the right question is that how will you deliver light to brain or other system? So so this was solved by the nanobiologists. So you must be aware about the upconversion nanoparticle.
Yeah.
So you take this upconversion nanoparticle to the neural system where you want to activate these things, and then basically this upconversion nanoparticle can absorb infrared light and it can emit the visual. So you don't have to put optical probe invasive way
inside.
Inside. So this has been done just 5 years ago by different Japanese group, and now it is like booming like anything, because that is the one. Otherwise, it was a hurdle that you can't poke the optic fiber everywhere in the brain, heart, and all those things. You can't do. So I think the idea is that once you float some idea, then technologies people will join, and this kind of research need multidisciplinary, transdisciplinary.
Exactly.
You can't have just one biologist to do all those things. Engineers are involved, chemists are involved, laser physics is involved, and many people are there. So that's why, and that is the reason I have extensive collaboration, national and international collaboration.
That's very nice. Good. Congratulations. Thank you. Yeah.
Sunil, Rakesh here. I think this is a fantastic talk. And I was just trying to comprehend in the area of photodynamic therapy. So what happens, usually there is a conjugated system that is to be given to the patient. And when you apply the light, it absorbs light and goes from the
You have muted yourself, Rakesh. Unmute it, please.
So what I was looking at, if you find a gene which you can express to have a protein, and that protein is capable of harvesting light, right? To a level that can
So actually the thing is that then you have to use basically photodynamic therapy work in little different way. They have this UV caged or other light sensitive caged component, and that is delivered by the nano carriers or other places. So I think ideal would be that if we have a protein based nanoparticle, and this protein based nanoparticle go unloaded with your drug which you want to take it. But the second challenge will come how to deliver at the right place. And I think all nanotechnology basically suffer with that. And to find this one, once you are at the right place, then I think even photodynamic therapy without the photoreceptor protein is also doing very good job, especially in cancer thing. But here another opportunity is coming, Dr. Rakesh, that is basically photoimmunology. So what you can do, you can take these basically genetically encodable cells, and if you are able to regulate immune response itself, and that immune response is targeted to mostly the cancerous tissue or the neighboring area, and it in such a way that you can basically evoke and kill at that itself, which we have done recently with the HIV. So what we did basically latency of the HIV we have broken by optogenetic method. So we simply deploy our CGMP gene to the viral system, this viral and cellular system, and then we have increased the CMP and CGMP, and because of that, this hiding virus which was not expressing, it basically came into system, and that therapy was going on, then it basically killed it. So if we are able to control that immunological aspect directly to the place where basically you have to deliver, then definitely it can be done. And your idea is also good that if you are able to load different drug and also activate by the different photoreceptor protein. Say there is no problem with the different photoreceptor protein right now, but their stability is a big question. You cannot, so for example, rhodopsins are very difficult and tricky protein, membrane protein. So you can't play them so robustly with the chemical tagging, loading and all kind of things. But definitely soluble protein can do that job. Okay. So the protein stability itself is a question. So it is better not to work at the protein level, take these genes which are encoding protein to a system, and then you try to control at that level.
That's a fair point. Yeah.
Any other question, please?
Thank you, Dr. Sunil, for sparing your time. Now this is time to next speaker, Dr. Madhukar Misra. Dr. Madhukar Misra.
Yes, sir, I am here.
Welcome. Welcome, Dr. Madhukar Misra. Madhukar Misra is Associate Professor in Department of Physics at BITS Ilani, and he is most welcome to deliver his lecture.
Yes, sir. One minute. I am sharing my screen. Okay. One minute. Yes. Next slide is visible?
Yes, yeah.
Okay. Can I start with this slide?
Your slides are visible. You can start.
Okay, okay, sir. Okay. Okay, so first I would like to thank the organizer of this conference for inviting me to deliver this talk. So the topic of the talk is the behavior of matter at extreme high density and dark temperature. So this is the brief outline of my talk. First, I will give you just some small motivation behind the topic, and then introduction, and then some challenges. And then I will concentrate on a particular work which is related to the Quaternion production and suppression. And then finally I will summarize my work. Okay, so let us start from this brief motivation. So, in fact, there are two parts in this talk. In the first part, I will try to give you the brief information about particle physics or high-level physics, which is the broad area of my topic. So, the particle physics or high-level physics especially moves around two fundamental questions of this universe, and the questions are what are the most fundamental building blocks of a matter and how they interact among themselves, how this universe in fact work, how does this micro-world behave. Micro-world means the world of electrons, protons, neutrons, neutrinos, and so many other particles. How these particles, how these micro-world behave, and how does this initial material world work? So these are the two fundamental questions of this universe. So the most of the time, this particle physics or high-energy physics revolves around these fundamental questions. Since particle physics deals with the study of matter at extreme high density and temperature, and that is why it requires huge amount of energy, and that is why it is called high energy physics. When we talk about the interaction between the particles of the micro world, then we need to talk about fundamental interactions. In fact, there are four fundamental interactions. First one is electromagnetic interaction, and the corresponding field theory is called quantum electrodynamics. And the second one is deep interaction, which is responsible for the beta decay process. And then gravitational interaction and the quantum field theory associated with the gravitational interaction is called the quantum gravity. And then the strong interaction in fact binds the quarks inside the proton and neutron. And the corresponding quantum field theory is called the quantum chromodynamics. So there are four fundamental interactions, and corresponding to each fundamental interaction we have a quantum field theory. So, in fact, what happens, suppose you have some matter, and then if we start providing certain amount of energy to the matter, then matter will be divided in terms of molecules. If we further provide energy to the matter, then the molecules will break up in terms of atoms, and again if energy provided to the atoms, atom will break up in terms of nucleus and electron. And if we further provide energy, then the electron will come out from the atom, then it will get a nucleus. And nucleus is not the smallest particle, it can be further divided into other smaller particles, I mean proton and neutron. And by doing experiments and by developing some theories, it has been found that even proton and neutron, they are not very big particles, they are made up of some other smaller particles, and these smaller particles are called as quarks. So how these quarks bind inside the proton and neutron, this is described by the strong interaction and corresponding quantum field theory is called quantum chromodynamics. Okay. So what is the property? What is the property of this matter? What is the property of the matter containing quarks? And these quarks, they are held together by a force which is described by QCD, and actually when there is a force between two particles, then there is an exchange of the third particle. This is a very basic concept of the quantum field theory. So when two quarks interact, then that interaction happens due to the exchange of the third particle, and the third particle is called the gluon. So gluon is the mediator of the strong interaction, strong force. So what will happen if we keep on providing energy to the material, then the material will be divided in terms of quarks and gluons, and this type of material is called the quark-gluon plasma. So the main question is, what is, whether this quark-gluon plasma can be produced in the laboratory or it's not, or it cannot be produced, or it cannot be produced in the laboratory. If it is produced in the laboratory, then what are the properties of this quark-gluon plasma? Properties means what will be the thermal conductivity, what will be the electrical conductivity, what will be the transport properties, and what are the different thermodynamical properties of this quark-gluon plasma. So this is extremely tedious work in order to find out the properties of this quark-gluon plasma. So the properties of the matter at extreme high density and temperature means the property of this quark matter. And this quark matter, this quark-gluon matter, if this is found, in the experiment, then this will exist only for a very small interval of time, and suddenly it will be converted into a normal electronic process. Okay, and this type of this phase transition is is called the hydronization. Okay, so and the next question is there any correlation between the matter properties at extreme low temperature and matter at extreme high temperature? So quark-gluon plasma is a matter at extreme high temperature, and matter at extreme low temperature, the cold atomic gases. So cold atomic gases, whether is there any link between the properties of the cold atomic gases and the quark-gluon plasma? This is another question. And after the study and after the experiment, it has been found that there is some link between the properties of the matter at extreme low temperature and the properties of the matter at extreme high temperature. So, okay. So it is explained to me that this type of matter, the quark-gluon plasma, this was formed a few microseconds of the Big Bang. Okay, so this quark-gluon plasma was the initial stage of this universe. Okay, and it is also believed that this type of matter still exists inside the core of celestial objects like neutron stars. Okay, so various phenomena people are observing which are related to the neutron star, and in order to explain these phenomena, people are developing different theories. And in developing these theories related to the neutron star, they are assuming that neutron star is in fact made up of quark-gluon plasma. People are assuming that they are made up of some other materials. Okay. So the matter at extreme high temperature, the quark-gluon plasma, this is studied theoretically using QCD, quantum thermodynamics, the quantum field theory, corresponding to strong interaction. So there are two parts of the quantum thermodynamics. One is called the perturbative QCD, and another one is called the non-perturbative QCD. Perturbative QCD means the matter at extreme high temperature, and non-perturbative QCD means matter at slightly lower temperature, not at very high temperature. So what happens when we study matter at extreme high temperature, then actually there are two important properties of the QCD. One is called the asymptotic freedom, and another one is called the quark confinement or color confinement. So asymptotic freedom, what is asymptotic freedom? Asymptotic freedom corresponds to the property of QCD at very, very high temperature, extremely high temperature. So we know the force acting between the two charged particles, which is called the electromagnetic force. And this electromagnetic force is very well understood, and the corresponding field theory we know this is called the quantum electrodynamics. Why in quantum electrodynamics is well established? This quantum electrodynamics is well established since various observations related to the quantum electrodynamics are completely verified by the experiments. Okay, so that is why QED is well established, and in QED, what happens when we have the two charge particles, the force acting between the two charge particles increases when we decrease the distance, and the force, and the same force decreases when we increase the distance. This is the property, this is the force which is given by the Coulomb's law. But what happens in the case of QCD? What happens in the case when we have two quarks? When we have two quarks, then the force or the interaction is governed by the rule of QCD, not by the rule of QED. In QCD, situation is completely different. When we have the two quarks, then the force acting in the two quarks decreases when we decrease the distance between the quarks, and the same force increases when we increase the distance between the two quarks. This is completely opposite to what happens in the case of QED. So the force, the strong force, becomes very, very big at extremely lower distance, at extremely smaller distance, and smaller distance, small distance corresponds to high energy scale. So at the high energy scale or at the low distance scale, this QCD force, the strong force becomes very, very big, and this is called the asymptotic freedom. And the 2004 Nobel Prize was given to the discoverer of this asymptotic freedom. So asymptotic freedom is a very, very important property of the QCD, and this corresponds to study of matter at extreme high temperature. Quark confinement is the another extreme of QCD, and this happens at lower temperature. So at a lower temperature, with the quark confinement, quark cannot be isolated. Quark cannot be isolated. We have actually hadrons. Hadrons, they are, this is the name of a family of particles, and hadrons are divided into two groups, baryons and mesons. So baryons consist of 3 quarks and mesons consist of 2 quarks, 1 quark and 1 antiquark. So we try to remove quark and antiquark from the mesons by providing energy. We cannot do that. One cannot do that. By doing experiments, one cannot isolate single quark. This happens due to the color confinement or quark confinement. We don't have any mathematical proof or experimental proof for quark confinement or color confinement. Color confinement or quark confinement, this has been proposed in order to explain this phenomenon, in order to explain that no isolated quark is observed. No isolated quarks, isolated quark cannot be observed. This is the experimental fact, and this is explained by proposing quark confinement or color confinement. So asymptotic freedom is well established. Asymptotic freedom is mathematically developed. The theory of asymptotic freedom is mathematically developed, and but quark confinement or color confinement is not well understood. It is just a concept. Maybe it may be explained commercially, but so far it has not been understood very well. Okay, so this asymptotic freedom tells that when we provide energy to the material, then beyond a certain energy, quarks inside the baryons or quarks inside the mesons could become almost free, and baryons and mesons would dissolve, or proton and neutrons inside the nucleus would dissolve, and we would get a matter consisting of quarks and gluon, and this The confined state of matter is called the quark-gluon plasma. So whether this quark-gluon plasma exists or this quark-gluon plasma doesn't exist, if this exists, then what are the properties of this? This is one of the very important and of the high energy physics. So how to provide this huge amount of energy to the nature? Okay, we cannot provide this huge amount of energy by putting matter into a furnace. Okay, we cannot do that. This is done by colliding two heavy nuclei, or this is done by colliding two elementary. Nucleons. Two nucleons are two nuclei. We can first accelerate this nuclei or nucleon, and then we can allow to collide, and then due to this collision, huge amount of energy is released, and that energy is used in order to dissolve proton and neutron. And then one can observe quark and gluon. The matter consisting of quark and gluon, and then one can study the properties of quark-gluon plasma. But again, there is another problem here. Designing this type of experiment in which we accelerate these nucleons or nuclei up to a very, very high energy. This is very, very tedious work. This is not a very simple work. This is a very, very complicated work. And this type of experiment becomes very, very costly, extremely costly, and this is not possible to build by a single country. So most of the experiments related to high energy physics, they are just a very large collaborative experiment in which several professors, scientists, and engineers belonging to different countries across all over the world are involved in building experiment, accessing data, analyzing data via grid computing network in almost real time. Okay, so due to involvement of the several people from very remote locations poses many challenges. They cannot frequently visit the experiment. So frequent visits of so many people from different countries to access data is very, very costly and impractical too. So ever since forces scientists to think about the solution of this problem, and due to this problem, scientists at CERN discovered two important facilities, two important technologies. One is called the grid computing, worldwide Large Hadron Collider computing grid facility, and other one is very important component of the internet, and that is called World Wide Web. WWW. This WWW was invented at CERN by a British scientist, Tim Berners-Lee. Okay, so we can say that the very important component of the internet was discovered at CERN, which is called European Center for Nuclear Research. This is the biggest laboratory of this universe, biggest laboratory of this world. Okay, so one can say that this grid computing and this worldwide web WWW are the byproducts of the particle physics. These are the byproducts of the high energy physics. Okay, so few experiments here: Large Hadron Collider at CERN, Relativistic Hadron Collider experiment at Buffalo National Laboratory, USA. These are the few machines. These are the collider experiments in which both the particles are accelerated at a very high energy by providing huge amount of electric field, and magnetic field is also used in order to bend the particles. Okay, so the main important component of these machines are accelerators and detectors. Accelerators are used in order to accelerate the particles so that we could get huge amount of energy after their collision. And detectors, they behave just like a telescope, or they behave just like a microscope. I mean, they detect the particles emitted input at the time of production. Okay. So, so what happens? So there are many other outcomes. There are many other important outcomes of the particle physics, like PET tomography, positron emission tomography, and then cancer treatment using cobalt-60. This cancer treatment using cobalt-60. This machine is nothing but a portable tabletop accelerator experiment, accelerator facility, which is used at many places. And then the next example of this outcome of the particle physics is MRI machine, magnetic resonance imaging, in which nuclear magnetic moment of the nucleus is used in order to find out, image of the internal organ of the body. And few other areas of the high energy physics, QCD phenomenology, quantum chromodynamics phenomenology. There are various predictions of the quantum chromodynamics, QDP, quantum quark-gluon plasma. This is one of the prediction of the quantum chromodynamics based on the asymptotic freedom. Similarly, there are various predictions which are given by the quantum chromodynamics, and so we need to validate the predictions of the quantum chromodynamics, and this is called the QCD phenomenology. The standard model of the particle physics. There are other quantum theories associated with the other fundamental forces. We need to validate these theories. So, in order to validate these, predictions of these theories, we need to develop experiments, and few experiments are called fixed target experiments, and few experiments are called collider experiments. We call collider experiments when both the nuclei are accelerated and then they are allowed to collide. When we fix one nucleus and other nucleus is accelerated and they are allowed to collide, then we call fixed target experiments. So fixed target experiments have now become very old experiments. So the current experiments are collider experiments. Okay, so other areas of high energy physics is neutrino physics. Neutrino physics is also a very important area in which we study about neutrino mass. Earlier it was thought that neutrinos are massless particles, but now it has been found that they are massive particles, they are not massless particles, although they have very tiny mass, very small mass, but they are not massless particles. So neutrino mass and neutrino oscillations, in fact there are three types of neutrino, there are three flavors of neutrino. One is called electron neutrino, and then muon neutrino and the tau neutrino, which are called the three flavors of the neutrino. And these and in fact the neutrino, one flavor of neutrino is converted into another flavor of neutrino, and then another flavor of neutrino is converted into other flavor of neutrino. So the conversion of neutrino from one flavor to another flavor, this is called a neutrino oscillation. So neutrino oscillation is not the normal oscillation, just like oscillation of a simple pendulum or vibration of some object. Neutrino oscillation is the conversion of one flavor of neutrino to another flavor of neutrino. So this neutrino oscillation is very, very important, and it explains, explain the atmospheric neutrino problem, which is a very important observation related to the neutrino physics. So INO experiment, Indian Neutrino Observatory, this experiment is being built in India. Ice Cube experiment, Super Chemical experiment in Japan. These are the same experiments related to the neutrino physics. Physics beyond standard models, super symmetry, super gravity, extreme theories. These are the some. Okay, sir, I'm taking too much time.
Yeah.
Okay, fine. 5 minutes I can take?
Okay.
Okay, fine. Okay, so there are few other challenges, but I am just speaking. Speaking that. So the next topic is related to the quantum physics, and first is the matter which is found in the Heavian collision experiment that suddenly converted into normal hadronic matter. So we cannot directly observe it. So this matter is observed indirectly by measuring certain observables, and charminium suppression is one of the observables. What is charminium suppression? So charminium suppression is in fact suppressed production of charminium. Charminium is bound state of C and charminium quark and anti-charminium quark. So this charminium is also produced in the case of nucleus collision, and it also produced in the case of proton collision. So in the case of nucleus collision, I mean how much charminium is produced. So charminium, less number of charminium is produced in the case of nucleus collision as compared to the proton collision, and this suppressed production of charminium is called the charminium suppression. So this is the model that I have recently used, in fact, in order to explain some data from Large Hadron Collider experiment, and these are some details related to the model, and the final result is described in this slide. I'm coming to this slide, the final result. This is the final result. This is experimental data. So our prediction is in fact compared with the experimental data here, and you can see comparatively, reasonably agreement between our theoretical prediction and the experimental observation at the Large Hadron Collider experiments. So on the basis, and this model is in fact assumes that the matter, the quark-gluon plasma, this evolves according to the rules of the hydrodynamics, and this in fact this quark-gluon plasma behaves like a strongly interacting matter having a very small viscosity to entropy ratio, and this small viscosity to entropy ratio is in fact observed in the case of cold atomic gases. So on the basis of this, one can say that the property of quark-gluon plasma is somewhat related to the property of the cold atomic gases. Okay, so there are many challenges. There are some other challenges related to high energy physics, but I'm not going to bring about that. Okay.
Dr. Mishra, thank you very much for giving your valuable time to this conference. The valuable lecture will definitely provide new idea in the conference.
Okay, thank you.
Now, I invite a new guest speaker, very energetic and young speaker, Mr. Tarun Krishna, researcher in the field of artificial intelligence in Dublin University, Ireland. Mr. Tarun Krishna, please deliver your talk.
Hello, am I audible?
Yes, yes.
Yeah, we are hearing.
So, good afternoon, everyone. So, before I start, I would just like to introduce myself. Myself, Tarun Krishna. I'm a first-year PhD student at Dublin City University, and my research topic is in self-supervised learning. But the thing today I will be exploring the ideas of how can we exploit artificial intelligence for sustainability and social good. So it will be not, so the presentation will be more of a, how could we exploit in the direction of social good, but not from a research perspective, but what's going on in there. So when we talk about sustainability, it's very imperative to talk about social good because they go hand in hand. We just can't talk about sustainability without talking about social good. So before we go further, it's very important to know like how we have been using AI in our everyday life. So very first example is in your Gmail. When you use your Gmail, there is a categorization algorithm going on the background, which basically categorizes your email into spam and not spam. And the moment you do some Google search, the recommendation that you see is again a AI algorithm giving you some sort of recommendation based on your queries, whatever you type. Google Maps, we use it in our everyday life. Again, the fingerprint sensor, the selfie camera that you have. When you use your selfie camera, you get a bounding box on your face. That's basically a face detection. That's an AI algorithm is running. Netflix, movie recommendation. Amazon shopping portal, when you search your products, that's an AI recommendation. So it's just like friction, which is unnecessary evil. So AI is just like that. You might agree or disagree, but it's just like that. So if I have to define AI, like what is artificial intelligence? So a very technical way to define this is AI refers to the simulation of human intelligence in machine that are programmed to think like humans and mimic their actions. And if I have to put it in my words, I would say like artificial intelligence is the study of techniques that somehow give the machines that allow machines to learn, to reason, and to act accordingly to attain their goals, whatever. So the goals has to be some sort of task. So just to give you some idea of like the current trends in AI, I would like to display two videos. I'm not sure if I can play them. Let's see.
So you just go back to switch off the presentation mode, and then I think you can play it.
I think it is played. So this one that you see on the left is basically an autonomous vehicle. So the thing is like, this is not a futuristic video. This is something out there. There are cars in Europe and US where they just run in a full autonomous mode. And this problem is very complex. It's just not one single problem of autonomous vehicles. It's amalgamation of different problems. You have to do this. You have to think of this lane detection. You have to think of the cars that are passing by you. You have to think of red light signals. You have to look for zebra crossings. You have to make sure there are no pedestrians. So there are a hell lot of calculations and mathematics going on there to make this thing fully functional. So I think I will just stop this one here, and the other one is like this is the problem of motion control. So you see a robot performing a flip, just like we humans do. So having said that, so the ultimate goal of AI is to somehow to learn from your environment so that you have the ability to reason about it. And once you have this ability to reason, you could perform some sort of goals, or you could act accordingly. That just we do as a human. So we think, we reason, and we take actions accordingly. So, but the question comes as, okay, So the question comes is, okay, AI could do that much thing, cool, that's awesome, but can we exploit it to improve society and fight social injustice? So there are a hell lot of social problems out there, and there are a hell lot of AI research which is going on there. But what we are really interested in is basically we are interested in this gray region, like where we want some AI solutions for our social problems. Because the thing is, like, most of the AI that we are using is wrapped around for high-tech companies, or I would say like for privileged people like us who have access to internet, who have access to smartphones or smart TVs. But what we really want is, can we use this AI to help the deprived parts of this world who still don't have access to mobiles, smartphones, or can we elevate poverty? So when we talk about these goals of social justice, there has to be some sort of plan. There has goals like, okay, we want to look into this direction of security and justice. We want to look in the direction of economic empowerment. And the thing is, there's a whole notion of this sustainable development goals from United Nations, which basically talks about this idea, of security and justice, crisis response, economic empowerment, and education. So if you look at this, chart, this is basically what it describes. So on the left, what you see is basically the UN goals for sustainable development, which is lives below water, affordable and clean energy, clean water and sanitation. And on the right, what you see is the use cases of artificial intelligence that can be used to improve those particular goals. So that means there is like immense challenge for AI for when we are talking about sustainability and social goods. So let's look at these challenges for AI. But the thing is, these are challenges, but as well as there has been solutions for these challenges as well. And there are open problems still for the researchers to look into. So let's look at these challenges. So the first and foremost is basically deepfakes. When I say deepfakes, a simple example is fake news that we almost get on our WhatsApp, and I'm very pretty sure we all must be forwarding 1 and 2 fake messages to each of us without knowing that it's a fake message. So it's a great challenge for us how AI could be used to detect those fake news. And one such video to explain this is this. So you see this. So this is completely a fake video where Obama didn't said anything, but it was presented as he said anything. So this is the problem of deepfakes, and it is very much. And you have to look into it. Just look into the Indian politics scenario at the moment. You'll find a lot of deepfakes video, and you'll get it like how this is being exploited to raise some propaganda or to raise some riots. So this is a problem of social security, and AI has to look into it, and there are solutions for it, but still there are many open challenges for that. So if you see these images, they are really good images, to be honest, but the thing is these people don't really exist in real. So these are AI generated images. These are unknown identities, and the thing is, like, with unknown identities, they are boundless. I don't know, like, there could be many scenarios like where these could be like, these unknown identities could be exploited. So these are the challenges with deepfakes, fake news, and all sorts of things. The second challenge is basically healthcare and medical, and the AI has been quite useful in this directions. I would say, like, now there is a huge dependence of AI in the direction of healthcare and medical. So basically, AI is basically used for assessing this cardiovascular risk factors using using the images of your retina. So there are algorithms for tumor detection where you just pass the image of the particular infected area, and AI will generate like give you the predictions. Okay, is it the tumor? Is it cancerous or is it not cancerous? The recent trend is again, the recent trend is basically on robot-assisted surgery, where a robot could perform the surgery, but it is still a futuristic approach. Although I won't say this is not happening. There are some parts around the world where they are using to perform minor surgeries using robots. And I'm sure, like you might have in like previously in the afternoon, there would have been a talk by Mr. Ajay Singh. So he would have given you some idea of nanorobotics, and AI is being exploited in that directions as well. And who knows, like COVID-19 predictions. Yeah, so during this pandemic, AI has been quite useful. In COVID-19 predictions, AI models are making predictions about like total number of cases in the next week or in the next month, total number of deaths in the next month. So these are mathematical models who make predictions about the future. But you may say, like, why predictions are important? But the question is, predictions are important because you can plan accordingly. So if I say, okay, in the next month there will be 1,000 COVID cases, so you can accordingly order particular number of ventilators. You can arrange that number of beds in your locality in some particular hospital or something. So that's why predictions are important. And this and the thing that you see on the right is basically AI is being exploited to predict or to restructure or to generate protein structures. And this has been one of the big breakthroughs of AIs in year 2019. So AI has been able to predict protein structure. Now you may say, like, why it is so important to know about protein structure. In order to make any drug for any disease or anything, you need to have those protein structures, and it is not easy to predict those protein structure, and AI is being doing it for you. That means it has reduced a hell lot of hours of work from your researchers and doctors. So yeah, so AI has been quite fruitful in this direction of healthcare and medical. And the third is remote sensing. Remote sensing is like there's a huge opportunity for AI being exploited in the direction of remote sensing. So so one is basically damage assessment. So what you see on the left is basically a scene from like a satellite image of a particular region before some sort of storm or cyclone, and what you see on the right is the scene of that particular area, but it is after a storm. And what AI has predicted on the left, you see it has segmented the area, and you can see like there are clear bifurcations between the house, locality, road networks. But once after a storm, you see like there's no clear bifurcation. So this is how you could make damage assessment, and then you could accordingly release packages for that particular area. So that's how AI is being exploited for damage assessment. And these are basically sustainable. These are basically sustainable goals. I would like to affirm on that. Like these are sustainable goals, and we are looking for social goods. Floating plastic detection, like plastic pollution, is, we all know, like it has been. It has been just destroying the nature. So how can we use AI for detecting plastic things? So this is how AI is being exploited. So you have this satellite image of oceans. You give it to your AI model, and what it does, it predicts, okay, this is plastic bottles, fishing nets, plastic bags. The thing is, like, you may ask, like, you can just ask, you may say, like, okay, you could just ask 2-3 persons to just go out and just collect plastic, but it's not that simple. You have to build up a whole entire company for doing that. You need to buy ships, you need to buy boats, you need to buy equipment just to make sure, okay, is that a plastic or it's a fishing net? But the AI system just give them an image, it will do it for you. So it saves lots of money, it saves lots of resources. again, the another thing is flood forecasting. So what you see on the left is basically AI model predicting, forecasting floods for a particular region if that happens. So given that you have a flood forecasting, so you can plan accordingly. You can do better disaster management. So this is a flood forecasting, and what you see, this is from a river from Hyderabad. So it's a model. It's basically modeling if a flood happens, how it will spread in the entire region. So you could plan better. AI has been used for wildlife management as well. Like you could just keep track of your animals. So these are basically how you keep track of animals in some wildlife sanctuary or some forest. And this could help you for better management of your animals and keep track of poaching. And finally, the last thing is basically agriculture. The current hot topic in India is basically how could we exploit AI for agriculture. So what you see, so we all know this problem of feed detection. Like these are grasses which just grow up in your main crop fields and just destroys whole crop. So AI is being used for detecting these feeds, and once you know them, you could just cut them out. AI has also been used in quality assessment of the food, like to make sure it is ripe or it is raw. So basically, so how it is done basically, you have these drone cameras, you have these drones and they have these cameras, which is basically AI powered chips, those cameras, and it will just go over your farmlands and it will give you, okay, this fruit is good, this fruit is not, it is still too ripe, so better make sure you take it off your lot. So that's how these predictions are made. So I would say there are like immense opportunity and there are immense challenges for AI for sustainability and show like social good. So if I have to conclude with, so I would say like, I would just quoting it, say like AI should be exploited to its core for the betterment of the society. That's how I think like AI should be exploited. So yeah, that's it. Thank you.
Thank you, Tarun Krishna. We were listening a young intelligent scientist, which is not artificial at all. Artificial intelligence is really interesting, intriguing, and extent interfering. Very useful in so many ways, frightening at times. Thank you, Tarun. We can have a few minutes discussion, 2 or 3 minutes. We have time constraint, but the topic is very interesting. So we invite panelists for a brief discussion.
Yeah, Tarun, very interesting talk. Can I say something? Hello, am I audible? Can you hear me?
Your voice is clear, Dr. Azhar.
Okay, so Tarun, my question is, okay, artificial intelligence is a field, I think, that is expanding very rapidly, and it is, I mean, peeping into all parts of our life, I mean, daily to daily life. So one thing I want to ask, how much depth it has into how much depth in artificial intelligence has in predicting how advanced is the technology, basically, I would say, in predicting these COVID-19 based infection recently I have heard. Because a lot of app, I mean, they are suggesting which basically you enter whether you are positive or not. And based on that, every person who is non-infected, they would come to know that how far you are from other person who is infected. So which is very interesting because basically you can prepare yourself how you are exposed to the transmission of this coronavirus. Tarun, are you there?
I think there is some technical issue.
Okay. because this could be very interesting. In India, I think corona infection is rising rapidly. I mean, it's like a crazy rate. In single day, 16,000. And I was thinking those apps which are available in India, and if the government pull out a plan to make aware of everyone, so there will be very rapid information sharing and a kind of preparing the uninfected person to avoid such kind of a scenario. So artificial intelligence is deeply involved in this particular kind of scenario. So yeah, anyway,
Okay,
yeah, I
so
can discuss with him privately.
I can just tell you one thing from Indian side.
Uh-huh.
So already Indian government have deployed several apps, and one of the very important one is Arogya Setu.
Uh-huh.
About infected person around your area, about the person who are basically taking
video.
or so this information is available at the resolution of 500 meters.
Okay.
So this is a very interesting basically thing.
Okay. Yeah, yeah.
It is operational and it is in my basically phone. So I'm also using it at least twice a day. I can see it, how the situation is in my campus. I'm from Jawaharlal University. So now I can tell you that there are 4 people, and this information is correct because we also have official information who is infected and who is under treatment, quarantine, and all those things. So this app is working, and moreover, the global effort is also taken very nice shape. So there is a COVID desk from US side which can provide all kind of information. So for COVID, I think this will take 1 or 2 minutes. There is no dearth of information right now. Okay, in terms of looking at molecular biology, looking at disease biology, looking at, so whole world have done aggressive research. I have never seen this kind of research in last 20 years. Okay. So, basically there is no problem with the information now. Only thing we have to find out solutions.
Yeah,
whether it is at the prophylactic level or it is at the drug level. And drug also, AI is deployed, molecular biology is deployed, immunology is deployed, and then repurposing is also going on. And repurposing also, you won't imagine almost each and every target have been finished by AI, and already paper is published in Nature or all those places. So currently the scientific community, or we all, are suffering around the globe to have solution for COVID. So that credit goes to really international basically efforts and consortium research, that every information is on platform. You name anything, it is there. Okay, so this is what I do. Okay, I'm done. Thank you.
Yeah, thank you. Yeah. Tarun, very nice talk. Tarun, congratulations. Very nice talk.
Yeah, Tarun, it's very good. So you are taking up very right career at the right stage.
Yeah.
Yes, Rakesh, I'm ready. Yes.
Yeah. Please unmute.
Yes.
कॉल करिए बोलेंगे नहीं टाइम लगता है
First aid level.
Those slides are visible.
Dr. Rakesh, can I start with my presentation?
Yeah, the slides are ready.
Yeah,
you can proceed.
I have shared my slides.
Yeah.
Okay. So first and foremost, I would like to extend my very heartiest thanks to the organizers and especially to Dr. Rakesh Pandey for providing me this platform of a galaxy of academicians and intellectuals to discuss about my importance of my subject in the current scenario of environmental sciences. Basically, I am dealing with the bioinformatics, and the previous lecture was based on the artificial intelligence, was also very informative and very important in the current scenario. As Dr. Tarun and many of panelists have discussed about the importance of artificial intelligence, including our Indian, this app, Arogya Setu, and many more. So I can say the bioinformatics has the pivotal association with all kind of these advanced branches. So not only in the healthcare, not only in the education, but bioinformatics has also put its many effort in the area of environmental management and ecological management and environmental management. So here I would like to share some of the important points. Basically, in the current time, all of us are seeing that data management is a big issue. We are COVID patients data, control data, and a number of data. The bioinformatics is the branch. The first and foremost important thing is the data management, data information, and data compilation and curation. So when we talk this field in concern to environmental management, definitely we can coin a new term by the name of eco-informatics. As actually bioinformatics is the kind of protocol. We can't say it is a kind of external discipline. It is a kind of protocol that can be opted in the healthcare by the computer-aided drug designing, eco-informatics, neuroinformatics, immunoinformatics, whatever, wherever you want to apply, you can apply this field as a protocol. So when we are applying this field for the management of environment, for the management of data in the terms of ecological management, we can coin the term eco-informatics. So by self-explaining, it is an interdisciplinary framework for the management and synthesis of ecological data by advanced computational technology. The management of ecological data aims at facilitating data standardization, retrieval, and sharing by means of metadata. Actually, in the current time, data and metadata concept, whatever the data we are regenerating by the existing data comes under the metadata. We can, and very much artificial intelligence is based on this metadata. So it is object-oriented programming also. It involves object-oriented programming. Sorry. Analysis and synthesis of ecological data aims at elucidating principle of information, processing, structuring, and functioning of ecosystem. Actually, these kind of protocols are being monitored, are being opted in different fields. And when we are foremost, actually, basically, many of my work have been done in the area of environmental biology, that I will be discussing later on, that gives a very good and strength towards the eco-informatics. In the current time, forecasting of ecosystem, forecasting meteorological information, they all are being developed by the computational and hybrid models. So these models are very importantly playing, as the term is self-explaining, bio means the biological information and its amalgamation in the information technology. So the both when combines, it gives the terms bioinformatics. Exactly in the terms of ecological data, when it's interpretation, analysis, orientation, then it gives the eco-informatics. So different kind of hybrid model systems are being made to go with the environmental biological management. So basically you can see this diagram. Previously we were facing the problem of in vivo experiment, then we shifted towards the in vitro experiment. Then in the current scenario, we have shifted towards the cellular level, then to the in silico experiment. In the current time, most of the people are doing with the COVID research or interaction of COVID proteins with the different molecules, with the different drugs in the terms of repurposing by using these kind of computational tools. So from gene to protein to cell to organ to organism. So exactly when we want to know the interaction of these biological molecules in relation to the environmental science, we have plethora of data by the microarray data, by the genomic sequence data, by the physiological data, metabolomic data, metagenomic data. So these kind of everything is want to be interact with the environment. So ecological and environment management is no doubt a very integral part of bioinformatics. Like suppose you want to know the effect of lead in relation to fish, aquatic fauna. You want to know about the lead interaction with the any plant system. So all kind of interaction could be visualized, could be monitored by different kind of computational tools which are freely available and give the pivotal results of your research. So based on pivotal results, we can go for further validation of our experimental analysis. So no doubt bioinformatics, it reduces the cost as well as it reduces the time of experimental evidence for the scientist or for the biologist. So these kind of interaction like the autotroph producers, heterotrophic consumers, then decomposers, how these things are being channelized in the system. They all are these kind of all interactions. First of all, the main important thing is the database management. So all information has to be maintained in different kind of databases. Like here I have mentioned in the right corner of my slide, like genomic sequence data, then phenotypic data, then community data. In the current time, we are very much focusing towards the community data. Okay, so if there will be no discipline for maintaining these kind of data, then how the further researchers could take over the opportunity to go in the more deep, to go in the more application part. Then environmental data, then climatic data, then genotypes of population, structure, functioning of ecosystem. So these all kind of networking, these are all kind of interactions give the scope of our future informatics. And no doubt the predictions are very authenticated in the current time as our tools are highly refined. If you go to design a drug, if you go to repurpose a drug, if you go to design a new pesticide, a herbal pesticide, if you want to go to design a new predictive tool, different pipelines are available. Even in the current time, I think most of us are aware about the high throughput data analysis. So next generation information, different platform like iTorrent, Nanopore, Roche 450, they all are giving the high analysis of data, high throughput generation of data, and this data gives the very good interaction after the analysis of our results, either in the terms of environmental interaction or in the terms of health or in the terms of community based studies. So these kind of approaches are currently in being used. They are the different sources from where the data is being generated. Most of the people are having in their mind that bioinformatics means database. Not only database. Database, management of database is the first and foremost important part, because if you are going to design any tool, any software, then your previous information in the terms of database is required. Like in the current time, near about 7,000 genomic sequences are being entered in the NCBI. Near so many structures of COVID-related proteins are in Protein Data Bank, PDB, RCSB. And by these sequences, by these protein structures, system biologists, computational biologists they are predicting the different kind of interactions of this COVID. They are trying to finding out the new vaccines target. They are trying to find out the phylogenetic relationship that this virus belongs to which category, and whatever the innovative ideas that can be attained, that can be taken out by these information. So database is the first and foremost important step for maintaining, for doing any kind of experiment. You need information. Exactly in the terms of bioinformatics, if you are going to deal with any kind of experiment, you need a basic data. Data is something like the building blocks of a child game. And by these building blocks, you can make a building, you can make a bridge, you can make a train, and whatsoever. It depends on your own innovation and your curiosity. So the basic data sources of data in the case of environmental management comes by the citizen science, by the federal, state, and the private data repositories. Then by the indirect sampling and passive surveillances are also available. Academic data is also available, that what kind of courses are being offered, and n number of data. They give the different amalgamation of data source. Then we build the data sets. Data set building depends on the data quality, privacy of concern, because I don't want to share my data. You will be happy to know that our IGIB has done a number of sequencing parameters with the COVID genome, but still, due to certain privacy, the data is not being publicly available. They are trying to train their data, they are trying to validate their data, then it will come up to the public domain. Then flexibility should, size of data sets, then temporal and spatial scales of data sets. All these all together makes the data building of a data set. Once your data set is being built, then there are 10 checkpoint, the checklist, 10-point checklist is available for the ecoinformatics studies. Identify your research questions, what you're going to add, what is your basic problem. Then identify the primary pre-existing data, then seek out complementary data streams, then access data privacy issues. If I'm not able to disclose my data, you cannot use my data. So privacy, copyright issues are also comes under these privacy issues. Then understand the data set and evaluate potential basis. Then design data management and workflows. Workflow depend upon your problem, upon your issues, what kind of problem you want to resolve. Accordingly, you can draw your workflows. Suppose if I'm trying to identifying the vaccine candidate from the high throughput data of COVID, yeah, XYZ, any of the disease, yeah, for any plant, yeah, for any test. So I have to design my own objectives. Then accordingly, I have to set over my own pipelines. Then I have to take over the tools accordingly. Then I will analyze, yes, now I will fit this data here to get this information. So these workflows depends on your problem, upon your own issues. Then clarify, define response, explanatory variable, and evaluate potential confounding influences of your results. Then select an appropriate framework, then conclusion and inference, and then integrate finding with other studies. Whatever you have found it, you have to integrate with some of the relevant studies. So this is the ecological informatics versus ecological modeling. In the current time, people are dealing with the artificial intelligence. They are doing with the ecological modeling, differential equations, thermodynamics, multivariate statistics by using the heuristic approaches. And ecological informatics are also, and all these things give to direct a hybrid approach. When all these mix, then it gives the hybrid approach towards our study. So there are certain very important, very informative websites are available, which are dealing with the management of these kind of data. Some are doing the freely available. Some are having some charges. So I have listed some online resources, those who are freely available, like ecoinformatics.org. Here you can get the information about your problem. You can get the information that you are trying to deal with this issue, what kind of protocols you have to use, what kind of tool you have to use. Then there are certain tools that I have listed over here, which are very importantly being used by the by the biologist, by the environmental biologist, like so analytical and modeling tools. The analysis and modeling of data is one of the core activities of ecological science. Here I would like to just take 2 minutes. Like suppose you have identified a very important protein of your test and you want to design a herbal or a natural or a synthetic biopesticide or something commercial pesticide. So, and you know the this protein is very important and you have to target this protein for stopping its activity as a pest. So you need the 3D structure of that protein. For designing of that 3D structures of protein, either you have to isolate that protein, characterize this protein, and go for the crystallography or NMR, or there are computational modeling tools are available through which you have to take the sequence. Most of the sequences are in the current time available in sequence database. UniProt is one of the very important database of protein sequence, either of plant, animal, human. So from there you can take only the primary sequence, and with this sequence you can model the tertiary structure of your target protein, and then with that tertiary, because protein is functionality, and that with this tertiary structure you can proceed up to the designing of your biopesticide or commercial pesticide. So these are the certain very important modeling tools are available in the concern of homology modeling. And exactly there are variety of the shelf software packages which are existing for analysis and modeling of your data. This is the structure modeling that I have discussed regarding the homology modeling, and these are the analysis and modeling, because when we are dealing with huge amount of data, we have to do some statistical analysis as a biologist, as a scientist, and because without the statistical analysis, there is no significance of our study. So there are certain tools like Kepler. R is one of the very commonly used package in the current time, driven by Ross and Ross, and it is many of the packages are freely available. And most of my one of my student, she has developed her own protocol and accepted in NCBI and all, and I am in the process of being copywriting. And by using the R and all for analysis of gene expression, differential expression of genes. So by using this protocol, you can identify differentially expressed gene in human, in plant, in animal, in any of the ecological system. So these kind of protocol are being developed. So there are some metadata specification, AML, NBII, FGDC. So from where the true sense information can be taken in the context of environmental biology. As Dr. Rakesh said, this conference is very much concerned with the environmental sustainability. So most of the time I am focusing on the databases, on the tools which are used in this concerned area. So there are the certain data repositories where you can submit your own data. Suppose you have identified a very good information, yeah, you have designed a good data, yeah, you have designed a good structure or something like that. So there are certain very good repositories are also available in which by your own authorship, you can submit your information, and later on that information can be used by the different people like us on the basis of public demand. So there are certain data, GetBank, CLIMDB, HerbetNet. So vegetation plot database is the WedgeBank, centralized server to provide open access of long-term meteorological records. HerbNet are the collaborative effort by institutions in United States, Canada, and Mexico to establish a network between databases of herpetological collections. So these are the some listed database in reference to the ecological management, and many more are yet. The current research in ecological informatics focuses mainly on 4 major feature areas: understanding information, processing, and evolution in ecosystem. What kind of process is being taken? What kind of phylogenetic relationship is being established? It is orthologous, paralogous. What kind of things are this? Then we can go for the computational management of this ecological data, then computational analysis and synthesis of this data, that means the interpretation of this data. Then we can go for the hybrid modeling of your desired set of data. Here I'm sharing 1 or 2 of my work that I have done with my PhD scholars. Now they have already doing their very good in different states, different countries. This database was designed by one of my scholar, Dr. Anshul Tiwari. Currently he is in Harvard, and he was awarded with the gold medal in scientific conference. By using this data, suppose PestEye, a tool, online tool for pesticide degradation gene sequence data. Here we have entered the different gene of microbes that are found to be used for the pesticide degradation. And suppose you have identified a new gene, new sequence, and you want to know the similarity of your known sequence with the with how much this your sequence is found to be the pesticide degradable. So you can compare the information of your sequence by putting in this database, and it will give you results on the similarity index basis. So pivotal study was that in this database, I have taken near about 100 or 150 sequences of those those micros protein which are found to be important in pesticide degradation. So if our unknown nature of sequence are being compared with these nature of sequence by doing the alignment, there are many kind of alignment, multiple alignment, pairwise alignment. So by using these kind of alignment technique, this database will give me the results that how much percentage of my sequence is having the pesticide degradation capacity. So novel targets can be identified. New microorganisms can be used for these kind of pesticide degradation. So another work I have done with my scholar, I have pasted some posters, and this in silico identification and construction of microbial gene cluster associated with biodegradation of xenobiotic compound. Here I have identified the certain very important gene and made a microbial cluster, and the paper is published in very good journal. So in silico we have identified that yes, this gene is important, and altogether this cluster can be made, and once the cluster can be used for the expression analysis, it can give the synergistic effect for the xenobiotic compound degradation. So exactly I have identified some very good antiviral compound in the current time. This camphene extract of camphor is found to be the very good antiviral compound by using the in vitro, by using the in silico analysis, and after the in vitro validation, it came into the existence. The camphene is being shown a very good antiviral agent against MX protein of Bitarunda family virus. And because this barramundi is found to be the very important fish of cultivation, and mortality rate is very high because of the MX protein. So we tried to screen out certain herbal antiviral compound that could be eco-friendly with the system, with the culture, and even in the pond, and we can dump it so that the viral infection can be prophylactic or it could be protected. So this camphene is, and exactly in the same way we have identified the nimbolite. Nimbolite is very important constituent of neem, this Azadirachta indica, and found to be showing the, I have submitted one of my project also. So it is showing also very good efficacy with some protein of fish. So exactly in this way, we have also developed some databases by the terms of neuroallergens, that what are the allergy compound that are showing their interaction with the different proteins of our neurological biology. And then we have based the neuroallergen knowledge-based information system, and we have done virtual establishment of phytochemicals as a neuroprotectant against pesticide. So there are the certain things which are found to be important. One very important database is the Neurotox database. It is a neurotoxic and international conference we have presented, and it is showing the very good feedback. So unfortunately, in the current time, I am not being focused for the revised version of Neurotox, but later on I am in the process. So by these kind of work, we can proceed for different kind of our own analysis, small protocols for the analysis of environmental problems, environmental issues, given good good outcome, give good results. That could be eco-friendly, and that could be more cheap also. Okay, because our second India, we are in the agricultural land, so there is a huge demand and huge need in the current time to target, to find out the pesticide, because we cannot deny that we won't use pesticide. The huge amount of population is here, so we have to find out the remedy against that. Of course, we can limit the doses and all, but still there is a demand for pesticide degradation tools, for pesticide degradation, sequences, for clusters, and these are my students working in the different areas of bioinformatics. And she is Lady Garima, she is working in the area of agricultural bioinformatics. So I think, thank you once again, and paper is open for discussion. And thank you, Rakesh sir, for giving me again this opportunity. I hope I'm in time.
Thank you, Prachi, for delivering such a nice talk on information technology. As you rightly said, it is not only database, although data is very important, as we see in case of COVID-19. There are insufficient data to produce vaccine, effective vaccine. Once sufficient data is available, only we can produce the vaccine. You have very good papers also. Congratulations for that. And I am looking forward to collaborate with you on certain aspect, and I will talk regarding that.
That's my fortune, Dr. Rakesh, and I will. So actually, I was a little bit afraid of time, so I was a little bit fast because it is going to be 5 o'clock, and you have already instructed me to be in time. So, and I'm most thankful to all organizers, especially you, and you are most welcome. And it will be my proud privilege to have any kind of association with you, Dr. Rakesh, or any of the panelists. Thank you, sir.
Thank you. Thank you. So I would like to express my all of the panelists who spared so much time and participated throughout the conference, the morning session, as well as in this session, especially Dr. Sunil Kataria. Thank you very much, Dr. Rakeshwar Bandichor. He has been with us since morning. And Dr. R.C. Dubey, Ajay Vikram, Arun, and Prachi Srivastava, Dr. R.C. Dubey. So, lot of thanks, and hope we will have the similar discussion tomorrow. Very fruitful discussion, and tomorrow's session will start at 9:15. So till then, thank you very much. Have a nice day.
Thanks a lot, Dr. Pandey.